All right, so we're recording, so everybody be civil. All right, well, I want to welcome everybody to the 4th annual Translational and Transformative Informatics Symposium. We started this in 2017, the brainchild of Chen, and we were in a location called the Edge of Chaos, which is the 4th floor of the Lister Hill Library here at UAB. And now we are at the other edge, the other side of the Edge of Chaos, I think, switching to our virtual mode today. 2018, we had a keynote by Ying Zhu, searching for roots of cancer formation and development in the chemistry of cellular acid-base homeostasis. And the meeting had a strong cancer focus, but really spanned bioinformatics to clinical informatics. Last year, we were a pre-conference symposium for the annual conference of the Mid-South Computational Biology and Bioinformatics Society, or MC-BIOS. So that was a much bigger conference that we hosted here. The theme of our meeting was bioinformatics applications, and we focused on scientific findings and how they're enabled with bioinformatics and functional genomics techniques, and including genomics, proteomics, metabolomics, epigenomics, and various other applications of next-generation sequencing. So this year, the theme is informatics research to improve biomedical research. And the reason for that theme is that I think a lot of people think that informatics is not about research, it's just about writing computer programs to analyze other people's data. But it really is about developing new ways to represent, visualize, and think about biomedical information to help other people do their jobs, whether it's basic science, clinical research, or patient care. And we have testable hypotheses, and it's a, it is real science. So today's world of learning health systems, which we're going to hear about in a couple minutes, the information that we have to represent spans a spectrum from genetic sequences to human population factors of health and disease. And so we really need to embrace the notion of team science, where we bring in people with a range of expertise to work across that spectrum. And we doing translational research, also spanning the spectrum, not just from one step to another, but from basic science all the way up to the clinical setting and the public health setting. So anyway, to get started, I'm pleased to see the breadth of topics this year. And before introducing our keynote speaker, I need to do some thank yous because it's the only chance I'll get to do that. First, I need to thank the Scientific Program Committee, Chen, Alex Rosenberg, Zechen Chong, Amy Wang, Matt Wyatt, Wayne Leon, Tiago Colicchio, Min Gao, and Brittany Hussein, who have all contributed and especially have come together in the last 2 days where we decided that we really needed to switch this over to a virtual conference. And I would say it's gone pretty smoothly considering how quickly we had to do everything, although I shouldn't speak too soon. We'll see how it goes today, but we at least managed to get everybody organized to do this virtually. Second, I want to thank the Informatics Institute staff for handling all the logistics, especially Amanda Carballo, and she was assisted by Heather Watts, who is our communications director. They and the Scientific Program Committee have made a huge effort to make this conference move from the cancellation phase, which is where we were headed to a physical event, to its rebirth as a virtual one. It is virtual, but the lunches are very real. So if you are a speaker or poster presenter or a paid registrant, be sure to stop by at the Cudworth lobby to pick up your lunch if you're here on campus. It'll be between 11:30 and 1 p.m. And apparently we didn't cancel the coffee service this morning for the coffee break. So we have a huge amount of pastry and coffee outside the vault here on Tinsley Harrison 142. So again, come by. Just don't all come by at once. It would kind of defeat the purpose of this mass gathering. One other group I need to thank is the sponsors. So Center for Clinical Translational Sciences, CCTS, Department of Microbiology, the Comprehensive Arthritis, Musculoskeletal Bone and Autoimmunity Center, Department of Pathology, the Department of Cell Developmental and Integrated Biology, and the Department of Medicine. They've all made financial contributions as well as those intellectual contributions to the meeting. So thank you to all of them. So now my great pleasure to introduce Dr. Philip Payne. Philip got his Ph.D. from the Department of Biomedical Informatics at Columbia University when I was there, and his research there focused on the use of knowledge engineering and human-computer interaction design principles in order to improve the efficiency of multi-site clinical and translational research programs. After working at Ohio State University, he moved to Washington University as professor of computer science and engineering, where he's the founding director of the Institute for Informatics. And he has a number of broad research interests, including cognitive computing, machine learning-based approaches to discovering and analysis of biomolecular and clinical phenotypes, identification of precision diagnostic and therapeutic strategies in cancers and other clinical conditions, interventional approaches to the use of electronic health records in order to address modifiable risk factors for disease and enable patient-centered decision-making, and studying human factors and workflow issues surrounding the optimal use of healthcare information technology. Philip is widely known not just for his informatics research, but also as a thought leader. He has important publications on topics such as drug repurposing hypothesis generation, the role and impact of health information technology, and clinical applications of translational bioinformatics. He was elected a fellow of the American College of Medical Informatics, ACME, in 2013, and this is one of the highest honors you can get in the field in the US. So with that, let me turn it over to my good friend and co-author, Dr. Philip Payne, who's wondering why I didn't say anything sarcastic about him, and he will speak on the learning health record system. Philip.
Well, thank you, Jim, and I appreciate you reserving the potential for embarrassing stories, of which you have many that you could have shared. So it's a real pleasure to be here today. I'm glad that we're having this meeting virtually because I think it's really important, despite sort of the current state of affairs that we find ourselves in, that we continue the important work that we're all engaged in, which I think now more than ever is critical to the health and well-being of the communities that we serve as academic researchers, clinicians, and educators. So today what I want to talk about is a topic that I think is very relevant to sort of the broad focus of this meeting that Jim alluded to, which is this concept of how do we arrive at the behaviors that constitute a learning health system. And in particular, and as you can see from the title, I'm going to make an argument over the sort of next few slides during this presentation that the electronic health record is one of the most critical living laboratories in which this behavior can be exhibited. And that means we need to start thinking of it in that way as more than just a tool for medical legal documentation or billing purposes, but as truly a platform for innovation in terms of care delivery at the individual and population levels. So I will start with the obligatory conflicts and interests. I hope that you will just recognize this as an indicator of sort of the nature of our field of biomedical informatics. There tends to be a tremendous amount of cross-fertilization between industry, academia, and government, and I think that that's represented here. I often make the joke that having lived in the Midwest for a good amount of time now, that when I show this slide on one of the coasts, people look at me and say, that's it. And when I show this slide in the Midwest, people look at me in a very suspect manner. So I will defer to you to tell me how this resonates in the Southeast. So to begin with, as I just mentioned, the focus here is about how do we create a learning healthcare system? And I think we can all recognize that there's been a tremendous amount of dialogue nationally and internationally on this topic. And this has taken many forms, not only traditional academic discussions of the theoretical and practical frameworks that are necessary to build a learning healthcare system, but critical policy and strategic directions, for example, those that are set forth in the Institute of Medicine report that I've included an image of here on this slide. But I think the real takeaway here is that this is about a movement to start taking advantage of the myriad of data types that we generate in research, in care delivery, and at a population level, and start using that data to make smarter, more efficient, more impactful decisions for both patients and populations. And the simplest way that I often describe it to people is that we want to build a system where every time we interact with a patient, it's an opportunity to improve their care, their family's care, and their community's care, because we're going to capture all of the salient information, reason upon it, apply perhaps precision healthcare approaches at that individual level, but also create rapid feedback cycles so that the system is constantly improving. And I think this sort of stands in stark contrast to a more traditional sort of research to practice paradigm that we've seen over the last several decades that's more unidirectional, right? Research is translated into practice, and then there's rarely feedback from practice into the research arena. And that's really what we're trying to achieve by building the learning healthcare system. Perhaps one of my favorite examples of that is sort of visually this diagram by Jyoti Pathak and a variety of other colleagues, Abel Ko, Josh Denny, that appeared in JAMIA a few years ago, sort of creating sort of this idea of a virtuous cycle that is the learning healthcare system. And importantly, if you take nothing else away from this diagram, is the concept that the learning healthcare system involves sort of this constant cycle of designing care delivery, implementing it, evaluating it, taking those data, optimizing and feeding that back into continuous design processes, but also that it's not just localized, it also involves dissemination of new knowledge and practices to the broader community, as well as the intake of knowledge produced outside of healthcare delivery systems, such that we can learn not only locally, but regionally, nationally, or internationally. But this is really this concept of a virtuous cycle. And at a surface level, this is very appealing, but I think putting this into practice is quite challenging. And the thing that I want to emphasize here is that the individual constituent components of what's required to build a learning healthcare system are not particularly complex or sort of remarkable in and of themselves. They include things like, how do we actually instrument that care delivery or laboratory or other environments that are relevant so that we can capture high quality deep phenotypes across multiple scales, biomolecular phenotypes that we might produce through sequencing at the DNA or RNA levels, image-derived phenotypes, clinical phenotypes that may be captured in the EHR, behavioral data concerning the decisions and activities of our patients, not to mention the broader exposome, the environments in which they live, work, and play beyond the clinic and hospital. And if we do that, the next natural step in building this learning healthcare system is to figure out, how do we improve access to all of that data? The idea that there will only be a small number of individuals, data scientists, biomedical informaticians, health services researchers that have access to this full spectrum of data will likely preclude that virtuous cycle that I alluded to before. And then, if we achieve that level of performance, how do we leverage technology to close the evidence cycle? How do we make sure that we're bringing back that optimized evidence that's produced by this learning healthcare system in the right time, place, and format, delivered to the right person so that they can act upon it? And then, perhaps most importantly, and this goes to the point that Jim made earlier, as a science, and I think there's a science of informatics, not to mention there's really a science of learning healthcare systems writ large. How do we continuously measure and evaluate and improve using system science principles so that we're not, again, engaging in that unidirectional evidence generation concept? And one of the things I want to emphasize here, and I said it already, is that this is more than anything a behavior. It's not a physical thing. It's not just the EHR. It's not just the physical environment. It's not the people we have. It's all of those things working in concert, creating this behavior. Now, so far, I've cast a pretty positive image of what is possible if we build a learning healthcare system and why we might want to do that. Unfortunately, building the learning healthcare system is a bit more complicated than sort of those aspirational goals. And I want to talk just briefly about a few of the reasons why that's the case. So the first thing I want to talk about is some of the limitations of the tools that we have at the point of care, and in particular, electronic health records. And in this slide, I make reference to a recent report that was commissioned by the Kaiser Foundation with a very potentially hyperbolic name, Death by 1,000 Clicks. But the takeaway from this is that the human factors, the workflow integration, the contextual sort of situation of the EHR has fallen short of the expectations of our providers and patients for that matter. And that we've created systems that, while they may capture many of the data that's very salient to the care of patients, is not organized or presented or otherwise rendered actionable in a way that really fits the needs of our providers. And one of the things we've observed here at Washington University in a lot of our workflow and human factor studies is that the resounding feedback that we get from both patients and providers about the EHR is that the EHR, sort of as a physical manifestation in a computer or workstation in an exam or hospital room, has become a barrier between providers and patients, both physically and literally, that it is something that prevents sort of the effective practice of medicine, let alone the type of deeper thinking and interaction that our patients and providers expect. Another sort of critical issue that we are still seeking to tackle is how do we make sure that all of the vendor systems that exist in the clinical environment, in fact, allow us to move data between them into other environments that we might need for experimentation or innovation, and then back again. One of the terms that's often used in this context is information blocking. The idea that vendors are making it difficult, even with increasing adoption of standards that would allow for interoperability between various electronic health record systems, that vendors are making it difficult to actually achieve that degree of interoperability. If you've read some of the recent press around the newly sort of established interoperability rules from the Office of the National Coordinator, you would know that there's been a broad spectrum of responses from the vendor versus clinician versus research community about the need for these. And not surprisingly, the vendor community is not that enthusiastic about opening up the doors to sort of interoperability with sort of third-party applications with the EHR, even though that's really important if we're going to innovate on top of the EHR in this learning healthcare system. I have to tell people that you sort of can think of the current EHR environment as being analogous to the internet in the era of America Online or CompuServe, this idea of sort of walled gardens where everything you need is inside the platform and there's no reason to go outside. And I think the history of the internet and many other sectors of the economy would tell us that that is probably not a rational plan. We also know that there's quite a bit of sort of hype cycle surrounding the use of artificial intelligence for better decision-making in these learning healthcare systems where we have more data. Probably one of the most prominent examples is some of the recent failures that have been experienced around the IBM Watson platform, particularly in the cancer domain, where transportability of the decision support tools built using really what is high throughput machine learning at some leading cancer centers was found to be suboptimal when going into community or other settings. And I think this speaks to the issue that, again, this is more than an engineering problem. This is a science problem. How do you build the evidence base to show impact, utility, and safety of these AI tools? But all is not lost because we also have excellent examples. For example, the work that Google has done interpreting chest CTs that have shown that in some contexts, these AI tools can actually outperform human clinicians in basic diagnostic tasks, in this case, finding potential cancers in lung imaging. And so, you know, when properly studied and evaluated and deployed as an augmentation to human cognitive abilities, these tools become very powerful and lead to that continuous feedback cycle that I alluded to earlier. And then lastly, there are a lot of big unknowns around sort of how the vendor community is approaching this space. For example, you know, what happens when companies like Apple or Amazon or Google start going direct to consumers with apps and tools that are ubiquitous and based on sort of mobile and other technologies that will start capturing a lot of data that is highly relevant to this learning healthcare system. And I think the nature of this relationship between industry, academia, care providers, and other innovators remains somewhat ill-defined and something that we'll definitely have to tackle, because as we move to a healthcare system that's value-based, that truly is a learning healthcare system, that emphasizes health rather than sick care, we're going to care a lot about these data that are captured where people live, work, and play, and not when they're in the clinic or hospital. And we're going to have to figure out how to, again, interoperate across these various platforms, much like I was alluding to earlier around the EHR. So how do we move forward given sort of the promise of the learning healthcare system and some of those challenges that I just talked about? Well, I'm going to take a brief moment and take a little bit of a philosophical soapbox. I think this will be familiar to Jim, but I actually think there's a third path that we need to consider. And, you know, traditionally when we think about research and innovation, especially in the health and life sciences, we tend to create this dichotomous division between basic science, where we come up with new methods and theories, and applied science, where we find out if they work in the real world. And we talk about translational science as a mechanism to move that knowledge from sort of that basic quadrant into the applied quadrant. But again, it's somewhat unidirectional, even in that modern sort of interpretation. But if you've ever had a chance to read this book by Donald Stokes, Pasteur's Quadrant, which tells the story of Louis Pasteur and his groundbreaking work, he describes a third path: use-inspired basic research, which is really rapid iteration between basic and applied science that's happening simultaneously, that you're innovating in terms of theories and methods, but you're actually applying and studying at the same time, and then providing those feedback cycles. I think this is emblematic of the learning healthcare system, but it's also emblematic of the critical role of informatics and data science in this context. And the idea here is there's not this unidirectional handoff, but rather the two are happening in parallel. And so that third formulation, as I just alluded to, really does stand in stark contrast to some of the sort of foundational principles that our modern healthcare research and practice enterprise are built on. If you're familiar with Vannevar Bush's report, Science, the Endless Frontier, he really argued for this dichotomy between basic and applied science, and it's made up much of how we approach this, whether it be in the academy, NIH, how we fund science, or how we train people. And again, I would argue that while that might have been rational at the time in which the report was written, with the computational and data capabilities that we have today, the ability to create, at least in the health and life sciences, these learning healthcare systems, if we are effective in doing so, that we can pursue that third quadrant that I talked about earlier from Pasteur's quadrant, which is use-inspired basic research, where we eliminate that dichotomy between basic and applied science. And I think that this is the critical issue when we think about how we position research, innovation, care, and education in the health and life sciences moving forward to achieve that learning healthcare system behavior, to really embrace this idea of use-inspired basic research rather than the artificial dichotomy that is more historic in nature between basic and applied science. So, if that is the case, what is the laboratory in which this use-inspired basic research is going to happen? How are we going to achieve this behavior of a learning healthcare system? Well, I'm going to argue now in the modern era that the electronic health record is truly the living laboratory for use-inspired basic research that will move the needle in achieving the vision of a learning healthcare system. EHRs have a true benefit of being real-time and patient-centered and ubiquitous in the clinical environment, in which much of the clinical and other types of phenotyping data that I talked about before are being collected and managed. And they are also built purposely to share information with other providers and organizations. Despite some of those concerns about information blocking as compared to a paper record, the EHR is the ideal tool when using standards that are available to us today to move data between all the different stakeholders that need to integrate, analyze, and act upon that data. And so when you look at this in sort of en masse, you realize that you have a ubiquitous patient-centered tool that's present in many, if not all of the places where we're capturing critical data that will feed into a learning healthcare system, that they can be extended in order to capture data beyond what we need for medical, legal, or billing purposes, and we simply have to do so intentionally. And they are built to share data across various stakeholders in this multidisciplinary world of what I'm arguing for as a learning healthcare system. And so therefore, I would say the EHR is the perfect living laboratory in which to conduct this work. As I already said, there's essential features. It's proximal to all the key stakeholders that we need to engage in order to achieve this vision. It can collect multiple modalities of data, from structured to unstructured, to imaging, to biomolecular phenotyping, to even sensing data now with some of our sort of common vendor EHRs. We can customize it. We may not always customize it the way that we want to, but the capability exists to do so. They are built to scale. And again, as I said, they also have this potential for interoperability and data sharing. And so again, the EHR really becomes that building block for deep phenotyping and, as I'll show you in a few moments, digital health interventions that allow us to close that evidence cycle in the learning healthcare system. Now, you may be saying, Philip, you know, that sounds really great, but we all know that the EHR is really hard to work with, right? That there are fundamental issues that we can't overcome right now that prevent us from doing what you just said. And I'm going to say that there are four main arguments right now about why the EHR is not an effective platform in this context. One is that there are a myriad of workflow and human factors issues that impact data quality and completeness. The second is that data privacy and confidentiality concerns prevent sharing of data, even if we technically can do so. The third is that the vendors make it difficult for us to do this type of customization. And the fourth is that sort of the interaction between care delivery, innovation, and IT delivery is often fraught with sort of friction. And I want to explore each one of those because I would argue many of these are more myth than fact. And the question is, how do we overcome sort of that prevailing wisdom and really get down to doing the critical work of building this learning healthcare system? So, and I'm sort of tipping my hat in terms of my thinking about this, I want to start with myth number one, that workflow and human factors issues cannot be adequately addressed, and therefore the data in the EHR is not useful for this purpose that I just described to you. So I would argue that we have a tremendous toolbox in the informatics community that will allow us to overcome workflow and human factors issues. And here you see sort of a listing of the various types of methods that have been developed and verified and validated by informatics researchers over the last several decades that allow us to study the use of healthcare information technology in context, identify opportunities for optimization, and then implement them to improve workflow integration and human factors. In fact, I think the only reason why we are not achieving sort of the benefit of these types of methods is that we're not putting them into use in sort of the operational environment. They've been largely the purview of the research laboratory and not operations. And this is about a somewhat false dichotomy between the informatics research arena and the delivery of these technologies in the enterprise setting. And I want to take a moment and focus on one of these methods, which is time and motion studies. So if you're not familiar, there's a vast amount of literature concerned with time and motion studies that tells us about how we can go into clinical environments and monitor the activities, the resource utilization, and time utilization of individuals interacting with the physical environment, including technology, and understand where there are opportunities to improve that, where we see sort of inefficiencies in terms of the use of those resources, interruptions in the environment, or other human factors problems. And one of the most common forms of time and motion studies are what we call continuous observation time and motion studies, in which we actually see sort of observers shadowing individuals in the clinical environment and tracking all of this data. Now, one of the problems with conducting time and motion studies is that they actually can be quite labor intensive and difficult to conduct, especially in multiple environments with multiple observers. One of the projects that are underway in my laboratory for the last almost decade now, working with colleagues the globe, has development of a scalable internet mobile-based application called TimeCat, which is a motion study platform. Now, observers to allow the performance of the observer and analyze them en masse. And we actually have a global user community of people from throughout the world that are using this tool today. I'll tell you as an aside, one of my favorite aspects of the project is we have people well outside of the healthcare domain, including professional sports teams, people in business and manufacturing, other places using, and it's available freely and not for profit. And I'll provide a URL in a moment. But see on the right side, some snapshots, for example, of the tablet-based version allows for multiple to observe analytics, including reliability assessments, as well as sort of quality assessment of the overall model. And so this is a powerful tool to allow us to conduct time and motion studies. And it's been used in lots of different places. And I'm going to cite a few articles, many of which are not my own work. The impact of task switching on nursing workflow in critical care environments. And this is actually with my colleague Po-Yin Yen, who's one of the other key inventors of the TimeCat platform, looking at the sort of operational impact of digital health technologies in clinical environments. Again, looking at sort of audit trails in the EHR in conjunction with workflow studies to understand whether or not the audit trails in the EHR tell us something useful about how people are interacting with these technologies. Looking at the role of workflow in capturing data at the point of care for disease registries, in this case in the rheumatology domain. And then also looking at the impact of digital health interventions on sort of healthcare quality, safety, and cost. And these are just all examples of studies over the last several years that have used the TimeCat tool to identify opportunities to optimize the use of healthcare information technology from everything from nursing workflow to purely research applications with lots of stops in between. So I would argue this is an example of a tool ready today that can be taken off the shelf, that can be used to optimize clinical workflow and the use of health information technology so we can trust and know that we're getting high quality data from the point of care. Second myth, data privacy and confidentiality concerns make it too difficult to extract data from the EHR. Basically, the rules and regulations that we live under preclude the use of this data, even if we can ensure that it's high quality and appropriate to the task. So again, I would argue that there are many different tools that exist in our informatics and data science toolbox that allow us to responsibly analyze clinical data while respecting privacy and confidentiality. And again, I've listed a variety of those methods here, but I'm going to dig into one of them, which is this concept of producing synthetic clinical data as a way of sort of protecting the privacy and confidentiality of patients while simultaneously ensuring that the data is high fidelity and useful for the various analytical tasks at hand. So at our institution, we have a unique partnership with an Israeli startup company called MD Clone that's actually built a very interesting synthetic data engine. And I'll show you in a moment using an information graphic how this works. But the idea here is this is a self-service engine that produces synthetic clinical data on demand based upon the contents of our electronic health record system, in our case, the EHR platform that we use for all 15 of our hospitals, as well as our 2 physician practice groups for about 6.5 million patients, or 12 million historically. All of that data is available in this MD Clone environment. And one of the things that we've done with this company is truly evaluate the performance of this synthetic data in comparison to sort of real world clinical data, right? The raw data from which it was derived. So we can find out, is this data sufficiently comparable that it's suitable for these analytical tasks, right? Is this synthetic derivative something that is high fidelity and represents the real clinical data without the risk of breaching the privacy and confidentiality of the patients from whom it's been derived? So how does this work? So this is the cheat sheet version of how this data synthesis process works. It begins with having all that data from the EHR in a data lake, in this case, an actual identified copy of the data where you can't see the individual data values, but you know sort of the broad contours of the patient population. And you can select a cohort of patients based on whatever condition or sort of outcome of interest you have. You then basically walk through a feature selection process in which you say, these are the data elements that I'd like to extract from the EHR for those individuals. And then the system will produce on demand in a matter of a few minutes a synthetic data derivative of those features for that population, provide summary analyses, and allow you to export it and actually analyze it. So how does that work in terms of protecting the privacy and confidentiality of the patient? So on the bottom half of this slide, what you see is sort of a series of 2-dimensional distributions, right? And you can think of each one of those distributions as being sort of a feature, right? It could be a laboratory value, could be age, it could be some other measurement of interest. Each patient has a measurement that sits within that feature distribution. And if you were to fully populate those distributions with all patients, you would actually sort of cover the full spectrum of those possible values given sort of where they sit in that distribution space. And thus the patients become a vector that passes through this multiple dimension sort of feature space as you see here, right? So here we have 2 different patients. They have a measurement in each one of these feature distributions, and we can create a vector that represents all of their measurements across time. What the MD Clone platform does is actually create new vectors where the individual measurements are not statistically different from a significant standpoint than the initial measurements, but they don't directly overlap. And what you actually do is produce a set of new synthetic vectors that satisfy the overall feature space distribution, that track with those real patient vectors, but do not cross over. So there's no mutual information shared between the original patient and these new vectors. The covariance and the other statistical features of that information space are remaining intact. So one of the places where we've used this, and it's actually quite interesting in the current state of affairs, is actually in sepsis risk prediction, a popular topic. And we really focused on a fundamental issue in sepsis risk prediction, which was the temporal dimensions of prediction. Could we move the prediction timeline back 6 to 8 hours before sort of standard clinical indications of sepsis for patients in general ward units in our hospital, such that our critical care teams could go see those patients before they may need to be admitted to the ICU? And again, we did a head-to-head comparison of real data extracted from the EHR and synthetic data to see if they were comparable. So this is the basic sort of concept, right? Like as I said before, starting with time of sepsis, moving back 6 hours, looking at all data captured within a 24-hour window prior to that 6-hour time, sometimes going all the way back to admission, using a variety of features. And while this is a relatively short bulleted list of 5 bullets, this represents close to 2,000 features extracted from the EHR. And then applying relatively straightforward prediction models like logistic regression, support vector machines, as well as clustering analysis to find out, could we achieve sufficient accuracy to predict who is going to become septic before they satisfy those standard criteria. In our case, a combination of the Sears criteria as well as suspicion of infection indicated either by ordering cultures and then starting antibiotics post result, or ordering cultures and starting antibiotics before the results came back because of the timing issues. Now, this is a diagram just as a sidebar that I describe as sort of the funnel of despair, because this happens in almost every predictive analytics project. If you start at the top and you look at sort of, here's all my patient encounters, isn't this great? I have 61,000 patient encounters. By the time we get down sort of this funnel and we find unique patients, those that were able to be diagnosed with having sepsis because they met the SEERS criteria plus suspicion of infection, and they developed sepsis more than 24 hours after admission, so we know they didn't come into the emergency room with sepsis, and had sufficient vital sign documentation. Again, this goes to data completeness. We go from 61,000 to just 377 patients. And I would argue almost every predictive analytics project has a funnel that looks something like this, which speaks to the challenges of sort of building adequate data sets given the way we presently use the EHR. But this is the important slide, and I could spend the rest of this talk, and I would love to, but I will not, as exciting as I would be about talking about this. But this is the really important slide, because what this shows is our best performing predictive model for identifying sepsis 6 hours before standard diagnostic criteria. You can see in the test set that our accuracy sits right around 84 to 85%, right? So that way we can find those patients, a pretty good performance. This is a random forest-based model. And the really important thing is if you look at the left-hand column, that is what happens when we train the machine learning model using our real data and then test it on real data. So the traditional way of doing things, just using data extracted from the EHR. On the right-hand side, we show the performance of the model when we train it on synthetic data produced using the MD clone platform and then test it using the real clinical data. And what this actually tells us is there's hardly any difference in the performance between the two, which means we can train machine learning algorithms on this synthetic data where we have no re-identification risk for our patients. It means we can have partners, we can engage students, it's no longer human subjects data research, and still know that when we go to run that machine learning algorithm for an active clinical decision support purpose within our clinical environment, that we have a sufficiently high fidelity model that can act upon our clinical data. And I think this is a pretty exciting finding, and we're continuing to press the envelope. We're doing everything from geospatial reasoning, traditional statistics to machine learning like this using this engine. Myth number 3, vendor constraints make it difficult to build and deploy interventions. So I want to close that evidence cycle. I want to bring back these interventions at the point of care based on all the cool machine learning and other analytics that I can do. Can I actually do that in the EHR environment? Well, again, we have a lot of tools and methods at our disposal right now that can be used to build sort of apps or other interventions that interoperate with the EHR. I'm sure many of you heard about FHIR and other vendor-specific APIs. We have common data models, data interchange standards, and then sort of combined approaches across all of those areas. And I'm going to show you an example of exactly what I mean. So this is a project called the SPHERE project. It involves a clinical decision support tool looking at modifiable behavioral risk factors for cardiovascular disease in at-risk populations. If nothing else, this slide satisfies the criteria that informaticians produce slides with boxes and lines, and then engineers actually have to turn that into working software. But the idea here is that we're taking historical data extracted from a data warehouse, combined with transactional data from the EHR during a clinical encounter, putting that together with a reasoner, in this case, that applies the American Heart Association Simple 7 criteria for modifiable behavioral risk factors for that data, and returning it at the point of care in a workflow-aware manner that enables patient-centered and shared decision-making around those modifiable behavioral risk factors. Now, thanks to our EHR vendor, I can't actually show you a real picture of our EHR, so you'll have to imagine that for a minute, but this is sort of a mock EHR that we use to value. But imagine here we're in an environment where we have a set of results for a patient, we're during the encounter, and what you see on the right-hand side is actually what the SPHERE tool looks like in use. In our case, this actually triggers within the Epic electronic health record environment inside the Epic environment using their Open Epic Web APIs. So let me blow this up a little bit. The idea is basically we pull all the data historically and transactionally from the encounter. We show how that data aligns with those American Heart Association Simple 7 criteria. We calculate a aggregate cardiovascular health score, but we also show the individual components color-coded. And I want to draw your attention to the fact that for each one of those components, we actually have slider bars or other interactive components, because the important dimension here is that once this is displayed, the provider can show it to the patient and say, let's see what your score looks like if you get your hemoglobin A1C down, or your blood pressure, or you stop smoking. So it allows for sort of the if-then experimentation of what are the best modifiable measures that you could take, which is different than I'm sure what many of us have experienced when you go to your primary care physician. They say, you should lose some weight and get your blood pressure down. It's like a generic comment and then nobody does anything. This is much more concrete. We also have the opportunity to collect over mobile from the patient's additional sort of dietary and activity data that can be used to augment this. And one of the things that we've learned in this study, we used a user-centered design process, is that engagement of the clinicians and workflow integration. And this is my illicit picture of the Epic EHR, so I'll deny that this was included. But here you actually see it displayed in line with Epic during an encounter for a patient, and it's triggered via BPA. It tells you that this report is available and then triggers the tool. One of the things we've learned is it's just powerful because it actually allows us to sort of organize and deliver the information in an actionable format. And the important takeaway is that we found in a practice randomized study using a step wedge design across multiple safety net clinics, this was actually done in Ohio at the time, that we can show statistically significant improvements in both body mass index as well as hemoglobin A1C for those patients that received this digital intervention versus those that did not. And actually an effect that is not dissimilar to what would have happened if we would have prescribed new medications for glucose control or sort of medically supervised weight loss to these patients. And so the takeaway is that the digital health intervention actually changed clinical outcomes for these patients, and it was built inside of the EHR. And I think that's an important lesson, and we're doing this now in cancer survivorship, we're doing this in stroke, we're doing this now in behavioral medicine, a variety of other areas, and we see similar results in many of our studies. An interesting sidebar, this initial project was funded by Pfizer Corporation through an unrestricted grant. They were not as thrilled with our results because they believed that what would happen is we would demonstrate the use of the decision support tool would increase prescriptions for drugs for cholesterol and blood glucose and blood pressure control, which in fact it did not. It actually reduced new prescriptions by impacting modifiable behavioral risks in an effective way. But nonetheless, I think this is an important lesson. We can use these standards, we can build interventions, and those interventions can have impact on patients in terms of clinical outcomes, in this case, modifiable behavioral risk factors. And I think that's a powerful argument for the art of the possible and closing the evidence generation cycle. And if you want to learn more about it, and I'll make sure that a copy of these slides are available to you, we have a number of publications that sort of outline these clinical study results, in this case, in the SPHERE project, and we have a couple of follow-on projects, but you can take a look and learn more about the results of that study. And then lastly, and just briefly, I want to talk about myth number 4. IT organizations create too many roadblocks to doing this work, right? The division between innovation and informatics, sort of research, clinical care, and IT precludes all of the things that I was just talking about. Well, I would argue that there actually are good models that many of us understand from high-performing organizations that allow for us to do all the things I talked about. And there's not really sort of any magic bullet here, but it involves things like being very clear on the roles, responsibilities, and capabilities, and achieving sort of shared leadership across applied clinical informatics and operational IT. And making sure that we actually have formal processes for evaluating and deploying these interventions at scale that span our informatics and data science researchers and the IT organization, so we can build these rapid learning systems. And I would particularly emphasize that third bullet because part of it is creating the environments in which that happens. Here at WashU and with our partners at BJC, one of our biggest activities around our Epic EHR right now is creating an innovation environment, a parallel environment to our EHR where we can experiment, scale, and find out how these interventions work, both with external companies and internally developed innovations, and actually have a clear change management and deployment process where we can push those interventions into the clinical enterprise when they are ready to scale. And that's a really important factor, right? Because it's different to say it works in my lab versus I'm going to deploy it to 15 hospitals and 2 physician practice groups. That's a different scale of intervention, and we need to be able to do that, but the process is not the same as getting something to work in a laboratory setting. So part of that is understanding, like I said, roles and responsibilities. And I would just emphasize that, informatics is not computer science, nor is it information technology, but there are fuzzy boundaries between the fields. And so it's really important that we learn how to navigate those fuzzy boundaries and build multidisciplinary teams that can move sort of the needle across basic science to applications. Now, I would point out back to my argument about sort of that third way, that use-inspired basic research, that informatics in sort of the middle point of this spectrum most definitely fits in that quadrant because of the nature of its ability to conduct both basic and applied science and make that sort of multidisciplinary and translational activity happen. And I think one of the things we have to talk about in the informatics community more frequently is informatics translation, which is what I'm alluding to. Not dissimilar when we talk about translating new molecules or new diagnostics from the lab into clinical trials and then into general clinical practice. We need to do exactly the same thing around these digital health interventions and think about the translational blocks and how we build the systems to make that happen, working with our colleagues in both the computational and data sciences as well as in the IT world. And this is actually a model that Chris Longhurst and I have worked on. Chris is the CIO at UCSD, and we have a publication that's in the final stages of sort of review on this, thinking about what is the synergistic relationship between sort of healthcare IT and really research informatics in that translational juncture. And I won't read all of the things here, but what the key takeaway is that there is a synergistic cycle, much like the broader learning healthcare system, that starts with sort of the research arena, involves verification and validation of these new evidence, scaling and delivery in sort of real world settings, and then optimization and measurement of impact. And much of this needs to be shared across the IT organizations and the research informaticians if we're going to achieve sort of this shared vision of a rapid learning system. And perhaps one of the most important lessons, and Chris has done a phenomenal job at demonstrating this at UC San Diego, is having shared leadership and cross-pollination of these ideas between the informatics community and the IT organizations. So that knowledge base and that sort of shared understanding is made sort of part of the fabric of the organization. And he's demonstrated it, as has many other organizations. I'd also point out in this translational cycle that we need to recognize it's not just about the people that build software or analyze data, that perhaps one of the most important roles is what I like to point out as being the data translator. And this is actually from a really excellent article in Harvard Business Review, if you've not already read it. I think the data translator may be the most important person in the learning healthcare system, because they're the people that understand the problems in the real world, can help prioritize them, help us understand the data that needs to be analyzed, and how we will evaluate whether or not these interventions are in fact successful. And oftentimes in these projects, especially in the learning healthcare system, these are physicians or other clinicians that partner with informaticians, IT professionals, and computational scientists to achieve this shared goal. So to wrap up, just a few final thoughts. All of the things that I've talked about have been made possible because of this incredibly rapid evolution in the health and life sciences toward the adoption of computation and digital health technologies. And it's easy to forget that this has happened in a matter of a few short decades compared to many other fields, right? If you go back only 10, maybe 20 years, the state of the art in data management in many of the settings that I'm talking about would have been paper. Right? And therefore, we are still learning how to use these technologies in effective ways at the point of care, in research, or at a population level. We should not lose sight of the fact that this is a critical dimension of all the work I described, that we're still learning how to thrive and be successful in this new world order. And it has been made possible in large part because of the incredible advances in computation that are available to us today. This is something that sort of, if I was going to be a little bit more geeky, I would spend more time on. But it is amazing if you think that in the 1900s, we were talking about the state of the art being mechanical computers that were used to prove basic statistical hypotheses, like Bayes' theorem in the case of Babbage's analytical engine. To today, we have technologies like the image you see in the lower right corner, which are quantum computers, computers that can operate in multiple dimensions, and perhaps even more interesting because of the fundamental physics that underlie the way in which they work. If we try to observe them in action, we would actually perturb them such that they wouldn't compute as expected, right? So we've gone in 120 years from mechanical computers to computers that compute in multiple dimensions. That is a tremendous amount of advancement. And I think one of the questions we always have to ask is, how do we harness all this power to in fact improve the quality and safety of care for individuals and populations? Again, going back to this principle of a learning healthcare system. But if we get it right, what do we achieve? All the things that I talked about. Well, we would leverage all of those advances in computing to create a smarter, more precise healthcare system where every time we interact with a patient, it's an opportunity to learn, and as I said before, improve their care, their family's care, their community's care. That we would actually have these rapid evidence generation and delivery cycles that are sort of incumbent to precision medicine, and that we would use all of the big data technologies really at a systems level that are available to us to pursue that type of use-inspired basic research that I talked about before, that rapid translation between basic and applied science that drives all of the things that I just alluded to. And if we did that, and I think we are capable of doing so, we would truly have an integrated and high-performing healthcare research and delivery system. And I think we can all argue that we're not there yet, and that there is a tremendous amount of work to be done to achieve this idea of integration and high performance across all the spectrum of activities that I just talked about. So I'll leave you with one of my favorite quotes, which I think summarizes all the things I just talked about, which is that, you know, it's not about being the biggest healthcare system or having the most revenue or the smartest faculty, you know, that's going to make for success in this new world order that I just laid out. I think it's going to be about whether or not you're responsive to change. Do you recognize that the current state of the art is not where we need to be? And are you willing to make foundational changes in research, education, and practice, as well as population health to achieve this vision, right? To abandon sort of the things that we're comfortable with and actually evolve given all of the capabilities that are at our fingertips today. today. And I think that will define successful research, innovation, and care delivery over the next several decades. And it's really about that responsiveness to change that we need to embrace as innovators and practitioners in this field. So I'll just say before I sort of wrap up, if you want to learn more about what we're doing here at Washington University, take a look at our website, informatics.wustl.edu. If you don't ask me questions right now, you can always email me, and I do tweet, and I promise other than the occasional college football reference, almost everything else is related to sort of the current state of the art in sort of strategic thinking and technology around building learning healthcare systems. And so with that, I'm happy to take some questions.
Well, thank you very much, Philip. I'm sure that if the 30-odd participants were here, they would be applauding loudly. I ask people, if you have questions, use the Zoom group chat. Send it to all so everybody can see your question, and I will pass them on to Philip and moderate those. So I take your last quote from Darwin to be a compliment for our ability to adapt this conference to the virtual mode, so thank you for that. And I have, while people are thinking about the questions they want to ask, I have a question, or maybe a compound question for you. So the ability to link outside applications to the electronic health record is something that we've been able to do one way or another for a long time. And so the more data we can get out of the EHR, the better, and the more creative things we can do. The problem is closing that loop, because you get this thing that says, okay, here's what you need to do, doc. And then the doctor has to write it down on a napkin and then go back to the EHR and type it all in. And so we don't close that loop for the active part of the decision support. The other part of that, and this is the compound part, is that the way that we deliver decision support now, either as a pull technology where you say, hey, I want to use this application to help me make a decision, or push technology in the form of alerts and reminders. They kind of work, although we have a lot of fatigue, alert fatigue and information overload. But as we scale up our understanding of the interaction between the genome and patient care, particularly things like pharmacogenomics, the amount of information that we're going to be able to deliver to the clinician and the patient is going to go up by orders of magnitude. And how are we going to adapt to that kind of level of intrusion of knowledge into our, otherwise blissful, doctor-patient relationship? We're just going, here's what I know, and here's what you should do. Suddenly the computer goes, no, doc, here's 10,000 things that you don't know that you need to know, and you better do the right thing. So how are we going to scale? How are we going to close that loop, change the model so that we can scale up to this and be able to, at the same time, incorporate new knowledge without having to write a new alert or a new application every time we come up with new knowledge, but rather build some kind of an actionable knowledge base that this learning health system can actually make use of.
So thanks for the really easy question, Jim. I like that. I would start by saying, I think the project I just described, the SPHERE project, is an interesting example of sort of closing the loop, in that tool is designed using, in this case, the Open Epic web APIs to provide that decision support and then actually record the outcome of that decision support alert back in the EHR, right? So it's pulling data out of the EHR, reasoning upon it externally, and then bringing back sort of the aggregate result, as well as sort of anything that's generated through that patient provider shared decision-making sort of episode in the notes for that patient during that encounter, so that it is recorded at least in a somewhat durable way. And I think that's just a glimpse. It's not a perfect solution, but I think that is about closing the cycle, not just getting data out, but injecting data back in. And I think we have to get more comfortable doing that in terms of injecting data back into the EHR. And it's something I think that people don't necessarily understand. Or are not comfortable with, to be honest with you. So I think that's sort of my first answer, is we just have to get more familiar with that. I think with regards to things like, bringing more sort of critical information like genomic data or other types of data, and I see a question about that. as it relates to sort of it's too much data for us to currently process when we're already overloaded with alerts. I think this speaks to the fundamental human factors and sort of knowledge engineering issues around how we build clinical decision support. You know, right now in most organizations, we have a situation where, you know, it's almost like first come, first serve. People say, I want an alert for thing X. Everybody needs colonoscopy, and we're like, okay, clinically, okay, we'll go ahead and build it, and now it's going to trigger for everybody. And so, you know, someone comes in to see, you know, say they're, you know, a rheumatologist. That rheumatologist really only wants to see alerts that are relevant to that current encounter, like a drug-drug interaction. And while, you know, the fact that the patient needs a diabetic foot exam or has other gaps in care may be relevant, you know, that's not the most relevant thing given what's happening in that encounter. And so we don't filter for that right now, right? We don't really use that context, let alone do we use more advanced sort of interactive models like what I showed you in the SPHERE world, so we don't just end up with a list of best practice alerts. And so I think there's a lot of fundamental work to do to think about how do we contextualize, how do we use knowledge engineering methods to sort of model the knowledge base in a way that's reusable, something we've known for decades, right, in the informatics community, but is not reflected in the way decision support is currently implemented in EHR platforms, and then actually use more advanced sort of visualization and other interactive technologies to make that actionable in that context. And so the good news is that's a lot of work for us to do as researchers. The bad news is we have to sort of break ourselves of sort of the current addiction to, well, let's just write another BPA if you're an Epic site or whatever the equivalent is in Cerner, where we end up with these hundreds of alerts. And I think in the genomic space, it'll become even more complicated because we're going to have a lot of information where the providers receiving it may not be fully familiar with sort of the context of those results, especially when we move beyond sort of known variants and we get into sort of that fuzzy area of variants of potential significance, right? And so we're going to have to do some fundamental work to figure out what is the right way to deliver that information. And I'm not convinced that anyone actually knows what that looks like right now. I will share here, we're actually experimenting with a model sort of stolen from the sort of social media world, which is to use an activity feed paradigm, not dissimilar to something like Twitter or Facebook, to allow for sort of all of the potential alerts for a patient to be displayed in some sort of panel within the EHR, but then allow for sort of context-specific filtering and prioritization, hopefully better than the way Facebook works. But the idea is that that would allow for an individual provider to set their preferences based on that encounter, or for us to enforce a preference based on the nature of the encounter, so that we can prioritize but still have some visibility to the full spectrum of alerts available to a patient.
Okay, good, thank you. So Chindo Hicks asked how we can integrate information from the EHR with genomic information. And
Right.
I think you might mean integrating genomic information into the information that's already in the EHR.
Right. So, I mean, my response to that is a couple fold. So I think one of the issues, right, is that genomic information, if we're talking about taking the output of, say, DNA or RNA sequencing, is fundamentally different in structure than the information that we capture from clinical encounters. So we have to begin with sort of an understanding that the data structures that we use under the hood in the EHR are simply ill-suited to storing or manipulating that type of data. So the first step is that that data ideally lives in a different environment that's optimized for storing and acting upon that data. Second of all, and actually goes to a point you made, Jim, is that we have to separate sort of the data from the interpretation, which means we really need to use these knowledge engineering-based approaches where we allow for the graceful evolution of the knowledge base used to interpret that sequencing data as new evidence becomes available, right? So we don't want to store interpretive results. We want to store interpretable results alongside a knowledge base that allows for real-time sort of scanning or rescanning of that data to produce results. And then number three, we have to figure out, back to your original question, how do we create some sort of interactive paradigm within the EHR if we're trying to use it for clinical decision making that brings that interpreted data to bear alongside the clinical context in a way that's actionable for that provider. Also understanding that we will occasionally need to rescan that data and produce new results as new evidence becomes available, and produce that in a way that can update the provider and patient on those new understandings of their sequencing data. So unlike other more traditional laboratory tests, it's really a dynamic system that needs to be treated as such. It's going to constantly change over time. But I think there's going to be sort of a, as I describe it internally here, sort of a separation of church and state between, you know, the clinical data and the genomic data, and then we need the bridging technologies that bring those together in context especially care delivery, so that we can provide the current and most efficacious interpretation of that data for that provider and patient when they need it. And I think one of the problems right now, the current state of the art, is often to treat them like discrete laboratory tests, right? I have a variant of known significance and I'm going to store it as a trio. You know, basically here's the patient, here's the variant, and here's what it means. And it gets stored in the static form. It's not dissimilar to a white blood cell count or any other laboratory test. And I don't believe that's an adequate representation of what really is dynamic data. And so we're sort of flattening the dynamic data into a way that fits into the current way that laboratory data is stored in the EHR.
Okay, great. Let me ask if there are any other questions. Get them in now, folks. Use the chat. That's that little speech bubble thing at the bottom of your screen if you bring your mouse down to the bottom of the screen. Or maybe it's on the side, depending on how you've got your screen arranged, or at the top or somewhere else. Okay. Well, you can have Philip's contact information. Philip, I can send out the link to your slides if you like.
Yeah, that's fine. Yeah, feel free to.
You want to remove the...
Yeah, I'll create a PDF version of it and send you a link, and take out the illicit epic screenshot.
Exactly, okay.
Yes.
So I'll wait for that, and then I will send it out to the distribution list of people who registered for this. And if anybody has signed on and you have not registered for this, then I don't have your email. Just send me an email, seminoj@uab.edu, and I will add you to the list. Philip, thank you very much. Sorry you couldn't be here in person.
Likewise.
The WBHM event was canceled, so you didn't miss that, but you did miss some really good meals in Birmingham.
Well, you'll just have to, once this all calms down, you'll just have to invite me to come down and visit again.
I will do that.
All right. Well, thanks, everyone. I really appreciate the time and attention.
All right, thanks. And of course, you're welcome to hang out and
right.
give us the, let's see, maybe I can take it back.
Great. All right, thank you.
I'm going to mute Philip, and now I'm going to unmute Jake Chen, who's we're going to go right into the next session. Oh, there he is. Oh, he's somehow unmuted.
Oh.
Okay. Jake, I'm going to make you the presenter.
Okay.
And then you can share your screen.
Right now, my screen says a host disabled participant screen sharing. Oh, okay. Now I can.
Let me just make sure if you were unmuted, maybe somebody else is unmuted.
Do you see my screen now?
I will when I stop looking at the. Yes. Okay.
All right. Let me know when I can start.
Go ahead.
All right. Well, good morning, everyone. I guess this is a 10-minute tool talk, and I'll start with a fresh morning BEERE. The BEERE is a tool that we published last year. It stands for Biomedical Entity Expansion, Ranking, and Exploration. So what I'm going to do in the next 9 to 10 minutes is to walk you through how this tool works and what type of problem it's going to solve. And hopefully I'll get your interest to explore it further, either alone or with us. So this tool is currently hosted at this website, Discovery Informatics UAB edu/BEERE. When you get to the tool, you will see immediately two types of input. One is that you can actually enter into the search box a list of genes. And then the second is you can also search into the search box a list of terms. So what you're going to get out of this tool is everything that in the knowledge database we know about the interacting partners of these genes or the semantic neighborhood terms of the input terms. And you may think, well, what is the use of that? So imagine that you are starting with some interesting gene, candidate genes. You say, is coronavirus, is ACE2 is very important for viral binding. but then you don't know what's going on afterwards, and you may actually enter those to get some information. Or you may actually be interested, in our case, in drug repositioning. I'm going to show you some queries. And then this is a tool that helps you not only expand, but also rank the information such that you get something relevant and to explore it in a visual environment. So just for some facts, we have almost 3 million protein-protein interactions captured in the database. And we have 20 million predications that we took from a database called SAMDB. Those SAMDB extracted the entire PubMed and all the open part of the PubMed Central, and just extracted those statements into a subject, predicate, object format that's available for this search engine. And that represents about 7.6 million gene to PubMed reference relationship, and up to 100 million facts to PubMed citations. So that's a lot of information that we put behind the scenes. Suppose that you want to start with a list of genes, and you query the database with your list of genes, and you can get an expanded list that may actually augment the missing information from your list of initial genes. You can actually examine the protein-protein interaction network to filter out additional information, and then the tool will generate and prioritize the list of genes. If you are not satisfied with the result, you can iterate back and come back with a list of genes that are actually further as input as you sift through the process. And then in the end, you get a network that looks something like this. Especially interesting is that this is a platform that we've been using more commonly for drug repositioning now. For drug repositioning, what's important is you not only have the information about genes, you also have phenotype information, you also have candidate drug information that's already been published in the literature. So we have very comprehensive list of terms and categories terms that we use to expand those the drug terms. And so I'm not going to go through the details, but I'll just walk you through the example of drug repositioning using BEERE in a case study of glioblastoma. Glioblastoma, many of you know, is a very aggressive form of cancer that currently cannot be treated with a drug alone. And even the survival with radiation and surgery is very, very poor. So we're looking for drugs that potentially can be repurposed from other cancer treatment for this disease. What we start is a list of genes from TCGA, then there are only 6 of them that are very significant, 20 mutated genes from COSMIC database, and 200 genes that are functionally relevant from OMIM database. So we go through this merge them as an input, and we get a ranked list of GBM genes. So you can see TP50, GFR, and so on. And then we get expanded GBM genes, and we can actually get to a network that look like this. And this network contains phenotype terms, like a disease named glioblastoma. And in some cases, it contains the genes, MYC, HDAC1. In some cases, and it's very interesting and surprising relationship, for example, with amyloid beta, which is heavily implicated in Alzheimer's disease. So we can actually rationalize what potential information we can extract and make use of from a graph like this. So this is actually coming from the real world result that we're really happy and then presented to our collaborator for radiation oncologist treating GBM patient at UAB. So when you look at this list, in the top 10 drug list, the one that I covered are actually either currently being used experimentally or being pushed into the cell biologist at UAB as candidate drugs to examine, especially temozolomide, and then the other ones, and we're really excited that they were ranked high. And we believe it's simply because that this information consists most critically, not only the gene information, but also the gene ontology terms and the candidate phenotype terms that make this network more useful and coming up with results that's more interesting. And so there are some examples, and then we're having these results published as posters, and then I won't go through the detail. But essentially, we not only, the idea is we can not only find drugs that are beneficial, but also we can find drugs that are potentially harmful and worsening the potential disease recovery processes. And we also have these semantic network graph where the edges actually indicate this semantic relationship that exists. And then you can actually examine the underlying papers behind it to whether believe or disprove this drug as a potential candidate for repositioning. Let me switch gear in the last 3 minutes a little bit to tell you that we're really interested in finding new applications of this. On Tuesday, I did a seminar and talked about biomedical informatics of COVID-19, which has an underlying single-stranded RNA virus called the SARS-CoV-2. And there are lots of tremendous interest in the past 2 months to find repositioning target. One of the most famous one is remdesivir, which actually came out of the UAB's antiviral center. And then there's an interesting letter that showed up in Lancet last month. And it was just essentially looking at ACE2 and some of the related biological mechanism. They come up with a rheumatoid arthritis drug, and also make the Lancet as a letter. And this is really intriguing to us because then we thought, why not try it on our tools? So I just did a very simple query term. Let me see if I can quickly show you. Oh, I only have 1 minute, so I probably don't have time to show you. Let me just walk you through. I used a combination of terms: coronavirus, antiviral, ACE2, TNF-beta, anything that I as a layman have learned from reviewing the literature, a few of these terms, and then try to search. This is a list of genes, and there are actually a gene set that has 10 genes from actually the related database pager, and then I got that genes, and then for this example I showed you, these are the input terms, and then these are the expanded terms that were retrieved. As you can see that there's not only the regular virus-related drugs retrieved, and there is also the semantic terms, and then interferon. Very interesting, yesterday I was having a conversation of John Mons, and he has stocked up some TL7, which is interferon beta-related drug, in his pocket, and just in case. And I was able to actually verify that interferons probably would be relevant to this coronavirus.
Jake, I'm sorry, but as you noted, you're out of time.
Okay.
And you're not. I can see you're not close to done. So thank you very much, but I do need to move on.
All right.
Our next speaker is Zechen Chong. I'm going to activate his microphone and make him a speaker as soon as I can find him in my list of.
Hello.
Oh, we can. Now, how is it? So I guess people can unmute themselves. Good. I'm going to make you the speaker, Zechen, as soon as I find you in my list here. Why am I?
Jim, I cannot share my screen right now. Can you?
We'll be able to in a minute here. Okay, you are going to be co-hosts now. Good, we can see your screen.
Can you see the screen now?
Yes, yes.
Okay, perfect. This is Zechen Chong. I'm an assistant professor of genetics at the Informatics Institute. Today, I would like to share our recent work on a suite of software packages designed to solve specific biological questions using the third generation sequencing platforms. So our lab is a small lab, but we have an ambitious mission. We are making sense of biology using bioinformatics. There are 3 lines of research focused in my lab. We develop innovative software to solve specific biomedical problems. This software is freely available on GitHub. We also perform hypothesis-driven data analysis. We start from a specific scientific question. We will propose a hypothesis, and we will use the consortium data and some other public data, and we will develop a computational modeling method approaches to analyze data. And finally, we will interpret the data and solve and address the scientific questions. And we have extensive collaborations, both on campus and nationwide. Today, I would like to focus our recent work on software development on the third generation sequencing platforms. So the third generation sequencing platform consists of 2 major techniques. One is synthetic long-read sequencing, which often uses barcoding, etc. techniques to get long-range sequence information from short reads, like conventional Illumina sequencing reads. For example, the 10x genomics linked reads sequences is such a platform. The linked reads are usually tens of kilobase pairs, and by overlapping the linked reads, it can generate long, accurate phased blocks. For example, this is the phased results. This blue line shadow is a phased block of 50 megabase pairs. That's very, very impressive. And the other is real-time sequencing. It includes 2 platforms. One is PacBio, a smart sequencing. The other is nanopore sequencing. PacBio platform uses a polymerase in a small vial. As the polymerase repeatedly traverses and replicates the circular molecules, the DNA template is sequenced multiple times to generate a continuous long read. It's called the CLR read. The CLR reads can be split into multiple reads called subreads by removing the adapter sequences. And by sequence multiple times, the subreads can generate a consensus sequence. It's called CCS reads, which has much higher accuracy. And the other platform is the Oxford nanopore sequencing, ONT. It can directly sequence a single-stranded DNA sequences by measuring the current changes at the base go through nanopore. So the read lengths of both platforms are very long and can range from 1 to 100 kilobase pairs. And the nanopore, they have a kit which can generate ultra long reads. It's up to 1 megabase pairs, even longer. So that's very impressive. So why we use the long reads? Because the short reads cannot access many of the human genome or other genomes high GC regions. The long reads can overcome that drawback. And long reads can potentially resolve some of the complex genomic regions. For example, the MHC region. And we know genome have lots of repetitive sequences. For example, human has over 50% of our genome are repetitive sequences. It contains, for example, short tandem repeats, large tandem, and segmental duplications. And there's lots of structural variations in the human genome and other genomes. And phasing the genome into 2 haplotypes, paternal maternal haplotype, is historically very challenging. And the good news is that long reads the cost, the sequencing cost, dropped dramatically recently. So it has the potential to replace short reads in the near future, I would say. So we developed several tools. I will briefly list all of them here. This is a meiotic recognition. Meiotic recognition is essential biological process. It facilitates the transmission of exchange genetic materials between paternal and maternal chromosomes. And it plays an important role in increasing genetic variations. Fitting paternal and maternal haplotypes is critical for accurate meiotic recognition discovery. However, previous short reads-based methods cannot get an accurate phasing result. The meiotic recognition results are not reliable, and the breakpoint resolution is usually low. So we developed a tool named MRLR, which uses the linked reads, the 10x genomics linked reads, long-range information, is phased information to accurately and comprehensively identify the meiotic recognition. This work was accomplished by my former postdoc, Peng Xu. And the stratification are important genetic variations in both healthy and diseased genomes. They are large genomic rearrangements, including deletions, insertions, duplications, inversions, translocations, and complex forms, which can involve and combine multiple simple SV types. Computationally, it's challenging to comprehensively detect all forms of stratifications using short reads because their sizes can vary from 50 base pairs to up to several mega base pairs. There are many forms of stratifications. Each stratification has its own signatures. SVs tend to appear in repetitive regions where short reads are not easy to be mapped correctly. The long reads sequence data can overcome the drawbacks to a greater extent. For example, the long reads can sometimes cover the full-length sequences, full-length repetitive sequences. With this opportunity, we developed a DBreak for long-read stratification detection. It supports both PacBell and nanopore data. The work was accomplished by my graduate student, Maggie Chen. Another hot topic in terms of long reads application is the de novo assembly. The long reads have the potential to reconstruct much longer continuous sequences from read fragments. There are several tools developed, but there are not many tools developed yet. However, de novo assembly is still a non-trivial problem. For example, some segmental duplications are longer than the read length, than the long reads even. The heterozygous are still very high, and still there. In addition, the real-time long reads have a high error rate. For example, the error rate is as high as 10 to 15%. There lacks a tool to evaluate the performance of these assemblers. So we developed an inspector to examine the long reads de novo assembly results. This work was accomplished by Maggie too. She will have a poster presentation today. You're welcome to attend and get to know the work in more details. As introduced, the long reads de novo assembly is a very hot topic in the community. So we also touched a little bit. Instead of solving large genomes, we focused on bacterial genomes, which seem more amenable. Here I use C. We found that bacterial genome assembly is not that intuitive. It's not an easy problem too. There still exist long segmental duplications and tandem repetitive sequences. The circular structure of bacterial genome.
You have 1 minute.
Okay, always make the conventional assembler fail at the boundaries, and the assembler reference is not accurate enough for downstream analysis. So we developed BEERE assembler. This work was accomplished by my student, Feng Yuan. She will also have a poster soon. Feel free to ask her at her presentation. And we can use the short read, long reads to also characterize gene fusions. Gene fusions are important variations in cancer genomes. They are important for tumorigenesis, development, and progression. They are also potential drug targets. For example, the famous medicine Gleevec can specifically target BCR-ABL fusion genes. So the long reads has the potential to solve that. We developed two Floyd to characterize gene fusions from long read transcriptome sequencing data. This work was accompanied by a research assistant in my lab, Yiqing Wang. He will also have a poster presentation. You can join that session soon.
Okay, Zechen, I need to stop you. Thank you very much.
Okay.
Okay, next is Alex Rosenberg. Alex, you are co-host.
Is this visible?
I'm going to shut down the audio.
Okay. Good morning. Today I'm going to talk about bioinformatics and tools to study B-cell repertoires. So why do we study B-cells and their repertoires? There's a lot of applications because B-cells are so critical in the immune system. There's applications for human disease in autoimmunity and allergy, and more timely for vaccinology, vaccine design, and infectious disease. And it's also important to study repertoires of B-cells to understand questions of basic B-cell biology. Before I go into a lot of details, I just want to highlight some of how we've applied these tools to problems in the past. So we've used repertoire sequencing of B cells to understand better the pathophysiology of lupus. We've used it to help understand differences in vaccine strategies in an HIV trial. And we're also currently using repertoire studies to combine with microbiome data to understand the development of allergic disease in infants by comparing urban and rural communities. B cells are lymphocytes, and they're part of the adaptive immune system. And they come in many different varieties. There are naive B cells that have not experienced antigens, and memory B cells that have experienced antigens. And B cells, these cells can also differentiate into plasma cells, which can secrete antibodies. B cell or membrane-bound antibodies, which are part of the B cell receptor and secreted antibodies, are very diverse. And these are proteins that are specific to particular antigens. And this diversity comes about by two main categories. First, it's the result of recombination of different gene segments during B cell development in the bone marrow, and the V, D, and J segments shown here. And there's different, it's a combinatorial process. And this joining is very sloppy. So there's non-templated insertions and deletions. And so the immunoglobulin gene here is specific per B cell. And so as a result, this can allow for a very diverse antibody potential repertoire. And then furthermore, during an immune response, there's proliferation, mutation, accumulation of mutations and selection to fine tune the antigen receptors for antigens during a response. And the particular gene that we sequenced to actually measure B cell receptors in the repertoire, the ensemble of these B cell immunoglobulin genes, is we sequence this, the combination of the V, D, and J segment. And this corresponds to different regions, the CDR regions, which are especially susceptible to hypermutation and can result in the antigenic specificity. And repertoires can generally be characterized as a diverse or alt polyclonal on the left, and this might be what you would expect for a naive repertoire, and oligoclonal on the right, which you might get from a clonal expansion. The way we take the sequences when we sequence these genes and then reconstruct the clonotypes or lineages they come from is we put them into bins based on which gene recombinations led to those sequences, so the V and J and the CDR3 nucleotide length, because those three parameters, if those are the same, then they could have come from the same progenitor B cell. Furthermore, we divide these into more granular bins by clustering. And so in this example here, the orange highlighted lineage has 5 sequences from plasmablasts and 2 sequences from memory cells. I'm not going to talk about the pipeline we use to process all of this data, but all of this sequence information gets deposited into databases, and we have a variety of ways of accessing this. We've got direct SQL queries, or we have another system we're developing with Darrell Hill in my group is a Python, Plotly, and Streamlit platform to have a web interface for analyzing these data. But what I'm going to show in the last few minutes is the MATLAB toolbox that we've developed, the IgRep Toolbox, myself and Chris Fucilli. And So MATLAB is very similar to R. It's an interactive scientific computing platform. And essentially what you do is you instantiate an IgRep object, and this object can have many properties. This essentially opens a connection to one of these databases. And you can access the different properties the way you would for most objects in most languages. There are different methods that can act on the databases or the objects, and there are different functions that take the object as an input to do various visualizations. And so I'm going to go through a couple of visualizations really quickly. This is one way to visualize clonality. So if you take these lineages or the clonotypes and rank them from smallest to largest, we can see that there are differences based on the different cell types that in clonality. So for example, the naive B cell has lots of very small clones and that are very diverse. So that there are a lot of lineages here. Whereas in the memory and plasma cells, as you might expect, and this is a flu vaccinated subject, we would see expanded clones that might have specificity to whatever is causing the immune response. We can also quantify this using the Hill Diversity Index, which I won't go into, but I just wanted to bring this up to show that we have various ways of looking at this data. We can look at mutation frequencies, and we can filter this down even further by dividing into isotypes. So these are distributions of mutations. So as you would expect, naive cells are not mutated. Memory and plasma cells can be highly mutated. We can take clonality plots and brush them with some other attribute of the data, in this case, isotype composition. So the top and bottom is the same data set. We're just, in the case of the top, we're brushing it with IgM percentage, and on the bottom, IgG percentage. And this, again, is what you would expect, that naive B cells are mostly IgM, and memory and plasma cells are IgG and IgA typically. We can look at the distribution of recombinations of V and J genes as a heat map. So we have V genes as the columns, J genes as the rows, and this is a fairly typical uneven bias of the distribution of recombinations that you might see. So the color corresponds to the percentage of the total. We could also look at clonal relatedness, and in this case we have three populations. We could do this between cell populations, time points, different tissues, and the ribbons connect lineages in multiple populations or time points that are clonally related. And so in this case, we can see that the center population is related to which is day 28, is related to day 14 Tibet positive cell on the left, more so than on the right. So the orange and yellow are things going from the center to both left and right. Blue and green shades of ribbons are things that only go from center to left or center to right. And so this kind of visualization immediately gives you a lot of information about things like clonal persistence in some populations, but not others. And finally, we provide for the unlimited set of questions you can ask, we provide an SQL interface so that you can ask any number of queries. I'm not going to go through this, but very specific application-specific queries and the results can be read into a variable, so you can exploit all the other functionality of MATLAB to do other analysis. And with that, I'd like to thank all my collaborators. And I want to point out, by having a lot of different collaborations on many different projects, this is a great example of not only helping to advance the science, but also the collaborators can help to generate requirements and help decide what kind of questions become the standard analyses that we want to use to expand this toolbox. And if anybody is interested in contacting me, my information's right here, afr@uab.edu. Thank you.
Okay, Alex, thank you very much. I'm going to pass control over. We don't have time for questions, but you did end on time. I'm going to pass control to Elliot Lefkowitz, who's going to make the next presentation. Elliot, you're in charge.
Okay. Can you see my screen now? No.
Yes. Do we wait? No, we don't. Not yet. You see your video.
I still have host disabled screen.
Oh wait, I'm sorry. I was hiding a dialog box. You should have control now.
Okay. Come on. Okay. That should do it.
Yes, we can see
Okay.
it.
Good. All right, so what I want to talk about today is some of the recent changes that we've made to our microbiome analysis pipeline to really incorporate some of the current tools that are being used by a lot of different groups these days. And so what we're doing is we're trying to assess the association of the microbiota, the microorganisms that inhabit an environment, including the environment that makes up our bodies. And we're trying to measure that microbiota by measuring the genomic composition of the microorganisms in whatever habitat, environment, body location that we happen to be interested in. And so in order to measure the genomic content of the microbiome, we're doing that in two ways, or you have two ways that are possible. One is through using 16S ribosomal RNA sequencing. The other is using metagenomic sequencing of the total microbiome DNA present. I'm not going to talk about that today. What I'm going to talk about today is our 16S microbiome ribosomal sequencing pipeline. And so that pipeline first starts with the sequence reads that the Heflin Center provides after the PCR is done by the UAB Microbiome Center, and then Heflin Center provides us with the sequences, and then we put the sequences through our pipeline. And this is the pipeline that we call QRAP for Chime Wrapper Analysis Pipeline. And the previous pipeline we were using was version 3. Now we're up to version 4. And these are the various steps, but I just want to concentrate on the steps that are actually have been changed recently. And so recently what we've done is we've incorporated a tool called DATA2 that provides more robust filtering and denoising of the sequence reads. And then it clusters the reads into what we call amplicon sequence variants. And I'll talk about what those are and how they differ from OTUs in just a minute. And then for assigning the taxonomy to the reads, we now have replaced the Green Genes database with the more up-to-date Silva database. And then I'm briefly going to show you the web-based report that provides a variety of different types of visualizations and statistical analysis of the data that you get. So as I indicated, two main changes to the pipeline, the use of DATA2 and the use of the Silva pipeline. And so as I indicated, DATA2, instead of reporting OTUs, that is operational taxonomic units, which previously were generated by a program called U+ that was part of the Chime pipeline, OTUs was a rather simplistic way of filtering and denoising of the sequence reads that went into the that were generated by the sequencing. And so it simply used a threshold of 97% identity, clustering everything together that was at least 97% of the sequence reads coming off the sequencer in terms of getting a better quality of sequence. And I'll show that visually in a second. It assumes that all sequence errors fell within this particular threshold, and the threshold really was arbitrary. And you also, by doing this, lose information on species and strain level identification. DATA2 produces what we call amplicon sequence variants that uses a much more statistically dependent quality-aware model of Illumina sequencing errors to denoise the data. That also allows us to use 100% identical sequence clusters to be able to now look at and get species and strain level resolution of that data. And so here's an example of what you get. You start with what's actually in the sample. The green, red, blue, and gray dots represent what's actually present in the sample. Once you sequence that, you have some errors, as you see here, by the additional dots that surround the basic sequence that you get in the larger red, green, blue, and gray dots. Now, if you use traditional OTUs and the clustering we used to use, you would just take the 97% distance around the initial dot. And you see here that mass out everything that would have been unique in the gray species or the green species, and all you're left with is the sequence reads that are part of the red or the blue species. Instead, DADA2, with its special statistically based denoising algorithm, leads you with each of the authentic reads and the exact sequence that those reads represent. And these are the reads that we then identify using taxonomic identification in the Silva sequence database. This is just a representation of the particular algorithm that DADA2 uses, and you can see it's based upon knowledge of the types of sequencing errors that Illumina sequencing tends to make. And with knowledge of those types of errors, it's able to denoise the noisy spectrum that you get after the PCR amplification. So as I said, the next step that you take when you have these sequences is to identify what those sequences are using a database. We used to use the Green Genes database. It was last updated in 2013. It has not been updated since. We found that using some of the research that we've been collaborating on with the vaginal microbiome, that we had to extend the Green Genes database in order to add sequences that were not present. Now the community worldwide on the microbiome analysis and us as well are switching to mostly the Silva database. There's some other databases that people use, but the Silva database, which is being kept much more up to date and has many more sequences and pretty much had all the sequences that we had to add to Green Genes, so we didn't have to update the Silva database. The most recent release of Silva is 138. We're actually using 132 because Silva hasn't yet released the CHIME compatible database format that we could use, but we'll update as soon as that's available. So in order now to make the taxonomy assignments, what we do is we take the sequence reads, CHIME filters out sequence reads that don't pass its quality score. We then cluster the reads using DADA2, and those clustered reads are then identified according to their species using Mother and the Silva database. And that gives us the ASV table that gives you the particular sequences and then lists the read depth according to each one of the samples. And then all of that's provided in our microbiome report, which gives you access to bar charts of the particular sequences that are present in each one of the samples and the read depth that is for each of the samples. You get information on alpha diversity, the diversity within each sample that's provided through rarefaction plots. And then we also provide beta diversity plots and principal coordinates analysis that allows you to visualize the relationships between each one of your samples based upon their microbiome composition. And they can be color-coded if we have that information according to different types of metadata. In this case, female and male samples, showing you distinct clustering of the different reads from the different samples. So I want to thank Liam Vanderpaal, who did the work to update QRAP to QRAP version 4 and incorporate DADA2 and Silva, Ranjit Kumar, who originally developed QRAP, Dongquan Chen, who is the person who analyzes most people's microbiome data at UAB these days, and then Casey Morrow, the director of the UAB microbiome facility. Thanks.
Thank you very much, Elliot. Right on time. We have time for one question. If somebody wants to put a question in the chat box. Right. Well, not seeing one. We can certainly reach out to Elliot separately. So with that, we are going to pause for about a 15-minute break. Okay, so we're going to stop for a 15-minute break. We will reconvene at 10:30. Thanks. You don't have to break your connection.
Back. All right, welcome back. We're now about to begin research talk session number 1. And our first speaker is Chindo Hicks from the Department of Genetics at Louisiana State University. The title of his talk is Clinical Utility of Polygenic Risk Scores and Racial Ethnic Health Disparities in Common Human Diseases. So, Chindo, you should be able to share your screen.
Yeah, can you see the screen?
Not yet.
Oh, okay.
And I should also make a note that these talks are 20 minutes long, so please leave a couple minutes for questions.
We want to turn on the recording.
Okay, where's that?
Click on that thing up there. See, right there.
Yeah.
I know. Where the heck is the recording? Oh, there it is. Oh, I guess we went bust. Okay, I thought we were bust. Well, we're still not seeing Chindo's screen.
Yeah, I can see the screen is still saying that. Okay.
Go ahead. Go ahead. You should be able to share now.
Can you see it now?
Not quite. We're seeing you.
So share. Let's see. Okay. Share.
Oh, now we're seeing something.
Yeah, I was. Oh,
Yes, we got it.
Good.
you can see it now?
Yes.
Oh, okay. Okay, so I'd like to share with you some of the work that is going on in my lab and some of the interests that we have and a few things that we are trying to get started working with Jake and Dr. Kov. So I'm going to talk about the clinical utility of polygenic risk scores and racial ethnic disparities in common diseases. So basically, I'm looking at polygenic risk scores and how that can exacerbate health disparities if the minority or underserved populations are not included in polygenic risk scores. So I'll give some examples to that effect. So the outline of the talk is genetic susceptibility. So basically, I'm going to focus on the polygenic diseases, not Mendelian diseases. And then genetic susceptibility as measured by the single nucleotide polymorphisms, genetic susceptibility variants. I then look at the polygenic risk scores, the clinical utility, and give some examples, how clinical use of current polygenic risk scores may exacerbate health disparities based on our population studies, and also the measures and the elimination of health disparities, how that can be done, and then some thoughts about future research directions. So when we talk about genetic susceptibility and the polygeneticities, basically with respect to common human diseases, this basically comes from genome-wide association studies. Following the completion of genome-wide association studies, there has been a lot of growing interest in terms of using those variants or summary statistics of those genetic susceptibility variants to develop polygenic risk scores. So for those who may not be familiar with the genome-wide association studies, the design is very basic. You simply have the cases and the controls, and you do genotyping of a large population. And then basically, here is, for example, the distribution of all the variants that have been genotyped. And this is the distribution of the two populations. And basically, we are looking for the risky variants in here. So essentially, you quantify that and you select the SNPs that have the smallest p-values. This is just a Manhattan plot of all the 22 chromosomes. So here we have the number of genes and how many SNPs that we select from here. So basically, once you have done that, you end up with this data here. You have the SNPs themselves here. You have the alleles here. and you have the genes here. And then you have the matrix here, basically the effect. In this case, we are looking at triple negative versus ER positive breast cancer. So we have the measurements here is the measure of effect size, or you can also use the odds ratio. So these are the metrics, if you are going to use the summary statistics, these are the metrics that you use to calculate the polygenic risk scores, or you can also use the, this one. So essentially, what we are talking about is simply aggregating these values, essentially calculating the total effect size. So where do you get those resources? So these are basically, this is a cartoon from the GWAS catalog. So these are all the diseases. So everyone can calculate polygenic risk scores using summary statistics. And these are the variants for each of those diseases. And these are just the diseases that are available. So that would be one way of calculating polygenic risk scores using this information. Alternatively, you could use the UK Biobank here, where you have many variants that have been genotyped in a population of 500,000 people. That data is available to the public. So you could use this, or you could actually estimate the polygenic risk scores here using this as the discovery cohort, as I'll show in the next slides. And then you can evaluate the summary statistics. For our purposes, we are using the summary statistics, also this database here, and then we are interested in actually working with UAB in terms of UAB has developed the Alabama Genomics Initiative. I'm sure you probably will hear that from Dr. Koff. So they have genotyped that population, but that population is relatively small. So basically, we would like to use that population as a validation cohort using either summary statistics from the Genome-wide Association catalog, or using the UK Biobank, or if we can access, there's also another bank called the Japan Biobank, which is also available for use. So that's basically the background. So now we come to estimating of polygenic risk scores, as I said. So you have the variants. This is a polygenic disease. It could be any disease, cardiovascular, cancer, or anyone that you are interested in. So you have the variants here, and you have the effect sizes here, or you can use odds ratio. If you basically have two diseases, like in this case, we have two types of breast cancer, you can also just estimate the combined effect just by looking at cancer, breast cancer in totality, if you didn't want to look at the subtypes of breast cancer. So how do we estimate the polygenic risk scores, and what are the assumptions behind this whole idea of polygenic risk scores? First, we know that the sum of R, basically, which means the total effect size for all the genetic variants associated for the associated SNPs that are associated with the disease, is typically small. It only explained about 30% or less of the phenotypic variant. One possibility for that is that we have very large numbers with small effect sizes, and that was one of the reasons why we are combining the variants. But there's a general consensus that for most of the common diseases, there are many variants that are involved in that. Hence the calculation of the polygenic risk scores using the SNPs, or using the effect size, which is a p-value, or the odds ratio in this case. So computation basically, in our case, we can use this as summary statistics, as I showed from the GWAS catalog, and estimate the polygenic risk score. Or we can also obtain the independent target sample. Essentially, in this case, it would be the UK Biobank, and then we can find another data set that is the validation data set. In this case, for example, AGHI, or the summary statistics. And then you basically select those variance with small p-values. We construct the polygenic risk scores, which is essentially the sum of the alleles weighted by B, which is basically an effect size factor in a regression model. So basically it's a mixed effects model where basically you combine the sum of the p-values for those SNPs. Okay, so how to calculate that? This is basically the model. So you have the SNPs here. You estimate their effect sizes. Basically, you calculate the polygenic risk score. Is simply sum of the SNPs that are in there plus the W here, where W is the weighted sum of the weighted SNP. And then, of course, XI is the genotype, which you can rank according to 0, 1, 2, or 3, depending on which model you are looking at. Effect size basically will be, again, will be simply the p-value or the odds ratio. And the model basically assumes the additive effects. If you expand the model, it will look like this. Basically, it will look like this. Essentially, you have Y regressed on this, and then the error of estimate of the polygenic risk score. And basically, you assume that Y is going to be standard distribution of the mean of 0 before performing the regression. Number of SNPs you are using in the score, essentially, where that is the N. The B here, the effect size, and then here you have the fixed factor. So we estimate the polygenic risk scores. You have your polygenic risk scores. You look at the distribution, low effect size, a high risk, low risk, intermediate risk, and you can project that over time, because the whole idea of polygenic risk scores is actually to estimate the life list over time, over a long period of time. For example, if you are going to estimate that someone's going to develop Alzheimer's disease, definitely they're going to develop that maybe when they are 65 or in their 60s. So you can project that over time. So this is basically the distribution here projected over time. High risk is here, intermediate risk is here, and the low risk is here. So what about the clinical utility of polygenic risk score? So once you have estimated your polygenic risk scores, how can you use those in the clinic? So one way is to improve health outcomes via precision medicine or precision prevention. In other words, you are using polygenic risk scores to identify those people who are at very high risk of developing the disease.
Hello.
So, or you could use that for future risk identification of individuals at high risk to be prioritized for treatment. For example, I work on a lot on prostate cancer. You could basically identify people who are likely to develop aggressive cancer and then prioritize those for treatment and leave those who are developing indolent cancer for disease monitoring. You could also use that for improving the sensitivity. So let's look at some examples here.
Hello. Hello.
So here is an example of how to use the clinical utility of polygenic risk. So this is a cardiovascular disease risk model, where in the model here you have the polygenic risk estimated from the variance that you find there. And then here you have basically clinical risk factors, which leads me to the question I asked Dr. Payne earlier on, is how to integrate medical records with polygenic risk scores. So you have the clinical risks here, polygenic risk scores, you combine that and you end up with an estimated risk factor, and then you can prioritize those people. Here basically you simply have the distribution based on percentile polygenic risk scores. You can see who is at very high risk, who is at intermediate risk, who is at low risk, and then calibrate that. Here's another example here, also a clinical use. So for example, here we have populations here, and we have two models. We have model A and B. So we have the distribution here, and we want to look at risk prediction here. So we have model B, model A, and this is the central distribution here. These people here at the very high risk. These ones in black here at this very high risk here. This is high risk, and this average risk, and these ones probably at the very low risk. So basically, you can identify the people or individuals based on those polygenic risk scores and then rank them according to their risk rate. Here is another example here. For example, again, we have cardiovascular disease here. We have breast cancer, prostate cancer. I work on these diseases here. So we want to identify which individuals are at high risk, which ones are at average risk here, and which ones are at low risk by aggregating that. So the question was, how can clinical use of polygenic risk scores come in? And that's the area that I'm very interested in looking at. How can we mitigate the health disparities from polygenic risk scores? So here we have a situation whereby polygenic risk scores, of course, are very much supposed to improve biomedical outcomes via precision medicine or precision prevention. But the main challenge here is that most of the available polygenic risk scores right now are more accurate in individuals of European ancestry than other ancestries, and I would like to show that. So this is most likely due to the problem that most of the genomic studies, in fact, GWAS studies, are very much biased towards European populations. And so therefore, this can pose some challenges if you are going to make improvements that in outcomes that will only favor one population or some populations relative to others. So let's look at this first with respect to the current situation. So this is basically a distribution of all the GWAS data that I just showed in the beginning from the GWAS catalog. So we look at the distribution of GWAS data based on ancestry. So we have the individuals here in millions, and we have the total world population here. And you can see that this red population here basically represents the Europeans. That's basically, and you can prove that because this information is there in the GWAS catalog. You simply have to plot it. So you get into this situation. For example, you get to African Americans, you can see this is a very small representation. If you look at the cumulative data as reported by the GWAS catalog here at the bottom here, according to the fractions, again, you see that the majority of the people are the Caucasians, and the other populations are very skewed. So what about if you just looked at the demographic relationships of allele frequencies, because you need that information, and also you build in polygenic. So this is a cartoon where you have African Americans, Caucasians here, and Asians here, and you simply plot one population against each other based on the allele frequencies from the human genome. Again, you can see that the majority of the populations in the GWAS catalog are mostly the Europeans, and you have very small population. So what about looking in terms of prediction accuracy relative to European ancestry or individuals across the 17 different traits and also different continents? And you can do that now because that information is in the 1,000 genomes. Again, you can see that this is very skewed more towards the European Americans. These values are simply showing the distributions of and the predicted relative accuracies, and then the standard errors. And again, you can see that African Americans are here because they are not very highly represented. So if you look at the polygenic risk scores themselves, the distribution, and this is just data from the UK Biobank, Japan Biobank, UK Biobank Europeans, UK Biobank African Americans, Africans, and UK Biobank Caucasians. So you have the three populations. So if you look at these cartoons here, for example, here summary statistics, we are looking at Bank of Japan and the Europeans. You can see there are differences here. Also here, if you look at this one here, based on different diseases, type 2 diabetes and colorectal cancer, you see that there are differences. If you also look at that within the populations, across the populations, for example, African Americans versus Caucasians, for example, this is African Americans here, this is Japanese here, and this is Europeans. Again, you can see that the distribution of the actual polygenic risk scores are very skewed. Another way to look at that is simply plot the allele frequencies from the data set, from the GWAS catalog, or from the UK Biobank, look at the risk alleles for African Americans and the derived alleles. Basically, derived alleles will be alleles that are already computed in the GWAS catalog. And again, there you can see that once you build in the ancestry risk alleles, then you find that there's quite a substantial difference when you look at non-Africans versus the Africans. So what about just looking at all the polygenic risk scores that have been calculated over the last couple of years, or probably over the last decade, and you look at the frequency distribution of any diverse population. So here I have a number of studies that have looked at polygenic risk scores and the years, number of studies in terms of genome-wide association studies in this case. And then we are looking at the proportion in years. Again, you can see in blue that the Europeans are mostly represented. There has been an increase in the Asians.
You've got about a minute left, Chindo.
Yeah, sure, yes, thank you very much. Yeah, so basically, if you look at the distribution here, world population estimates versus polygenic risk studies, again, you can see that the Europeans are still very much overrepresented than the other populations. So with that, so basically the conclusions and the future research direction that polygenic risk scores are posed to improve biomedical outcomes through precision medicine and precision prevention. But polygenic risk scores currently available are several times more accurate in individuals of European ancestry than other ancestries. Genetic disease risk can be misestimated across global populations under the current models because of underrepresentation of some populations. Then greater diversity must be prioritized in genetic studies to avoid exacerbating this problem. So basically, future directions, basically polygenic risk also in African Americans and African Africans and other populations. And then we would like to integrate polygenic risk score with other information, somatic, genetic expression, and all of that. And these are the people here, basically, so far who are involved. Everybody is welcome to be involved who feel this area of research is interesting. These are people from my lab. Dr. Cove has graciously allowed us that we can actually use AGHI data to evaluate some of the information that I showed you here. And Chen is leading that effort from UAB. And this guy here basically emigrated to UAB from my lab. He's a student over there. And the funding, obviously, like everyone, we are supported by the CCTS and the other entities. So that's what I have. Thank you very much.
Thank you very much. We're going to have to move on to stay on schedule. Maybe we'll have some time after the 3 talks for some
sure
questions. So I'm going to stop screen sharing. Okay.
Okay.
Which one?
Well, you should not be sharing anything.
Okay. Oh, okay.
So you should. So if you give it Richard.
Where's that?
Go to participants, manage participants. There, here.
Okay. He's unmuted.
He's unmuted, but go to more. No, not on him. Oh, you could withdraw from check out, sure. More on Richard. Cost. Okay. Say yes. Okay, Richard, you should be in charge. No, he does that. He should do share. We're still viewing the screen, it says.
Yes, I don't see anything yet.
You got to stop sharing, Chindo?
Stop sharing? Okay.
Yes.
I stopped that, yes.
Okay, now Richard.
All right, and now I will start sharing. Okay.
That's going. The next speaker will be Richard Kennedy, Associate Professor in Department of Medicine. And his talk will be entitled Geriatrics as an Informatics Laboratory.
Great, we see your screeners.
All right, you see my slides now?
Yes.
All right. Well, thank you for the opportunity to talk today. And this will be a little bit different presentation from what we've seen earlier, more focused on the electronic health record and clinical informatics. So a bit of a different direction. First thing to talk about is why should we look at geriatrics? Well, one of the key things for informatics analysis is we need to have a large amount of data. And we certainly have that in the geriatrics, both nationally and here at UAB. With aging of the population, there's going to be 10,000 people turning 65 for the next 20 years. So the geriatricians have a lot of job security. And even though older adults only account right now for about 12% of the population, they're one of the significant drivers of the healthcare system, accounting for about a quarter of the outpatient clinic visits and about a third of the hospitalizations and the prescriptions that are given out. If you look at it a little bit more locally, here at UAB Hospital, we're the third largest academic medical center in the country. We're up to a little over 11,000 hospitalizations per year in the over 65 population, and an even greater volume in the outpatient clinics and in prescription data. So there is a lot of information out there. Other reason to look at geriatrics is that it's an ideal situation, I think, for a lot of informatics applications. Geriatrics really targets very complex syndromes and risk factors. Things like delirium or acute confusional states I'm going to talk about more in just a few slides, or frailty, basically debilitation in older individuals. We also focus a lot on mobility, activities of daily living, being able to get around in your environment, being able to do a lot of the activities that are really needed for day-to-day activity like grooming, bathing, dressing, or even more complex activities like being able to manage your finances or being able to make phone calls. Other geriatric syndromes that are often a focus are falls and polypharmacy. And you can see just from this list, these are not diagnostic codes that get put into the medical record like a myocardial infarction or a pneumonia. So we really have to look at ways of describing this and mining it from the data in the electronic health record, besides just the typical approaches of looking through diagnostic codes. It's also important to really focus on these because they are associated with a lot of things that we and patients care about. Each of these associated with higher risk of mortality, nursing home placement, healthcare utilization, lengths of stay. So it's very critical for the health system and for patient quality of life. And even though they are important, these things are not often recognized by providers in family medicine, general internal medicine, or if they are, they're very poorly documented in the medical record. So one thing that we have really implemented here at UAB is a virtual ACE, or Acute Care for Elders program, as part of our quality improvement. This is really an interprofessional training designed to help non-geriatricians deliver quality geriatric care through a variety of mechanisms. It's really focused right now on the inpatient setting, but the goal is to move this to outpatient, to the pre-surgical screening, to lots of other areas across the healthcare system. But this involves a combination of screening for common geriatric syndromes, which is actually done by the nursing staff because they have the most contact with the patients, and really do these types of assessment as part of their routine care. It's then coupled with protocols implemented by the nurses to deliver the first level of care so that we can stretch the scarce geriatrician resources, really apply those to the patients that most need them, and lesser levels of care that don't necessarily need a geriatrician can be handled by other providers. Where this really becomes important for informatics work is these types of screening assessments. So here at UAB, we have a assessment for cognitive impairment using the 6-item screener on admission for everyone over the age of 65. We're doing a delirium assessment using the new desk scale every 12 hours during the hospitalization. Pain assessment using the visual analog scale every 12 hours. Mobility and activities of daily living are assessed at baseline, at admission, and every 3 days after that. So just take a moment to think about that. You know, for example, with delirium, we're getting an assessment every 12 hours on 11,000 patients per year, and we have data now going back over a 5-year period, encompassing about 41,000 patients total. This is where people in biostatistics, informatics, a lot of the data sciences really start to get very interested just at the amount of things that we could be doing.
Hey, Richard?
Yes.
We're having some issues with seeing your slides. Can you stop sharing for a moment? We're going to switch over to the slide deck that you sent.
Certainly. Let's stop sharing. All right.
And was there a dialogue to let me share?
No.
Shrink the window a second. Sure, dialogue didn't pop up. There, no? Okay, no, I guess not. It's not. Let me try sharing again. Share screen. It's not letting me share. So you made me a co-host. Go ahead.
I did.
Click. What does it say?
Oh, shoot. Yeah.
Okay.
No, it's,
My co-host. You can take Richard off being co-host.
okay.
But we'll leave his microphone on.
Oops. This one?
Yeah.
This one?
Yep. And co-host. Yes.
Okay.
Let's see if it's going to let me share. It's still not letting me share. Just didn't want wants to. What would you like to share? Oh wait, okay.
Yeah.
All right.
Okay.
All right. And
Okay. Is this your slide?
yes, we can go on to the next slide.
Okay, go ahead.
So this is just showing how well these measures have been taken up by the hospital system. This is actually just from our first 2 units, so we've even got better numbers now, but you can see we're getting about 90–95% uptake on these various screens. So these are being implemented very reliably by the nursing staff and being collected on patients. So like we were talking about earlier with the electronic health record, problems getting this data into the system, we really have a good way of doing that, at least for our virtual ACE outcomes. So next slide. This is an example from our work that we've been implementing here at UAB, really trying to identify delirium in the electronic health record. We know that delirium is very common. It's really conceptualized as an acute confusional state. The way that you can think about this is, as people get admitted to the hospital, they become very ill. If their body is not working, their brain also does not work very well. And the way that manifests itself is that they become confused. We know that among older adults, about 40% of people will have a delirium at some point during their hospitalization. So it's a very common condition, but it's often not recognized. If you look at diagnostic codes, only about 3% of Medicare billings have delirium. And even among the electronic health record, it's not very well documented as such. There was a study done where they did an expert consensus panel of keywords that they should search the chart for in identifying delirium. And when they actually applied these to the electronic health record, only about 11% of definitive delirium cases based on research evaluation had delirium or one of its synonyms mentioned in the chart somewhere. So we're really not describing delirium very well, at least from a clinical perspective. One thing that happens is a lot of non-specific terms are used. So instead of saying that a person is confused or delirious, the nurse or the other providers may describe it as the person is acting strange. Or they may describe symptoms of delirium, like wandering about the hall or not being able to remember things, but not giving a specific diagnosis. And there are definite tools that can be used for doing a manual review of charts and identifying delirium using these types of non-specific words. But this could be automated now that we have the increasing use of electronic health records. So next slide. And that's really what our grant is focusing on. We're going to implement a natural language processing algorithm that can search the EHR for these various symptoms and terms for delirium, and then be able to classify individuals without any type of manual intervention. And this really uses a data mining approach rather than going in and identifying specific keywords that we've shown have not been very effective. We're going to look for patterns in the data rather than testing a priori hypotheses. And the fact that we have these delirium screening results on thousands of patients using our virtual ACE project does allow for us to calibrate these algorithms against a large amount of data and really verify their accuracy. Our goal with this project is really that we can export these NLP algorithms and be able to implement them in hospitals that don't have the resources to do an in-person screening the way that we do here at UAB, but they do have an adequate electronic medical record that can be used for implementing these types of algorithms. And then be able to implement more community-based delirium studies rather than focusing on academic medical centers that have really been the base of delirium studies up to this point. This can also be used retrospectively, going back and mining all of the EHR data that has been collected over the years and do more epidemiological studies of delirium. Next slide. And although that's our current focus, these NLP algorithms can be combined with other EHR data to prospectively predict delirium risk. So this is much more of a long-term goal, but we would hope that we could incorporate a dynamic risk prediction for individual patients that is incorporated into the medical record. And as additional data is gathered on the patient, either from laboratory imaging or even from text that's entered into the medical record by physicians or nurses, we get a dynamic update that then tells us what this individual's delirium risk is on any given occasion. And then we could actually trigger certain actions and recommendations based on a threshold of their delirium risk. And as was mentioned earlier in the talk about the learning health system, this can be difficult in terms of, alert fatigue. Really what we've geared our virtual ACE toward is automatic intervention. So not necessarily alerting providers, but developing protocols that can be implemented. And there's no reason this couldn't be automated. For example, that at a certain level of risk, there are nursing protocols that are automatically triggered that don't necessarily need an MD or a geriatrician there to implement them. And then we may trigger a geriatrics consult for additional expertise when there are certain risk factors or certain level of complexity again, to really use our geriatrician resources in a much more efficient manner. Next slide. So that's really what we're working on at this point. But the really big picture is we want to develop a infrastructure that can really deliver better care to geriatric patients. And Informatics will be a key component to this. This has been part of our Pepper Center grant submitted to NIH that we're resubmitting in the upcoming months. We're really going to develop a longitudinal database that's going to combine this virtual ACE and EHR data for geriatrics research. And Informatics will play a key part of this because many of these predictors and outcomes are not very easily identified using standard approaches like coding algorithms or looking for particular medications. We're really going to need something that approximates much more what a human chart review could do, but that it can be automated for these thousands of records that we have available. And this will form a basis for a ongoing improvement in geriatric care. Some of it will be research focused, really being able to do population-based studies of hospitalization in older adults, not really focused on specific conditions, but really looking at general aspects of adult care. We can also use this to implement pragmatic clinical trials, doing system-level interventions on various units or throughout the hospital, and then monitoring how these types of interventions improve care based on the response in our virtual ACE system. It can also be used as a learning health system, going back to some of the earlier talk, that not necessarily looking at specific interventions that would be done by individual investigators, but more across the entire hospital, that we use this to gather information on various geriatric quality indicators and be able to monitor changes in the care of patients based on system-wide changes. Next slide. So just to sum up, I think that geriatrics really does offer a unique opportunity for Informatics system. The population is going to continue to grow for many years with the aging of the overall population. And there are multiple initiatives that are underway nationwide to improve the assessment and care of geriatric patients. I've focused on what we have available here at UAB because UAB really has been ahead of the curve in implementing these types of improvements. But this is a nationwide problem, and there are efforts really going on across institutions throughout the nation to implement these types of systems. This is really important for Informatics because these types of initiatives are really going to generate a wealth of data on geriatric patients that we have never seen before. And we need to be able to harness this type of data for research and for quality improvement in this population. And I think this is a unique opportunity for the informaticians and as well for the clinicians involved in this type of work. And so at this point, I'll acknowledge my collaborators and funding. And if there's time, we'll go ahead and open up for questions.
Yep, we're out of time.
Okay, thank you very much. I think we need to keep moving because we're a little behind schedule. Maybe we have a few minutes of the next talk for questions for any of the three speakers. So if you can stop sharing your screen, please.
No, I stopped.
Oh, you stopped it? Okay, never mind. So the next speaker will be Bruce Cork, Professor of Genetics here at UAB, and his talk will be called UAB Genomic Medicine. Are you muted? I can't hear you.
Yeah, I was muted. Just in the process of sharing my screen, I also hid the area, so I think I'm okay now. Let me just bring this up. You're able to see the slides okay? Are you able to hear me and see the slides okay?
Yes, we can, Bruce.
Okay, great. All right, well, I was asked to make some comments about informatics research in the context of genomic medicine here at UAB. I'll start with a maybe somewhat obvious but philosophical point, which is I kind of see genomics and information science as, in a certain sense of the word, synonymous. Genomics is the information science of the organism, and so the two obviously go hand in hand, but from a technological point of view, they are not as tightly linked as they could be. You've probably seen some version of this slide over the course of time where we look at the cost of sequencing of DNA since 2001, and it's obviously plummeted because of next generation technologies to the, pretty much at the $1,000 mark, give or take, depending a little bit on what you mean by sequencing and how complete it is. Moore's law, as you all know, I'm sure, covers the increase in the density of material you can put on a computer chip and how much more powerful computers become. And it, for a while, tracked with genome sequencing, but genome sequencing has outdone Moore's law quite a bit. And that's, in some ways, it's great to feel that way if you're a sequencer, but realizing that you need computational tools to interpret the sequence, that's not necessarily a wonderful thing. And indeed, sometimes those wind up being, at this point in time, rate limiting. So I'm going to talk very quickly about two broad areas. This is not intended to cover the entire waterfront of genomic medicine, but these two broad areas at the population level and at the individual level. Probably most of you are, in general terms, familiar with the Alabama Genomic Health Initiative. I've spoken about it many times, including at the UBright Symposium a few months ago, I guess now. So I won't go into huge detail about it, but remind you it's a state-supported initiative and consists of a collaboration of UAB and Hudson Alpha with the enrollment of two different cohorts. The population cohort are adults who are willing to consent to have genotyping done and return of results of actionable variants, which if any are found, they get genetic counseling and introduced to supportive care. The affected cohort are people with phenotypes who get whole genome sequencing and then return of results of variants that we think may be related to their phenotype. And ultimately, everybody is offered a chance to opt into participating in a DNA and tissue bank and a genomic database where genotypic data, as well as their electronic health records, if they're UAB patients, can be shared through the I2B2 database. I'm not going to go into detail about the return of results and our progress there. I've talked about that many times, and I don't think it's directly pertinent to the discussion here today. I wanted to focus instead on some informatics related issues. First thing to realize is that there's a 92% opt-in rate for sharing data and consenting to have specimens saved in the biobank, which we think is really encouraging. And when we survey the population, one of the prime motivations is the willingness to contribute to research. The CCTS lab under Jeff Edberg is where the biobank is. It includes a large number, as you can see, of plasma, aliquots, DNA specimens, buffy coats, and stored whole blood. And the information, this is updated now as of, I guess, about 2 months ago, so the numbers are probably increased since then. But almost 6,000 then, and now over 6,000, I think it's fair to say, chips from the population cohort. This is, by the way, using the Illumina Global Screening Array. An imputation has been done also by Mary Lynn McDonald, increasing the data density from about 700,000 variants to over 2 million. And then 444 at that point in time, annotations of genomic data from the whole genome sequencing cohort. So the resources that are there, which people can begin to use, are clinical data on I2B2, genotype data, Whole genome sequencing, I think some of this is straining the storage capabilities, and that may be in process. The imputed genomic data I just mentioned, the biomaterials that I've talked about, and participants are consented for recontact for future research, which is a resource that the community also can take advantage of. So some of the opportunities that this raises, you can ask if there's a variant you're interested in, how many people in the AGHI project share that variant? And for those that have records on I2B2, what are the phenotypes? So if you have a rare variant that is associated with a phenotype of interest, but not so rare that it won't be represented in 6,000 samples, and you want to know how many people here at UAB in this program share that variant and what do we know about them, that's there to be asked pretty much any time. With you can get general data, actually self-service, and more specific individual data with IRB approval. If you want to know simply, if you have a variant of interest, how many people in the population have it, you can ask that question quickly and easily. The only thing to realize, just parenthetically, is that when we return results, it's a CLIA grade, sort of rock solid, verified result confirmed by Sanger sequencing, but that can't be done for all 700,000 variants. So any variant you see in there, if it's a rare variant, you have to realize there could be genotyping errors. That has to do with validation for rare variants. So you need to be prepared to validate things if you're going to go on and study them. You can do GWAS and PheWAS studies, pharmacogenetic studies. Chindo talked about polygenic risk scores, but the data is in there to permit that to occur. We are launching this spring a pilot where we'll integrate into family practice a return of not only actionable variants, but polygenic risk scores and pharmacogenetic data. And we're also doing an outcomes analysis. If a person gets a result, what do they do with it? And how often do they actually have phenotype associated with the variant that's been returned? We're also very involved in the All of Us research program, which is the national program aiming to enroll 1 million people across the country to collect biospecimens as well as clinical and biometric data, as well as things like wearable fitness trackers. Our particular group, the All of Us Southern Network, is shown here, mostly Alabama, but also Mississippi and New Orleans. Next, please. Oh, I realize I'm doing it. Sorry. The data are actually now stored, data from not just us, but from the national feed in the Vanderbilt sort of data site, and you can access it through researchallofus.org. You're looking at a screenshot, which I created last night, which just gives you an idea of some of the kinds of data sets that are in there. There's over 300,000 people enrolled in All of Us at this point, and about 250,000 the last time I looked who had completed all of their relevant kind of enrollment procedures. So there's a lot of data in there. And there is soon to be launched, it is not quite up and running yet, a researcher workbench. So the way this operates is that if you have a research question, there's a variety of different things you'll need to do to get access, to sign on to security and ethical requirements, to have IRB approval and so forth. But with that, they'll create a workbench for you. You are not going to be able to download data from All of Us and just use it as you will, but you'll be able to work with the tools they make available on their site. And that will be announced sometime later this year. And it'll be available for any of the researchers here, of course, anywhere as well, to begin to ask questions with what promises to be an unprecedentedly rich data set. All right, I'm going to just now quickly segue to another kind of extreme. So those are really at the population level. Let me say something about what we've been doing at the individual level, and I'm going to focus on the Undiagnosed Diseases program, which I see as a kind of marriage, if you will, of old-fashioned medicine, depicted on the left, where the doctor is obviously raptly attentive to his patient, but with very kind of new fashion medicine, including genome sequencing. The workflow in the undiagnosed disease program is we get referrals, we do an initial review to be sure that they're appropriate and that we think we can contribute. Those that we feel are in that category get a detailed review, and then ultimately a multidisciplinary evaluation. Not everybody, but most get whole genome sequencing done at Hudson Alpha, or sometimes exome sequencing through commercial laboratories. The ones that don't get sequencing, it's because we didn't think that sequencing was likely to be helpful in answering the question. And many of our diagnoses are not genetic diagnoses. So fairly up-to-date statistics since this program began, which was in 2013, over 500 referrals, of whom about half, a little bit less than half, were accepted into the program. The reason a referral might not be accepted is either that it didn't seem to be a chronic condition that really was undiagnosed, or in some cases, they hadn't really exhausted the kind of more conventional diagnostic tools. So for example, we get referrals, people that haven't ever been seen by a neurologist, even though it's a obviously neurological problem, and we don't feel like we should be the first stop for those kinds of patients. There's a lot of referrals we get of non-specific complaints that we just don't feel we're going to be able to help with. So if those accepted, the numbers aren't going to add up, and I'll explain why in a second. 101 have been diagnosed. 41 are either ongoing, which I think is about 20 or 21, and the others are not diagnosed, but in an annual follow-up. And the balance are ones where we concluded but didn't achieve a diagnosis. So we're about 40-something% in terms of diagnosis, which we wish we were 100%, but when you remember that the patients we're seeing have generally wandered from doctor to doctor, sometimes for years, even decades, without answers, the fact that 40% now finally have a diagnosis is pretty impressive, I think. This is a list of some of the gene diagnoses that have been made. I'll make a point about this in a second. Obviously, you won't have time to go into detail about what all this means. And this is a list Michael Daniel, a T32 fellow working with us, has compiled of various of the variants of unknown significance that are being tracked, and where we're trying to do additional studies to see if we can validate these as potentially pathogenic. And that's really the interesting challenge these days in sequencing, is you get a variant that's never been seen before. How do you know that it might be relevant? Here's one quick example. It's actually what I think it might have been our first diagnosis. 3 sibs with an arthritis who were being treated with an anti-inflammatory regimen weren't responding. Their mother had some other orthopedic problems. It turned out all 3 of them were homozygous for this GNPTAB gene, which actually turns out to encode a N-acetyl glucosamine phosphotransferase, and really defines a syndrome that is a well-known syndrome where lysosomal enzymes cannot be targeted to the lysosome. And it turned out the anti-inflammatory regimen was the completely wrong approach to treatment. And although it's not an easy thing to treat, it did at least elucidate what was going on and lead to a modification of their therapy. We're now beginning to incorporate some informatics tools that others might be interested in. One's called Phenotyps. It's now a web-based tool that you can subscribe to. It allows you to, for what you can see in the screenshot, helps you to draw a pedigree. You can enter data about the patient that gets translated into HPO terms. HPO is the Human Phenotype Ontology, which is a standardized ontology for defining phenotypic features. And then it also allows you to share these seamlessly with Matchmaker Exchange, which I'll show you in a second. The other thing we're just beginning to look into now is called phenopackets, which is a standardized way of defining phenotypes. So you can actually take the phenotypic data and it creates a file, which you can see in various formats, which contains standardized information, not only the HPO terms, but also the various kind of demographic things, well, not named so much, but sex, age, things like that, can all be packaged in there. And it's a way of transmitting phenotypic data in a standardized format. And so we're beginning now to explore incorporating this into our workflow. And we're also looking at ways of linking the phenotypes with the phenopackets to make this a kind of way of standardized data exchange. We also do enter data into Matchmaker Exchange, most commonly either Phenome Central or Gene Matcher. Phenotypes essentially allows you to just click a box and it gets entered. And what that does is puts the HPO terms together with the variants that you have found into an international database. And anybody who's entered anything similar gets linked to you by email. Here's an example of one success with that. So the picture on the right is a patient we saw, and these are shown with permission of the family. In fact, these are screenshots from the publication that resulted. Patient on the right we saw many months ago, or actually a few years ago now, with heart failure. And as a matter of fact, he actually passed away subsequently of cardiomyopathy, weakness, joint contractures, cataracts, hearing loss, and some probably also dystonic posturing. And he was found to have this TOR1 AIP1 variant, which when we looked at it at first, it had never been seen before. Put it into Gene Matcher, and turned out the person on the left is in Germany. And I think you'll agree that there's a pretty decent resemblance just physically between the two. That's a bigger picture of that patient shown on the right. Our patient looked strikingly similar, although the one in Germany is still alive as far as I'm aware, but has almost exactly the same phenotype. And this resulted in a publication that was came out just a couple of months ago. We've had another example of that where Gene Matcher resulted in taking an N of 1 and expanding it to an N of several, and allowed the work to be published and define what is essentially a new syndrome. So some of the opportunities that exist here, we are creating from all of the variants that we've identified a database of unresolved variants that anybody in the community can tune into to find an area that they might be interested in doing further research on. Right now, we're not banking samples, but we actually have consent to do so. And we're considering banking at least blood and DNA and maybe IPS lines. We're launching a case conference that will allow people to hear about some of the cases that we've studied and where we have genotypic data to share. And I've already mentioned incorporating phenotypes and phenopackets into our workflow. So just to conclude, some of the resources genomic medicine has available include through AGHI, the I2B2 data, DNA and plasma, and data from whole genome sequencing. For all of us, we encourage you to explore the researchallofus.org site. In the Undiagnosed Diseases program, the variants I just described and formalized HPO terms for clinical data. And two things you just should know that are coming. One I mentioned, the AGHI pilot with family practice, but the other one is that Nita Limdi has spearheaded an effort for UAB to join the eMERGE network, which is a consortium of sites funded by NHGRI looking at merging electronic health record data with genomic data. And there'll be an upcoming site visit. We're hopeful that we'll be able to join this group. The grant review went very well. And the focus will be on polygenic risk scores, which I know Chindo has spoken with you about. So I will stop at this point, and we'll be glad if there's time to take any questions. Hope it was within the time frame here.
Okay, thank you. So are there any questions, speaker or any of the speakers? All right, well, thanks very much. Looks like the session is ending right on time. The poster session will begin at noon. And if anybody's on campus, you can pick up a box lunch at Cudworth.
And also just, hold on. It's good. Just pull my computer down real quick. We at 1 o'clock we have, or 1:30 rather, we have two simultaneous sessions. One has been down here, as Brittany was saying, and Brittany will be on this session, and I will be posting a link for the Zoom session for Min-Gauss session workshop on single-cell RNA-seq.
Okay, we're back. We're ready to start the poster session now. Can people chat to let me know if you can hear me? Okay, great. People can hear me. So I'm going to start with the poster presentation. And thanks for accommodating all the different formats and everything. I know that we had a lot of changes. People had to make presentations at the last minute, so I really appreciate that. Our first speaker will be Christopher Taylor. So let me open his presentation and get going with that. All right. And Chris, can you, I think you're unmuted. Can you try talking, see if we can hear you?
Yes, can you hear me?
Yes, I can hear you. Okay, let me, on your slideshow. All right, go ahead.
Okay. Do I have control to change the slides, or are you going to change them?
Yeah, you just let me know and I can change it.
Okay. So I'm going to briefly give you an overview of the poster that we have set to present. This is primarily collaboration with Christina Musney here at UAB and David Nelson, who is Indiana University, and then I am actually at Louisiana State University in New Orleans. Can you go forward? This study involves bacterial vaginosis, and it is the leading cause of vaginal discharge in women of reproductive age. It's associated with various adverse birth outcomes and also an increased risk for acquiring and transmitting HIV and STIs. The etiology of the disease remains fairly controversial, but epidemiologic data hints at the sexual transmission of BV-associated bacteria playing a primary role. And so in this work that I'm going to talk about today, we aim to investigate changes that occur to the microbiota prior to incident bacterial vaginosis, and we've used two different sequencing methods, and we're comparing those methods. So we're using 16S amplicon and shotgun metagenomic sequencing. Can you advance to the next slide? So this is what I'm going to show you today is a subset of some of the data from a larger longitudinal study. Again, the PI is Christina Musney, and she's at UAB. And we took 4 out of the women that were enrolled in this study, and we selected them. These were all women who got BV during the study. We sequenced them via both 16S amplicon sequencing and shotgun metagenomic sequencing. Can you advance the slide? So one of the major things that we saw out of our sequencing, again, we took 4 women and we did 6 samples on each woman, where the last sampling day, which is highlighted in the red box under each of these, when we looked at the percentage of reads via the shotgun sequencing that classified as bacterial, we saw a sharp jump at the very end in the amount of reads that were coming out as bacterial, as much as going up to 30% for one patient, going up for 60%, or even up to almost 80% for another patient. And this was seen across the 4 different patients that we sequenced. The only one that was a less dramatic increase was the last patient, K20. Can you advance? One of the interesting things to come out of this is that the reads that we found to be classified as Lactobacillus genus tended to decline leading up to incident BV. But when we looked at things via a shotgun reads that were classified as Lactobacillus, what we saw somewhat differed in some patients from what we saw via the 16S relative abundance. So the 16S relative abundance is given in the orange curve, and what you see on patient K12 is the drop in relative abundance really doesn't seem to come until the day of incident BV, which is the last time point. Whereas if you were looking at the shotgun read counts, what you see is the amount of shotgun reads that classified as Lactobacillus actually dropped precipitously in the second time point of sampling and stayed low. And the reason for the difference is really what happens is on the day of incident BV, a lot of obligate anaerobic organisms come in and they pump up the total number of organisms. And so from a relative standpoint, that's when you see the drop in Lactobacillus, but with the shotgun read counts, we see the drop much earlier. And if you look at some of the other women in the study, they track a little bit more closely when you compare the 16S relative abundance and the shotgun reads to one another. Can you advance the slide? So there are 3 obligate anaerobes that we are primarily interested in for an upcoming grant that just got funded, one of which is Gardnerella vaginalis, and we saw that across all 4 women on the day of incident BV, again the last day, we saw a precipitous jump in the amount of both shotgun reads classifying as Gardnerella and 16S relative abundance. Please advance to the next slide. Could you advance it?
Oh, sorry.
And so Prevotella bivia is another organism we're highly interested in, and we also saw in 3 out of the 4 patients a sharp increase on the last day, the day of incident BV, whereas there was not much of it via 16S or shotgun the whole time leading up to that. The only exception to that rule was K19, who did show a little bump early on, and then she showed a little bit of an increase at the end, but not as much as the other 3. Could you advance to the next slide? Adipobium vaginae is another organism of interest that's associated with BV. And we see, again, just like we saw with Gardnerella vaginalis, that it's fairly low all the way leading up to incident BV, but the day of incident BV, it jumps up sharply in both shotgun and 16S. Could you advance one more? And then in terms of lactobacillus, there are different types of lactobacillus. If we look at lactobacillus inners, what we see is that it generally declines leading up to incident BV, except if you look in the case of the woman K19, we see an increase when we look at the shotgun reads leading up to the day of incident BV, whereas the 16S relative abundance sort of tells the opposite story, that it looks like it's declining. So using this shotgun technique, we can actually look a little bit more closely at what individual species of lactobacillus are potentially doing without being handcuffed by the 16S methodology, which looks at everything out of 100% pi. Advance again, please. And the last organism I'm going to highlight is Lactobacillus crispatus, and that is generally regarded to be a protective lactobacillus, a good species of lactobacillus. And what we see in K18 and K20 is we see this progressive decrease. K18 loses it by the 2nd time point and then it stays flat, whereas K20, it sort of bumps up a little bit and then it drops off. And by the end, by the day of incident BV, none of our women have much of anything in terms of Lactobacillus crispatus left. Could you advance again to the next slide? In terms of conclusions, assessing the vaginal communities via the 16S amplicon sequencing provides us a nice broad overview of taxonomic composition. But in this study, we used shotgun metagenomic sequencing on a subset of those samples to get a more detailed view of the genetic material that's present in the community. We found similar compositions when we look at both methods, but one thing we did see was that shotgun metagenomics may be able to reveal differences in the timing of the increase and decrease of certain bacteria across a time course. And so with this pilot study, we've kind of laid the groundwork for an approach that we intend to use going forward, whereby we can use 16S amplicon sequencing on all of our samples to broadly profile the specimens, because it's a cheaper, less costly technique, followed by using the data from the 16S, we could select subsets of specimens that we want to interrogate more fully with the shotgun metagenomic sequencing. Could you advance again to the next slide? In terms of acknowledgements, this work was all funded by Christina Musney's K23 award. And then I'm actually supported by a multi-PIR01 award for this work. And we also received funding from 2 NIH awards. One was the UAB CCTS award that supports me, and another is an Louisiana Biomedical Research Network award supporting me. And all of the sequencing and work was done through our microbial genomics resource group at LSU. Could you advance one more time? And then, ongoing funding for this work, we're actually going to be continuing this work, which is supported by several recently funded awards. So the first 2 are highly relevant to the vaginal microbiome going forward. And then we have several other awards that are supporting my effort on this work going forward. One more slide, thank you. And then this is just a highlight of some of the recent publications and the collaborators we work with. We're working with Elliot Lefkowitz here and Liam Vanderpool, who are at UAB, and then Christina Musny, also at UAB. And we've published a few papers about this work. Thank you.
Unfortunately, we're a little bit behind schedule, so we don't have time for questions. So let me get set up to switch to the next speaker. Actually, while I'm doing that, there might be time for one question. All right, give me a second. Okay, our next speaker will be Yiqing Wang. Are you seeing the presentation, or
Hello?
are you seeing other screen stuff right now? Just maybe I can get a sense from people.
Okay. Hello? Can you hear me?
Yes, I can hear you.
I just wondering if I can show my own screen. I have some slides to show.
Oh, okay, okay, sure, hang on. You know, I think for some reason I have limitations as who I can make the presenter. And so I don't know if you can you send me your slides?
Oh, sure.
Appreciate everybody being patient and flexible. Let me just show the schedule so that I know that people are wondering what time they're going to present, and we had to make some switches. So let me do that. Okay, there we go. So while I'm waiting for the presentation, just take note of when you'll be presenting. Actually, Maggie, if you're available, maybe you could present while we're trying to work on getting Yiqing's presentation.
Amy, I just grant, this is Zechen. I just grant Yiqing the co-host. I think
okay.
Yeah, he can share the screen.
Okay, can you try, see if you can, I'll stop sharing and see if Yiqing can share.
So can you hear the slides now? Can you see the slides?
Yes. Yeah, just go to slideshow mode.
Good. Can I start now?
Yep, go ahead.
Okay. Hello, everyone. My name is Yiqing Wang, coming from Dr. Chong's lab. Today, I'm going to share with you my currently building tool named Philip. It's an efficient tool for discovery of gene fusions from full-length transcripts. Okay. First, what is gene fusions? Gene fusions is a hybrid genes formed by two previously independent genes. It also plays an important role in tumor genesis, and it has been found prevalent in major human cell lines. It's also critical of being a diagnostic and prognostic biomarkers. And the data used for building this tool is from SKBR3, a human breast cancer cell line isolated like 50 years ago. So here is the general workflow of our pipeline. After extracted from total RNAs, the messenger RNAs are then fed into site selection step. This step is used to detect different sites of transcripts. Then the transcripts are fed into the sequencer, the sequence machine. And we get the full length of reads. These are messenger RNAs. And then we align those transcripts to reference genome. Here we choose a Minimap tool as a default aligner because it can detect the chimeric reads, which can align to different loci. And then we only focus on chimeric transcripts, which align to multiple locus. And then, sorry. And then we only focus on the breakpoints of two different loci. After we cluster the breakpoints, because there might be some errors, so we allow a small shift. So those breakpoints are considered one breakpoint. Then we annotated those breakpoints to say where exactly it happens, to say if it happens in a gene coding region or some link RNA region. Okay, so now here's the results of the total fusions events we found. Here, the green bridge shows the inversion events. The yellow is the deletion events, while the blue is the translocation events. And then we looked for the fusion events studied by previous studies. We found 5 studies which reported the ASCII-BEERE3 gene fusions. And the totally there are 25 fusions reported. And our tool now can detect 15 of them, 60%. And here is the novel fusions that were not found in previous studies. There are 15 of them nearly. And I will choose one of them to see more details. Okay, here is specific examples of a novel gene fusions I found. It's IQ-SEC1 to a link RNA. And here is the alignment result of 2 loci. Here the reads are aligned to these 2 locus. And the same reads are indicated by same colors. Here, red, blue, blue, and they are linked together by this line, which gives a much more confident evidence of fusions. And further, we checked the whole genomic data of ASCII-BEERE3. And in this specific examples, we found a very robust evidence from genomic structural variation. It can cause gene fusions in these 2 loci. It just give a more confident evidence of the fusion detected. So in conclusion, we have successfully detected 15 out of 25 previously reported gene fusions. And we also found 15 novel fusions, which are not reported before, but with robust evidence. So this result suggests that Philip can actually and effectively detect gene fusions events. In the future, our plan is to improve our tool on more data sets. Like we're going to test if our tool can detect fusions from reads generated by nanopore platform, and if the tool can take the CLR reads, which contains more errors than currently the data sets. And we also want to simulate some data, which means we deliberately fuse some genes and we simulate as it's a real transcription data to see if our tool can detect all of them. So here is the reference for the 5 studies previously I mentioned. I collected the fusions of SKBR3 from them. Okay, thank you for listening. That's all my presentations. Do you have any questions?
Thanks, Yiqing.
Okay.
So most of the audience were muted. So you can, if you have any question, feel free to type in the chat. And
hey, the next speaker is Maggie Chen, and you know you're on there twice actually on the list of participants. I don't know which one is real. Okay, now I just see one. Okay, so I'm going to unmute you.
Am I going to use my own screen or yours?
I'm going to use my screen. So let me get that up.
Amy, you have Maggie's slides?
Yes.
Okay.
Oh, that's not the right one. Okay, that one. Actually, if you have it ready, I could let you use your own slides, Maggie. For some reason, I'm having some technical difficulties getting that to work. So let me make you the presenter.
Okay.
Or maybe Zechen, you can help me with that.
Did you ask me to open the camera?
Oh, sorry, I'm trying to get you to share. Sorry about that.
Just random Maggie as the co-host.
Oh.
What's that?
I just give permission for co-host for Maggie.
Oh.
Okay, so you can try to share, Maggie. See if you can push the share button.
Okay. Is it working?
Yeah, we can see it.
Yes, we can see it.
Cool. Okay. So I'm going to present my project today. It's called Inspector. It's a tool used for inspecting the long-read de novo assembly results. And we don't have much time, so we will not go through everything, just a little bit about background.
Maggie, we can't hear you.
No? Okay.
You need to speak louder.
All right. Okay. So de novo assembly has been a very powerful strategy for many types of bioinformatics analysis. And if you have a genome that you are interested in, you can do sequencing, either the PacBio sequencing or the nanopore sequencing platforms. Both technologies gives you a bunch of reads that range between So these reads have an average size between 10 to 15,000 base pairs. And the dual assembly is the step where you can reconstruct the original genome from the sequencing reads. So they start with finding the overlapping between the two reads by doing a read pair comparison. And they will also generate an overlapping graph, look like this. You will have the read and overlapping information there. If you can do the correction for the sequencing errors, and you can also connect two reads together if they have some overlapped region. And by doing that, you are able to connect all the reads together into one very long consensus sequence, which also have a very high base pair accuracy. And theoretically, we should have a same assembly contig with the original genome. So these two should be the same. But you also have a lot of sequencing errors in the read, and there are also complicated regions in the genome. So we always observe assembly errors from the assembly contig. And it makes very important for us to be able to evaluate whether assembly contig is like accurate or not. So what we do is we use the read as the faithful representation of the original genome and use the read to compare with the assembly contig and identify the assembly errors. And therefore we can know where are the errors and whether they are like confident or not. So this will be the major method of inspector, the way we identify the assembly errors. So we first align the read to the contig. Basically, we use the contig as the reference and find the location of the read on the contig. And if we observe some regions that are not present in the read, but somehow present in the contig, we know this will be an expansion, which means this region are not supposed to be in the genome, but it's somehow in the contig. So these are some extra sequence. And if we observe some regions that are present in the read, but somehow missed by the contig, we know this will be a collapse because they collapsed some regions that are supposed to be there. And the expansion and the collapse will be the two major types of assembly errors that we are identifying. We will also identify the smaller scale misassemblies based on the pile-up file of the alignment. So if we see a nucleotide in the contig, but a different nucleotide in the read, then we know there is a mismatch and we call it a misassembly. And we will also use a similar strategy to identify the smaller scale collapse and expansion as well. So these two will be the two major sources of the assembly errors identified by inspector. And we also have an optional input of assembly reference genome. So we also compare the contig to the reference and identify the collapse, misassembly, and expansion based on its own pattern. So this allows us to do some further analysis, like annotation. We are able to know whether the assembly errors occurred in the repetitive region or some gene coding region as well. So this will be the major method. And for the result part, we applied inspector on three popular assemblers, Canoe, Fly, and WTPG2. And here we show some plots to show the synchrony of the assembly contigs. And the top, well, the left and the middle panels are from the simulation data set, where we plot the position on the contig and the position on the reference. If we see a very straight line here, we have a perfect match between the contig and the reference. This is what we are trying to observe. And we see for the Canoe, for the Fly, and for the WTPG, we all have this straight line, which suggests they have very nice like mapping between the contig and the reference for the longest contig. And in the middle, we are showing all the contigs, both the larger ones and the smaller ones. And we observe these two longer lines in the Canoe and these two longer ones in the Fly, and the WTPG also has two. So this will be the two longer, like perfect match with the reference. And we also observe some shorter fragments. These are the shorter ones, or those are not perfectly mapped. So this will be the bad part we see in the assembly. On the right panel, we also have the real data. This will be the sequencing reads from the H02. It's a healthy individual. And we also did the same assembly, and we tried to apply the inspector on it, and we still see these two longer contigs in the canoe and these two ones in the fly. But in WTBG2, we did not observe any larger contigs that are mapped very well. Instead, we see a lot of small segments, which suggests the WTBG2 may not have the best synteny among the three tools. And we also have a table showing some numbers calculated by inspector for the three experiments. And we try to evaluate the assembly from three different aspects: the contig, statistic summary, and the structure assembly errors, and the smaller scale misassemblies. So for the summary part, we are able to count how many contigs they have, or how many contigs are longer than 1 million base pair, the size of the longest and the second longest contig, and the N50. So N50 is the popular term we use to evaluate the continuity of the assembly. If they have a higher value, it means it has a better continuity than the other. So canoe has the best continuity here, and fly does have best here, and the canoe again. And we also see the WTBG2 is having the smallest N50, which is matching with the previous result.
Maggie, unfortunately, we're just about out of time. I don't know if you can just maybe wrap up. Sorry about that.
Okay, cool. And we're also trying to show some number of structural assembly errors. And we also have the number of smaller mismatches per million base pairs. And these are the numbers. And we also are able to generate a N1 to N100 plot from the inspector. Basically, if we have the line on the top, it means it has a better result. So the simulation is very similar, and in the real data on the top, at the bottom, we have better result in the canoe and the fly, but worse in the WTBG because the y-axis is only from 1 to 14, but the other two are from 0 to 40. And we also have some IGV examples for the different types of assembly errors. Like this will be an expansion, and this is a collapse, and the smaller mismatch and the smaller collapse as well. So these are all identified by inspector, and they are supported by majority of the reads, which suggest they are highly confident. So about the conclusion, we are able to evaluate the assembly results from different tools, and we can do a comparison between those, and we can facilitate the genome assembly and other type of research. And in the future, we plan to really further improve the performance of inspector, and we also want to apply the inspector on more complicated genomes. All right, that's all. Thank you.
Thanks. Our next speaker is Frank Skidmore. Let me change the presenter real quick.
Amy, I will make my co-host to share the screen with us.
Okay. So
I think your name, rename your ID a little bit so that I can. I think this is FSCIDMOR is Frank, right?
it's FSKIDM.
OR, right? Yeah. We cannot hear you, Frank.
Can you hear me?
Oh, yeah.
Okay. Is it good good output? I mean, can you hear me? Is my voice loud enough?
Yes.
Yeah, we can hear you. So can everybody see the slides?
Okay, so I'm going to try to go through this quickly, and I think it's what I'm going to present is just a different way of thinking about approaching bioinformatic problems. It's grounded in fundamental physics, and I'm collaborating with Murat Tanik, who has developed a method of basically mapping out these relationships. You can go ahead and advance the slides, advance one slide. So I want to introduce the concept, introduce what's called the U matrix, and just invite collaboration or connections. You can go ahead and advance the slide. So we have some fundamental principles that, and This sounds really far out for a biological, but this is where we need to go, I think, in the future to sharpen the way we do our inquiries in big data. Basically, we fundamentally believe that all objects and communications in the universe are constructed from waves. There's no real solid matter. The universe is an anisotropic directional structure. The substance of the universe is actually one-dimensional. That sounds, well, basically, the math basically fits into a one-dimensional construct. Uncertainty is discrete, with the smallest uncertainty defined by something called Planck's constant, which is the smallest fundamental. So our current statistics are not discrete statistics. We basically allow things to be divided ad infinitum, but in nature, actually, there is not that capability. We are constructed from discrete lengths of time and space. Uncertainty increases over time and distance. So as things get further away, they appear to be more uncertain. And from our perspective, the universe is in a constant state of acceleration. And what this does, these principles basically bring relativity, general relativity into statistics. You can go ahead and advance the slide. So what does this look like? We can start with the concept of a point in time, and we can think about a wave or a structure that is one Planck constant ahead, one Planck constant behind. So that's that 2 pi h bar. That's the smallest fundamental length that we can have in nature. H bar is so we construct these as rings, and this actually derives from physics, where there's an understanding that light, for example, is a spiral structure. We posit that all structures are actually constructed from these spiral structures, that matter is actually constructed from rings. So we have 2 axes into the plane, one time, one space, and one along the plane, and that's basically the time axis. And as we move these structures on this one dimension of time, we actually create kind of a 4-dimensional representation, which we call space-time. Go ahead and advance the slide. So the principle of uncertainty, basically, as things get more uncertain, the original slide showed the 2 some distance apart, but uncertainty actually increases over time. And so we actually start to get a displacement, and those displacements start to give us dimensionality. The rotation, you'll notice there's a rotation, and that basically represents rotation through space-time. Rotation creates acceleration, which is one of our fundamental principles. Go ahead and advance. Then we add the principle of motion. So we can add the principle of motion through time. All objects appear to move through time at the speed of communication. In this case, it's the speed of light. You can say that we're all falling at the speed of light through time. This accords with Einstein's observations. When we move this construct through time, something very interesting happens. When I just take these 2 rings and I move it through time, I construct something which is essentially a hydrogen atom. And from the basic principle here, we can derive from first principles E, or the exponential function, which is derived from pi and properties of spin, something called the fine structure constant, which it basically defines the relationship between charged masses. And space and time appear as a 4D representation as this object moves. Now, we're talking about hydrogen atoms, but the point is that this is like the fundamental construct here. The model is scalable, and so all objects moving through time will follow these fundamental rules. So our model suggests that statistics from our observer point of view will be best modeled not as we currently model it, which is basically as a flattened representation. What we see is we're best modeled as a 3-sphere, a 3-dimensional sphere in a moving 4-dimension of space-time. And that added dimensionality gives us additional statistical power. You can go ahead and advance. you can advance the slide. So I'm going to introduce the U matrix, which is a mathematical way to represent this. It's a mathematical structure designed to model multiple events occurring over time and space. It models all communications occurring in discrete jump steps, with smallest jump steps defined by Planck's constant and quantum numbers. Those Q, N, L, and P, those are quantum numbers. They're derived from quantum physics. This incorporates quantum physics, but actually brings it into a way that we can use it to model statistics. Go ahead, advance one slide. So the basis is Fourier series of polygons, which is very different, which is a little bit different from a standard Fourier transform. And so what we conceptualize is that that we can model jump steps in space-time as jumps around a polygon. You can go ahead and advance one slide. So complexity of modeling can be increased by constructing irregular polygons, which allow us to capture complexity a little better. You can advance one slide. So I'm going to give you a practical, obviously we're not going to go through all of this in 10 minutes, but if anybody wants to talk more, they can, we'd be happy to talk. But here's an example of on the left, we have a particular signal, and we transform through the Fourier matrix, or we transform through the U matrix, which we have constructed. And you can see that there's a discrete difference between the two results. The fast Fourier transform, which is fundamental for how we do a lot of transformations in statistics and math, creates a certain structure. The U matrix creates, a different structure. The two structures are in some way equivalent, but the U matrix actually has richer information in terms of what it extracts from data. You can go ahead and transform. One more slide. So here's another example. So you see the.
And unfortunately, we have to wrap up soon because we're just about time.
Last slide. If you see kind of the scribble in the middle, basically all of those colors in the middle, if we transform on the left through the Fourier matrix, we get a particular structure. If we transform through the U matrix, we get a different, and what you can see is that the fast Fourier transform only does a limited job in capturing the complexity, whereas the U matrix actually captures the complexity more effectively. I've really blasted through this, and it's quite different probably from other things that you'll hear today. Basically the claim, you can go ahead and advance. I think the last slide might just have summary points. The claim we'll make today is that we have generated a model that encompasses time zero, and embedded features of the model give a natural explanation for many fundamental constants of nature. It creates discrete transforms of data that differs from all of our current statistical techniques. Basically, our model suggests that we're missing a dimension in our statistical modeling. Possible areas where this modeling might be useful include aging, genetics, and other areas where there are large data sets requiring statistical modeling. And if anybody wants to talk with us further, we're happy to have further conversations. We're done. Thanks.
Thanks. Our next speaker is Samantha Carlisle. Let me get her over there. Okay, Samantha, you can go ahead and start.
Okay. Thanks for giving me the opportunity to share my work with you all today. So I'm a bioinformatics scientist in the CCTS, so I work on a variety of projects. And today I'll be sharing with you one specific project that I've done in collaboration with the Udall Center and Dr. David Standard. Next slide, please. So as I said, I did this work in collaboration with the Udall Center, and we had many people who were part of this project. Next slide, please. So first, the study is looking at Parkinson's disease, which is really important because, as Richard Kennedy mentioned earlier, the population is aging. And with Parkinson's disease, it first usually onsets at about age 65. So we're expecting, as the population increases in age, we're going to see more and more cases of Parkinson's disease. Also, there's no known way to halt the progression of Parkinson's disease. And we do have a few ways to treat the symptoms, but even those aren't always working for patients. So there's increasing evidence that neuroinflammation plays a role in the etiology and progression of Parkinson's disease, and monocytes are a key component of the innate immune system. And in animal models of Parkinson's disease, monocytic activation is an early event and required in order to establish this degenerative phenotype. So in this specific study, we're going to look at monocytes derived from patients who have recently been diagnosed with Parkinson's disease. So they're classified as having early Parkinson's disease, and they haven't started any treatment courses yet. So our objective is to determine whether there is evidence for activation of peripheral blood monocytes in early Parkinson's disease. Next slide, please. So in this study, we do have a small cohort. We have 10 males who are controlled matched to 11 Parkinson's disease cases. And for females, we have 6 control patients with 7 matched Parkinson's disease patients. And from those patients, we collected peripheral blood monocytes. And using a negative selection method, we isolated only the monocytes. And from those monocytes, we're doing flow analysis and RNA-seq. And I'll be focusing on the RNA-seq today, which is what I have been working on. And with that RNA-seq data, we did validation in QC, and then we used that data to look at gene set enrichment analysis using specific gene sets related to different activation profiles of macrophages. Next slide, please. So briefly, just looking at the study population characteristics. As I mentioned before, it's somewhat of a small study. We have 34 patients, and they are age matched between cases and controls. And you can see from the PDQ-39, which is a way we measure Parkinson's disease, that the cases do exhibit a greater score compared to the controls. Next slide, please. So first, looking at the principal component analysis, we looked at the 100 most variably expressed genes in the RNA-seq data, and we saw a stark separation between males and females, actually. So from this, we decided to actually analyze males and females separately based on the advice of a biostatistician. And once we did that, and then again did a principal component analysis, we see a much better separation between cases and controls for this project. Next slide, please. When we then used hierarchical clustering and heat maps, when we looked at just both males and females together, we again see that actually the samples are separating based by sex, not by case and control. But for the purposes of this study, while that's an interesting observation, what we're most interested in is looking at differences between cases and controls, because we want to better understand the etiology and disease process in Parkinson's disease. So in subfigure B, we're looking at just the differentially expressed genes in females. And now we can see we are seeing genes that are specifically differentially expressed between cases and controls. And doing the same with the males, we actually don't see as great of a separation between cases and controls. And for us, this suggests there's more heterogeneity within the male population compared to the female population. And there may also be different mechanisms by which the disease is progressing in males and females, which actually isn't quite that surprising because we know in females, the immune system acts quite differently than it does in males. Next slide, please. So now we wanted to use gene set enrichment analysis. And again, we're actually using work from a previous group where they looked at different activation conditions to derive different modules linked to different stimuli of macrophages. And this will allow us to map patterns of RNA expression back to gene expression modules and activation states. Next slide, please. So when we did this, looking at the results, we actually saw only significantly enriched modules in the female cohort. And of those, 2 were very highly significantly enriched. And those were what we're calling module 7 and 8. And interestingly, both of those are linked to an M1 classical activation state. And these are driven most strongly by interferon gamma. And again, we think we're not seeing any significant modules in the male because of this increased heterogeneity within the males. Next slide, please. So in conclusion, we observed clear sex-based differences in monocyte activation in early PD. So the females had a much stronger signal than the males in this small study. Also, strong activation of blood monocytes with polarization towards this M1 activation phenotype in females is typically associated with pro-inflammatory actions that may lead to tissue injury. There were very few statistically significant changes in the gene expression patterns of early PD males. And again, we think this is because there's just more heterogeneity in males with the presence of subgroups, possibly. Next slide, please. So our observations are in line with clinical observations, which suggest differences in incidence, presentation, and progression of PD in males and females. So we actually see more cases of PD by about a 2-to-1 ratio in males than females. And these data highlight the critical importance of inclusive enrollment of both sexes in studies of PD and the need to consider that there may be sex-based differences in the underlying mechanism of disease. So from this small project, we're actually moving forward, proceeding with a very similar analysis on a larger cohort of patients. We're currently enrolling 60 cases and 60 control patients to kind of see if we can replicate what we saw here in this small cohort. Next slide, please. And with that, I'd like to, again, just thank all of the co-authors and collaborators that were a part of this project. Thank you for your time.
Amy, are you there?
Yeah, I think I have a delay that things are taking longer than I would like them to. I will start the, I don't know, Elliot, do you have Hope Gray's presentation ready? So I need to hand things off to.
I'm sharing it now.
Okay, Hope's sharing the screen. She can do it.
Are you ready for the rest of them? Because I have to step off for another call.
Only other one I see that's in the list is John Scholl's.
And there's also Robert Tyndall and Feng Yuan. So
I'm looking at what's available in the ATTIS folder.
Okay, well, then you might have to let them share whatever they have, then have them do it because.
Okay, so how many more?
And some of them are only showing the poster. All right, thanks.
All right, can everyone hear me?
Yes.
Thanks, Amy. All right, hello everyone. This is, I am Hope Gray. I'm a PhD student in Health Services Administration at UAB. My advisor is Dr. Sue Feldman, and my mentor is Dr. Wang Liang of the Informatics Institute. Today, I'm presenting on the applying diffusion of innovation to spiritual care documentation in the electronic health record. As you can see, the World Health Organization's definition of health does not include spiritual well-being. The Joint Commission requires that all hospitals provide access to spiritual care. Spiritual care or pastoral care is a holistic approach to patient care. And so chaplains document those visits in the electronic health record, just like any other member of the healthcare team, nurses, doctors, social workers, and others. The Association of Professional Chaplains has a standard of practice that board certified chaplains are to abide by. So this study assesses the adoption of chaplain documentation in the electronic health record since 2009's HITECH Act. So we're looking at the last decade, and the aim of this is the fact that evidence-based practice is something that many healthcare disciplines are trying to move toward. So chaplaincy is using evidence-based practice to show that what we do as chaplains is important work, as well as the fact that it can lend to quality improvement for patient care and patient outcomes. So for this, we did a comprehensive literature review of peer-reviewed articles about chaplain documentation. As you can see, the Prisma on the right, I started with 123 articles from PubMed, CINAHL, Scopus, the religion database, ATLA, and Atlas Serial, as well as PsycInfo. 13 of those articles met our inclusion and exclusion criteria. We used an Axel coding method in vivo for qualitative data analysis. And then we also used the Rogers diffusion of innovation theory to assess those articles. Our findings show that chaplains have adopted documenting in the electronic health record. 4 major themes arose, included spiritual assessment, the types of interventions, outcomes, as well as evidence-based chaplaincy. However, in the electronic health record, chaplains are lacking documenting specific outcomes for their patients and their interventions that they perform. For example, a chaplain might write in the record, spiritual care provided. Well, that doesn't give any information to the social worker or to the doctor what has been done with the patient when the chaplain visited. So we're finding that chaplains lack consistency in their spiritual screenings as well as their assessments that are documented in the chart. So in conclusion, again, chaplains have adopted the electronic health record. In looking at how diffusion of innovation occurs, Rogers shows that it happens in an S pattern. The S pattern is low at the beginning and it rises over time. And over time, chaplains are entering data into the electronic health record. So now what we would like to do is pull that data that has been input into the electronic health record through data extraction, find out what are those spiritual care interventions that are made. Sometimes those interventions are check boxes. They're not something that's written in an unstructured note. Also, we want to find out what patient healthcare outcomes occurred, such as what was the length of stay? Was the length of stay impacted? Was it increased or decreased by the chaplain visiting and providing spiritual care, and it will provide information regarding evidence-based practice for chaplaincy. The information that chaplains provide can also be utilized for social determinants of health and other psychosocial data. And lastly, we think that based on our analysis and the work that we do going forward, we'll be able to develop and propose standardized templates for unstructured pastoral care notes, also standardized screening tools for spiritual assessment. And lastly, the World Health Organization provides pastoral intervention codes that are in the electronic health record through ICD codes. We would then be able to propose expansion of those codes. Currently, there are only 4 that exist, but there's a lot more that chaplains do that could be captured if we expand those. So thank you for your attention today. Are there any questions?
I think we'll have to pass the questions for the sake of time, but thank you, Ho.
Not a problem. Thanks so much. And thank you to Dr. Liang and Dr. Feldman.
Thanks. Now, John Schultz, you should be able to share your screen and present.
Great.
Okay, we see your screen.
Awesome. All right, well, thank you all for giving me the opportunity to present today. I'm going to be really presenting my kind of more basic science research investigating roles for the Drosophila heterochromatin protein 1 family in transcriptional activation. So in order to ensure proper function in cells, DNA is packaged into a protein complex called chromatin. The protein composition of chromatin varies across the genome, and that exact composition is going to influence gene expression. In my research, I focus on a family of chromatin proteins known as the heterochromatin protein 1 family, or HP1 family. In Drosophila, there are 3 HP1 proteins that are expressed ubiquitously in somatic cells, and those are going to be HP1A in blue, HP1B in green, and HP1C in pink here. HP1 proteins are involved in diverse cellular processes, but I focus mostly exclusively on their function in gene regulation. HP1A has a critical function in the formation of heterochromatin, and so it's widely thought of as a transcriptional repressor, but it's also well documented that HP1A is required for the activation of a subset of genes. So in my research, I'm really trying to understand how HP1 proteins can be involved in repression at some loci and activation at others. And so here I'm investigating 2 models that can explain what determines whether HP1 proteins will act as transcriptional activators or silencers. Model 1 states that the effect of HP1 proteins on transcription depends on the surrounding chromatin context. So under this model, you would expect that HP1 proteins would be required for the proper transcription of genes located in heterochromatin, but if you have an HP1 protein binding in euchromatin, that it would silence that gene. And then alternatively, model 2 predicts that the effect of HP1 binding on transcription is going to be determined by the exact combination of HP1 proteins that you have at the transcription start site. So my goal here was to use available next generation sequencing data to test these 2 models and see which ones were, which of the 2 was really viable when you look at the data. I began by analyzing available ChIP-seq data for HP1A, again in blue, HP1B in green, and HP1C in pink, in the Drosophila genome from 3rd instar larvae. So here you can see the genome-wide binding profiles for all 3 of those, and the heterochromatic domains are highlighted in yellow. Summarizing these data on the left, you can see that for all 3 proteins, they have a majority of their enriched domains are located in euchromatin. But if you look at their binding behavior at protein coding genes, they're actually enriched within heterochromatin and on Drosophila chromosome 4, which closely resembles a heterochromatic state in the Drosophila genome. And then finally, if we look at their binding at protein coding genes, we can also see that these 3 proteins share a majority of their binding sites. So in particular, we see the largest number of genes fall into a category where either all 3 HP1 proteins are binding at the transcription start site, or you get a combination of HP1B and HP1C, but not HP1A at the transcription start site. So looking at their genome-wide profiles, we can see that although these genes are enriched in heterochromatin and on chromosome 4, a majority of their binding sites are in euchromatic regions of the genome. And then in addition to that, they share a majority of their binding sites. And then if we look at the expression profiles of HP1 binding targets across different chromatin contexts, so along the top, we're looking at the expression of genes bound by different HP1 proteins in euchromatic regions of the genome. And you can see that HP1A binding targets are in blue, and in gray, we're looking at the non-binding targets. And we have increased expression at genes bound by any of these 3 proteins. And that's true within heterochromatin as well. So knowledge of the surrounding chromatin context doesn't seem to predict the action of HP1 at the transcription start site. But if we go ahead and classify genes by the combination of HP1 proteins present at the transcription start site, so you can see along the x-axis, the Venn diagram is going to denote which exact combination of HP1 proteins are binding that particular set of genes. What you'll notice is that genes bound exclusively by HP1A, there in the middle, are not significantly expressed at higher levels compared to non-target genes. But genes bound by different combinations of HP1 proteins are expressed at higher levels. And so therefore, the combination of HP1 proteins present at the transcription start site does predict whether HP1 proteins will activate or repress transcription. And so if HP1 proteins are involved in transcriptional activation, we also want to develop an idea of a mechanism for how they can do that, in addition to how they mediate transcriptional repression at other regions of the genome. So previously, others have shown that HP1C has a role in mediating transcriptional activation through regulating promoter proximal pausing, which is a really critical step in gene regulation. So we investigated whether we see evidence of pausing at binding targets of the other HP1 proteins. To do this, we calculated a pausing index from available RNA pull-to ChIP-seq data, where we compare the read density at the transcription start site region of the gene to the read density across the gene body. In RNA pull-to ChIP-seq data, you see a higher peak at the 5' end of the gene as a result of that promoter proximal pausing. And when we plot this data as a metagene profile here, you can see that the genes bound by different combinations of HP1 proteins have higher 5' peaks, while genes bound by HP1A or genes that aren't bound by any HP1 proteins really don't have that same huge 5' peak. And then when we quantify that relationship as a pausing index, we're able to see that relationship play out in a really similar way to what we saw with gene expression. And so I've plotted the p-values of pairwise comparisons across groups in this heat map. But what you can take away from it real briefly is just that HP1A is not significantly different from genes with no HP1, and genes with different combinations of HP1 proteins present at their transcription start site are significantly different from those groups of genes. And so finally, just wrapping up, what we did was we looked at, again, available data from both wild type and HP1 null mutants, calculating pausing indices again. So on the left here, organ R is going to be our wild type Drosophila larvae, just for comparison. So that's what we were looking at earlier. And then going across, we see HP1A null mutants, HP1B null mutants, and HP1C null mutants. On the bottom, I've again plotted pairwise comparisons where yellow is denoting statistical significance. And then in the mutant conditions, you can see stars on the squares that represent change from what we observed in the wild type condition. What I think are immediate takeaways from this data set is that you really don't see much of a change upon depletion of HP1A when you're comparing that to the wild type. But upon depletion of HP1B or depletion of HP1C, we do start to see a number of differences that we didn't see in the wild type condition, or differences relative to the wild type condition. So depletion of HP1B or HP1C does seem to really impact RNA pull2 at these proteins binding targets. So in conclusions, our analysis supports a model of transcriptional activation by HP1 proteins, where the combination of HP1 proteins present at the transcription start site determines whether or not HP1 family members act as transcriptional activators or transcriptional repressors. And we see evidence that this transcriptional activation may be mediated through an interaction with RNA polymerase II in the pre-initiation complex. So thank you again for giving me this opportunity to present. I would like to thank the members of the Riddle Lab as well as the members of my dissertation committee. This project was funded by an NSF career grant as well as an NSF EPSCoR fellowship. And I can take questions if there's time or if we just need to move on. That's all.
Sorry about that. Thank you, John. Next up is Robert Tyndall. Robert, you should now be able to control and share your screen.
Yeah, I'm here. I don't have a button for sharing yet because the previous is up.
They should be off.
How do I stop sharing?
Okay.
There it is. Okay. Well, I'm Robbie Tyndall. I work with the Central IT group at UAB. And my project has included the Animal Resource Program, which I support directly. And we have solicited over time the School of Engineering for this project and the Research Computing Corps. We just completed an STTR grant, phase 1, to develop a device to monitor animals in research. And so hopefully this works well where I can just zoom to it. So this is the actual device. It can, the outside piece, the little green part, is a transceiver that operates in Bluetooth. And the inside thing there in aqua is the sensor pod or sensor array. We house temperature, humidity, light, motion, and ultrasonic vocalization monitoring in that pod. And so we think we have a good selection of things that we've developed with the evaluation process of design. And what we were hoping to do initially was just watch if there was an animal and create a automated census system to count the animals in our facilities. UAB having a very large number of rodents in the neighborhood of 26,000 cages on hand under study. So a very large and very tough thing to deal with. The sensors were selected basically over a need to identify environmental factors, temperature, humidity, light, because all three of those pieces were important for the metabolism of the animal and probably provide some valuable research to our investigators on the actual condition within the cage. The Laboratory Animal Association has been, and I hope I got that right, has been wanting microenvironmental data for a long time, and for the most part, it's not been available unless you did some specific things yourself. Everybody kind of assumed the inside temperature was the same as the outside. The inside temperature of the cage was the same as the room, which we have now proven in our studies is not true. We have found that there's a 5-degree variation between cages, and the reasons behind it turn out to be the population of the cage, where it's positioned on the cage in relation to the ventilation system, and also activity. So I'm going to go up here, and some of the things we can identify with our data collection is in the very first block here, we're receiving the humidity thing, and the data here is a accidental flooded cage event. And as you see, the humidity rises. Of course, in Alabama, the humidity could rise like that on any given day. So we have to analyze all of the cages nearby and pick out outliers with our analytics. Of course, down below, we've got, right below that, there's the motion detection. We either see the mouse or we don't see it. Temperature, we talked about just a minute ago, with the variations that are possible within a cage. That data is not depicted here. And then this bottom one where the lights are on and then they're off, and it shows the drop right away. I'm going to move over here to the ultrasonics piece, and this has been fascinating here and a lot of fun, thanks to the high performance computing core. This first black thing with the little dots in it, it's hard to see them, but that is a normal cage. It's actually 4 cages in normal condition where the mouses were creating their normal habitational sounds in ultrasonic frequency. Collected about 800 in about an hour over 4 cages, and that's the compilation of that. We've done other collections in other situations, and we are learning quickly that this has given us the ability to evaluate conditions that happen within the animal's environment concerning what he's experiencing. So in this top right-hand side, we've got a cage that has a forced, the first side of it is we forced a condition that was extreme. In this case, it was a flooded cage. And what we noticed was the normal sounds are shorter, and they have a little upswing to them. And we've identified that that's, well, I'll say we'll have to research it more, but we've identified the fact that that's probably a happier mouse that has the upward swings versus these long downwards things. Red also indicates that it's very loud. They're making a lot of noise here. And then down below, we're seeing the same sound shapes compared between the two models. And what we're finding is the blue is indicating a very soft, and these red ones that are, and green ones that are very similar in shape are well, they're louder, so, and softer. So we're now knowing that yes, a louder cage is something that could be a signal for distress. We believe we can pick up, well, we know we can pick up the presence of mouse pups using this methodology, aggression and fighting and injury, chronic pain. We're hoping to pick up dermatitis out of chronic pain. We can pick up mating, and we can also pick up the sexes within the cage. Mouse cycle, the mouse is their mating and reproduction cycle is very short, only about 20 to 23 days. which means that if we've not checked on our mice in a few days and we made a mistake in separation of the mouse, we can end up with overcrowded mouse, and 3 mice can become 20 something.
A minute left, Robbie.
Do what?
Minute left.
Okay. So the only thing I've got left remaining here is this was a special kind of grant. It's an STTR grant providing the NIH a way to create small businesses that can create value for multiple institutions. And so we started the company Tricorder Array Technologies to do that. The grant office has told us we must tell you about their the NIH thing. And because it's a small company, we have to let you know that the UAB Entrepreneurial Oversight Committee has developed a plan with Tricorder Array, and there are safe havens for people who feel there's some kind of violation of ethics. And pictured here is our wonderful team. I've got software engineers, biomedical engineers, undergraduates and graduates, and vets, and supercomputing experts.
Okay, thank you, Robbie. We have to move on now.
Thank you very much.
Thank you for your presentation. And the final poster, and hopefully we can get it in at about 5 minutes to keep on time, is from Emma Wang. Emma, you should have control now. Okay, we see your presentation. We can't hear you.
She's on mute. I need to be.
You're muted. I'm not able to unmute. There you go. Okay, try now.
Okay. Yep. Do you hear that?
Hear you now.
Okay. I'm Fengyan Huang in Dr. Zechen Chong's lab, and I'm going to present my project. It's a circular bacterial assembler, backsembler. So we know that there are a large number of bacteria on Earth, and it plays an important role in human health. Getting the full length of genome sequence provide a foundation for the basic research, translational vaccine, drug development, and clinical diagnosis. And the size of bacterial genome is not large, while most of them are 1 DNA molecular and have large proportion repeat fragments. And this made genome assembly difficult. Nowadays, there are numerous assemblers, and they apply only long nanopore or packed by arrays, only short illuminary arrays or hybrid arrays, while none of them are optimized for only long read assembler, high error rate, long reads, which can result in high misassembly rate. And most of them are not specific designed for bacterial genome, so they cannot generate circular genome. For only short reads assemblers, short reads cannot cover large repeat fragments, which make it difficult to resolve the repeat region assembly. Hybrid reads assembler take advantage of low error rate of short reads and structural resolve of long reads. While there are few tools available, Unicycle is one of few assemblers can resolve the circular genome while it runs ministem. Why? We found there are defects. So, so here we summarize the motivation for us to develop a new assembler pipeline. As that bacterial genome are circular, while most assemblers are not designed for circular genome, and only few ways like Unicycle can available for generating circular genome. However, it have defects. So also existing tools without preliminary error collecting generate low quality assemblies. So we developed BEERE assembling circular bacterial genome when they are only long reads or they are hybrid reads. In only long reads mode, is select top long reads and collect by remaining reads. Error rate can be reduced by this step. And collect reads and then assemble to initial assemblies. It then extract reads aligned at two ends and assemble them. Finally, emerge initial and edge contacts, remove overlap to form a circular genome. All blue mark steps are superior to other tools, for they can resolve high error rate and circular genome. The hybrid read mode here in the bottom are similar with only long read mode, but it applies accurate short reads instead of remaining long reads. To test the capability of backsembler, we use both simulate and real sequence data set. Simulate sequence will generate from microplasma arginine. The evaluation in table 1 show that backsembler performs better than other assemblies, since it generates a complete circular genome with the least misassemblies. So we can see here, the hybrid read mode of unicycler generate 2 contacts, but arginine just have 1 DNA molecular. In addition, unicycler generates several duplicates, shown as the bottom figure here. All other assemblies have more than 2 supplementary alignment clusters, which are the signs of misassemblies. And here, in the bottom figure of IgRep view of realignments, we mark the blue circle and red circle as the supplementary alignment cluster. So to detect the performance of backsembler and real sequence dataset, we run is only long read mode with microplasma and polyphony sequence. All the tools can generate 1 context compared to other assemblers. Backsembler have filled with supplementary alignments and non-cluster, while other tools have at least 1 error mark as red circle. When compared to, we also compare the structure of all assemblies. We found that most of the sequences are the same, shown as the colorful blocks, while there are few regions here marked as rectangle. They are unique in each assemblies. We also test both modes with arginine real sequence. WTDBG here run fastest while it got most errors. Can you spend a long time and extend much more long lengths? And it is unreasonable for that arginine genome size is about 700 kilobase pair, and unicycler separate the genome into 2 fragments in 2 modes. However, backsembler gets filled with supplementary alignments and non-supplementary alignment cluster. So we also check the misassemblies with IgRep view of realignments. A shows WTBG, B shows the canoe, C shows the unicycler, and D is backsembler. So all the here are the hybrid read modes. All the views supported evaluation in table 3. So finally, we got a conclusion that by assembling synthetic and real sequence data sets with only long read assemblers, WTBG, Fry, and Canoe, and hybrid read assembler, unicycler, and backsembler, our evaluation showed that long reads only assembler cannot produce circular genome, and although unicycler generates circular genomes, there are many misassemblies, and backsembler can generate circular genome, but and it has the least misassemblies on both data sets, synthetic and real data sets. So therefore, backsembler provide a better solution for bacterial genome assembly using long or hybrid reads, which will facilitate downstream and other bacterial genome analysis. So thank you.
Okay, thank you.
Opportunity.
Okay, so now we're going to switch to the special topics workshops. There are going to be 2 concurrent workshops. If you're interested in Brittany Lassen's workshop on machine learning, please stay here on this current Zoom meeting. If you're interested in Min Gao's workshop on single cell RNA-seq with Ubright, we've posted the link to that session in the chat for this particular group. So we're going to give 1 minute for people to switch around, and then also to give Brittany a chance to share her screen and take over. While we wait that 1 minute, I'm also going to say that in addition to the special topics workshop that Brittany has given, she is also giving concurrently a talk to our Info 101 undergraduate bioinformatics seminar class. And if you don't know, our undergraduate program now has a bioinformatics bachelor's degree program associated with it. And if you're interested in the undergraduate bioinformatics bachelor's program, please just send me, Elliot Lefkowitz, an email and I'll give you more information. So Brittany, I don't see that you should be able to share your screen now and take over. And I'll also
I'm almost there.
Okay, we can hear you.
Oops. Can you see it?
You can see your screen, yes.
Great. Oops. 1 second. There we go.
Getting ready, I'll introduce Dr. Brittany Lassen, who's an assistant professor in the Department of Cell Developmental and Integrated Biology. And she recently moved here not too many months ago from the Hudson Alpha Institute here in Huntsville. So Brittany, I'll let you take over.
Great. So today what I thought I would do, especially since this is focused towards the undergraduate students, is really talk about how we use machine learning to ask and answer biological questions. So I'll jump right in since we're running a few minutes behind. Whoops. There we go. So we're going to talk about why machine learning is helpful in biology, and then we'll talk about some current research examples from my group. So as many of you probably already know, genomics is a study of an organism's entire genetic code, its functions, and how it interacts with the environment. And so our goal really is to identify genomic variation associated with disease to improve patient care. And so that's what we work on in my lab. So the Human Genome Project was an international research effort to determine the sequence of the human genome. And it's really important for the work that we do because what it does is it really gives us kind of the scaffold for which all the big data that we generate and analyze from the community as a way to figure out where that goes. So you can think of the Human Genome Project as really being kind of like the scaffold here. And then the work that we're doing now is trying to fill in and figure out, you know, for this particular house or barn, what the differences are between it and the neighbors. So trying to understand how to put those pieces together. So what's enabled us to be able to generate big data and lots of genomic sequences over the past few years is the fact that this cost per human genome has really dropped over the last few years. So you can see here, we're at about $1,000 per genome now per NHGRI, though BGI says that they can go lower. So we may even see this drop further over the next year or so. And so sequencing technology is really what lets us be able to access all of this information now. And so just for the students, again, really what we're talking about is we really are looking at what the sequences of A's, T's, C's, and G's are in a given sample. And so a very brief overview of how we do that. And so my lab is a hybrid lab. We have both a wet and a dry lab component. So we do this ourselves in our lab. Is that we take fragments of DNA represented by these blue lines here. We ligate on universal adapters. So these are just sequences for which we already know. So we already know what the sequence of A's, T's, C's, and G's are in these orange barcode sequences. Then we attach them to a flow cell. So a flow cell is like a fancy microscope slide is what it looks like. So on that fancy microscope slide, we have a lawn of adapters that have a complementary sequence to this orange sequence. And so what that allows it to do is it allows, when we wash all of these different fragments of DNA with universal adapters on it, it allows them to stick to the glass slide. Then we amplify, so that lets us be able to see them. And then we wash over the slide different cycles of fluorescent nucleotides, primers, and polymerase. And so the way that works then, as you can see here, where we have our A, the T binds, and because it has a fluorescent probe attached to it, when we used to be lasers, now it's LEDs that you shine on the slide and it causes it to fluoresce green, and we take a picture. Then we wash the slide off and we do it again, and you can see here now we have a yellow because that's what's attached to guanine. So you can see if you look at this dot here, the second one from the right, you can see on cycle 1, these are different spots on the slide. It's green here, and then cycle 2, it goes to yellow. And so we sequence image by microscopy. And so these are really pretty fantastic machines. Like I said, we've got one in our lab. And it's what it allows us to do is to be able to read those individual bases. So a human hair is about the resolution that your eye as a human can resolve. And so a human hair is about 20,000 times wider than DNA. So these are very precise machines that we have to do this. So here's an example of what those pictures that are taken look like. And so you can see here up in the top left in cycle 1, I've outlined one spot and another spot across different cycles. And so you can see here that the green is a T, so it's a T. This bluish purple color is a G, and so it's here, and so on and so forth. So you can see the different cycles. Here it's green, 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 and we do that over and over again. And then we have software and algorithms that we've written as a community that will then go in and take these different spots, and read them out as the A's, C's, T's, and G's. So what we do in our lab is we look across kind of the entire central dogma of biology. So we generate data for everything that you see on this slide, and even some that I didn't include except for metabolites and protein abundance, but we also work with those data sets. And really our goal is, you can think of the cell as being kind of a big bag of information, or its patient sample as a big bag of information, and we're trying to figure out how we can extract as much information as possible out of that big bag that's in front of us. So we look at the DNA level with respect to both what the variants are, copy number changes, with DNA protein interactions, methylation, so that's epigenetic changes. We also look at the RNA level, so that includes messenger RNAs, microRNAs, long non-coding RNAs. I could fill up the entire screen with the different kinds of RNAs we look at, with proteins, as well as metabolites, and seeing how they interact with each of these. In the lab, we also use a lot of public data. So here's just a few of my favorite databases that we use. And so we think about how we can leverage big data from the public, including genomic annotations, phenotype ontologies, clinical data, to really enrich and further the data that we and collaborators generate. So just to convince you, I really want to drive home that this is big data. So I'll give you one example. So the Cancer Genome Atlas is a large NIH-funded project that includes multiple data types for over 11,000 patients across 33 tumor types. And the result of that project is almost 550,000 files with over 200,000 metadata attributes. So that results in over 2.5 petabytes of data. So this really is big data that we're talking about when we're talking about genomics. And so if you're like me, the thought of how much is 2.5 petabytes really is kind of hard to grasp. So here's a couple of examples. So it's about 5,000 years worth of MP3 songs. It would require about 50 million 4-drawer filing cabinets to store if printed. And it's estimated as the human brain's memory storage capacity. So this really is big data. And I want to remind you, this is just one data source. This doesn't include data that individual labs are generating or any of the hundreds of other large consortia data sets that are available. And it doesn't even scratch the surface for things like electronic health records or data from wearable technologies that we might also want to include when we're trying to solve questions about biology and that would help patients. So we really are talking about big, big data here. So in fact, some of my colleagues have suggested that instead of calling things astronomical, we should call them genomical. And so to drive that point home here, I've got a figure from their article, and it shows what the projections for data are by 2025. And so you can see here that Twitter, astronomy, and YouTube pale in comparison to the expected genomics data. And I want to point out that this is a few years old now, so the estimates based on the current technologies really are much closer or even exceed this 40,000 petabytes per year. So we really are talking about big, big data. I've told you about kind of the big data we generate in my group, and we have this big data out in the world that we want to make use of. And so why do we want all this data? So in my group, we want this data because we want to improve patient care by discovering mechanisms of disease, etiology, and progression, therapeutic targets, and circulating biomarkers. And so we think about how we can gather all that different information in that big bag of information. We're able to do that today in a way that we couldn't have 5 or 10 years ago because of technological improvements. So this is both at the wet lab side with sequencing technologies as well as in the dry lab side with compute availability. And so this coupled with lower costs and sample input requirements lets us have a hybrid wet and dry research lab where we can develop and implement these multiplex sequencing and integrative analysis approaches for disease research. So we're focused on like 3 big questions in the lab. So we think about how we can improve molecular diagnostic rates and pairing the right patient with the right drug at the right time. We think about how we can improve predictions, especially for small, heterogeneous, and/or rare disease cohorts. And we think about how molecular changes associated with aging and complex diseases vary across tissues and genomic backgrounds. And so all of these together are complex problems. So we have big data and complex problems in biology and genomics in general, but specifically in my group. And we want to make data-driven predictions to help patients end the diagnostic odyssey and prioritize potential therapeutics. And we want to be able to do this in a way that lets us be able to do the most good in the least amount of time. So we need to be able to automate model building. So having a cohort of grad students doing this by hand wouldn't be very effective. So the way that we do that is by using machine learning. So again, complex problems plus big data is usually a good indication that machine learning might be helpful. So let's talk a little bit about what machine learning is now that you kind of understand the problems that we're facing and why we think this solution might be helpful. So machine learning is a data analysis method that automates analytical model building. So it allows us to make data-driven predictions and discover patterns without explicit human intervention. So it's useful when we have complex problems and lots of data or big data, just like you just heard about, the kinds of problems and the kinds of data that you just heard about. Oops. There we go. So in traditional programming, what we do is we have a computer, we input data and a program, and we get out an output. Everybody listening to this has done this before. A calculator would be a very simple foil for this. So you might put into your calculator the number set 2 and 3 and the program addition, and the computer or the calculator would tell you that the output is 5. So how does machine learning differ from this? So it differs because here we give the computer the data and the output, and we ask it to tell us the program, the model, the equation, the algorithm. These are all names for the same thing that describe the relationships between those data and outputs. So taking our calculator example through, we would now give the computer the number set 2 and 3 and the output 5, and we would ask the computer to explain the relationships between those numbers to us. And if it does its job right, it would tell us back that the relationship is through the addition algorithm. So how do we do this in biology? So here our data profiles are things like genomics data, phenotype and clinical information, biological annotations, and the sorts of programs that we're trying to generate here are equations for predicting patient or system diagnosis or outcomes. So something about either the biology of the system, whether or not the cells responded to a drug, something about the patient, do they have a particular disease or not, or a certain outcome. And so that's what we're really trying to learn here, is by making use of all the data that we've generated, that collaborators have generated, and that has been generated in the community. So for example, we might be able to find a pattern that describes drug response. So I would just want to emphasize here that our goal isn't to make perfect guesses, but to make useful guesses. So we want to build a model that's useful for the future. So if we have a model that perfectly fits our data, then it may not be something that we're able to extend to new data that comes in. And so we work really hard on the statistics side to make sure that we find that balance. So you guys, just like you've all done model building before, you've all used machine learning before. So I think we're all about to get a lot of use out of the Netflix recommendation engine. When you have that queue that comes up that recommends different choices for you based on what you've already watched before, it's doing that based on different flavors of machine learning. The same thing with detecting fraud on your bank accounts. Every time I travel, I get a phone call or a text message from my bank saying, hey, you don't normally spend money in Prague. Is it really you? The way that they're able to do that is by looking for patterns in your spending that are unusual. They're looking for outliers. And then finally, I think you've all probably heard of Google and their self-driving car, and the way that they're able to respond to the environment and change course is through using different types of AI and machine learning protocols. So the advantages of machine learning are that they help us to identify trends and patterns. They have limited human intervention once implemented. They can continuously improve models, and they're flexible to multiple data types. The disadvantages of machine learning are we really do need lots of data. If you have small data sets, that's really difficult, though I'll tell you a different flavor of machine learning in a minute that we can leverage in those situations. Also, you really do need domain-specific and implementation expertise. So for example, in my group, we're the implementation experts, and in certain areas we have domain expertise, but in a lot of areas we really rely on close collaborations with both laboratory scientists and clinicians. Machine learning can have high error rates, and so having orthogonal data sets or orthogonal systems, being able to test your predictions and really understanding what those error rates are and what your tolerance to those error rates are really important. And then finally, interpretation of results. So sometimes it can be a challenge to understand which data pieces led to a particular prediction, though there are a lot of advances in that space over the last few years that I won't talk about today due to time limitations, but you should know that exists. So how have I used machine learning in my work previously? So I'll give you two examples where we've used machine learning to come up with improved cancer models. So the first was in early diagnosis of cancer. So what you're looking at here is called a ROC curve, receiver operating characteristic curve. And what you need to know is it's a way of looking at the true positives and the false positives of a model and being able to plot them out. And so if you have a line that falls on this identity line here, this black one that runs crosswise across the graph, that's essentially the same as flipping a coin. So you have a 50% accuracy of your model. And then the higher that the curve is above the line, the better your model is at predicting an event. So what you can see here with these purple and red lines, which is the same model for kidney cancer, trying to say, do these patients have kidney cancer or not, is using the same model using two different data sets. So the red one is data that I generated with collaborators, and the purple one is using a public data set to check our model. You can see that we did a fantastic job. We had a 99% area under the curve, which means that we're really good at being able to apply this model. And so using traditional methods where we only look at one data point did not give us this sort of prediction, these performance characteristics. it really did require us to have these machine learning models where we could look across a large data set to be able to get this kind of performance. And the second example is looking at, so this was thinking about diagnosis, is now looking at improved prediction of cancer outcomes. Whoops. And so for here, you're looking at a receiver operating curve again. And here what we were able to do was find patterns across 19 different cancer types to split them into different groups. And by virtue of those groups, which had to do with how much cell proliferation was happening in the cancers, it actually helped us to group cancers so that we could find pan-cancer models of patient survival that outperformed individual cancers alone or other sets of cancers. So for example, what we see here is that by looking at these cancers that had this cell proliferation phenotype, which was 7 different cancer types, we were able to get this better curve than looking at all the cancers alone or cancers that didn't. So again, it's really a matter of being able to use knowing the right biological question, being able to apply the right machine learning method with the right statistical analysis, and then the orthogonal data sets that really help to give us confidence of this. I'm going to give you a couple of examples from our current work here in the last few minutes. We're going to focus on rare disease for the next 5 or 10 minutes. So there are about 30 million Americans that are living with a rare disease, and the molecular diagnosis for those is less than 50%. Even more heartbreaking is that 95% of those do not have an identified therapeutic option. What that means is that there's this lack of rare disease clinical samples, because these are our patients that, even though there are a lot of them in aggregate, 1 in 11, these are patients who have a disease that is less than 1 in a couple 100,000. So there's very few of these patients. So there's a lack of these rare disease clinical samples and suitable preclinical models, so appropriate mice or tissue culture systems for research and development, and that makes it difficult to diagnose, but particularly to nominate, let alone test therapeutic options for these patients. We work on several different areas in the lab of trying to improve both the molecular diagnosis and the therapeutic options by using genomics and data science approaches. And so I won't focus on it today because of time, but for molecular diagnosis, we work on how we can use additional data types, integrative approaches, including machine learning, and then liquid biopsies to be able to improve this diagnosis. Great. What I will focus on today is tell you a little bit about how we can identify therapeutics using transfer learning, which is another type of machine learning. And then I'll touch on very briefly preclinical models and pattern matching. So I just want to point out that this is what we're talking about here is personalized and precision medicine. So we're talking about if we have patients here, in this example, there are cancer patients with colon cancer. They all receive a therapy, and some of the patients, do well on that therapy, some have no effect, and some have adverse effects. What we want to be able to do is to identify upfront which patients fit into those curves so we can give them tailored therapies and help them to all do better. I want to mention, because we hear a lot about precision and personalized medicine these days and about it being kind of new. What's new about it is the fact that we have so much data. So the data is both the genomics data that we're generating in the labs and in the clinics, but as well as data that we have about mechanisms of actions of drugs, about how cells behave in their cellular profiles change in the presence of drugs. For example, transfusion patients have been matched with donors according to a certain blood type for over a century. And we've been tailoring prevention for patients with inherited conditions for decades and tailoring treatments for patients' molecular profiles for the last few years. And so we're able to do this better and for more patients these days because we have all of this big data that's generated that we're able to leverage. So we think a lot about how we can help those patients with rare disease in the lab. And so we can certainly identify drug targets that then companies can go after making new molecules for. But we focus most of our time and energy on drug repurposing. And drug repurposing is a strategy for identifying new uses of approved or investigational drugs that are outside the scope of the original medical indication. And so we like this because it allows us to use de-risked compounds. We already know what these compounds do in a human body, and it helps us to potentially lower the overall development costs for rare disease patients for which there's never going to be millions of people taking that drug for that indication. So it may be difficult to get that developed properly. And it also helps us with shorter development timelines. So while we do this novel drug target approach by looking for genes, pathways, and signatures and transcription factors that might be helpful for these rare disease patients, I'm going to focus on the drug repurposing part here today. So I told you about how for machine learning, we had data and outputs and we were able to get a program out of it, and that would help us to describe drug response. But then I told you that we were going to talk about rare disease patients, and I said, you know, we need lots of big data and lots of information to be able to model these things properly. So what do we do? How do we leverage all the big data that's being generated in the community for rare disease patients who we can really think of as being like unicorns? We're going to see them once and we may never ever see one ever again. So how can we use this new technology and these new approaches to be able to help these patients? So the way that we're approaching it in my group is using transfer learning. So transfer learning is a machine learning procedure that lets us model a novel scenario by leveraging data from a related cohort. So for example, we may only see one unicorn, but we could go out and collect lots of data, about narwhals. And so they have shared features in their horns. We could collect lots of data about horses and their shared features in their tails. And at least in these pictures, we could go out and collect lots of data about deer because they're shared, they have shared features in their legs. So the goal is to transfer knowledge from one setting to another. So we can collect data about something that is more prevalent or to which we already have access to, and then we can transfer that knowledge to our new rare patients. So we're working on how we can do that for this drug repurposing nomination strategy. So the idea is for a bunch of genes, for lots of database samples. So this could be everything in the sequence read archive. It can be everything in the these other large perturbation encyclopedia where there are hundreds and thousands of cell lines that have all been tested with hundreds or thousands of compounds, and we've looked at what their gene expression profiles look like. We can do this for lots of different kinds of data. But the punchline is that we can take samples from multiple origins and conditions in a gene expression database or another omics database, and we can extract biological signatures. So for example, here I've mocked up maybe this purple box represents a signature for ATP synthesis, and the blue box is a representative response to drug, a particular drug. And so remember, I've shown you a cartoon here, but we're really talking about 20,000 plus genes across hundreds, thousands, or even millions of database samples. And so then we're able to take and we can transfer that learned pattern to our cohort. So for our much smaller cohort, we can say, well, this pattern or this pattern looks most closely like this pattern. So we think it's response to drug X. And so that's super useful because in our small cohort, we would never have the statistical power to be able to identify this signature as being important on its own. But because we're able to leverage these big cohorts, that lets us do it. And so we can use that to look for prioritized lists of drug repurposing candidates. And so this lets us nominate drug targets and repurposing candidates, which are really just phenotype modulators. So I'll just briefly talk about this last little bit so then we save a few minutes for any questions that you might have. But then the important thing that I want to drive home is that a prediction is just a prediction until you test it. So one of the things that we like to do is as I mentioned before, is that we match up with domain experts. And so what we can do then is we can say, we can have them go and test our predictions in either their cell lines, patient-derived iPSCs, mouse models, zebrafish, whatever the case may be. So for all of our projects, we have laboratory scientists with disease models that we can then test our predictions in. And we're also working on strategies for improving those preclinical model testing. So using machine learning and those sorts of analytical approaches to be able to figure out what the best cell lines to model a particular patient profile that's maybe has a rare disease is. So maybe the important part of that rare disease profile is this signature here, this red and orange signature. And so we can use that to say, okay, you know, this particular cell line best models the relationship between those genes. So that's the one we want to test in, or to make iPSC derived models more closely mimic adult tissues for which we're interested in. And those are just a few of the ways that we're working to kind of go through the entire process and be able to also improve how we're modeling our predictions. So in the last minute, I just want to mention, we talked about how one of the goals of the lab, though I didn't go into detail about it, is to try and improve this molecular diagnosis to give patients, let them stop the journey of trying to figure out what's wrong and be able to move on. But what about for these patients whom we still don't have a molecular diagnosis for? So for those patients, we can also use different pattern matching algorithms, which is really what a lot of our transfer learning and machine learning is. So we can say that for a patient with a molecular diagnosis, we can match those profiles to a patient without a molecular diagnosis. And it's also really important if you think back to that precision medicine slide where there were patients who didn't respond to a drug or didn't respond well to a drug. It is really important for helping us identify those patients who won't benefit from a particular therapy, because a lot of times these therapies, particularly for cancer patients, are very harsh and can make the patients really sick. And so this gives us an opportunity to be able to say, you know, hey, these patients are not good candidates for these particular drugs. And so there are examples in the FDA now for tissue and site agnostic indications. And so we're working on finding other signatures that unfortunately I don't have time to go into today, but we're working on finding other signatures to find these tissue and site agnostic indications. And so we're doing this all in the real world and here at UAB. So we just got funded on a project here where the idea is that for patients who have glioblastoma, their tumors are grown in basically mice avatars, so patient-derived xenografts. And what we're doing is we're starting up being able to generate and evaluate comprehensive RNA profiles from these mice. And so this requires, we have some molecular biology tricks up our sleeve in the wet lab side to be able to kind of grab all the different species of RNA at once. And then we are able to match these profiles to existing databases of cell lines to prioritize them. And then the second part of the grant is to be able to use those profiles and these transfer learning schema to prioritize the drugs, and then we'll be able to go back into the mice and see if our prioritizations are useful. Do they work? And so that lets us iterate back and forth between the wet lab, the dry lab, and the clinical questions to be able to improve our models. And it's you know, really a positive upward spiral. So, you know, we talked very briefly today about strategies we're using to end the diagnostic odyssey. Then we talked a little bit more in detail about pairing the right drug with the right patient at the right time, using these different machine learning processes. And so I think I'm gonna close here. So we've got 2 or 3 minutes for questions if you like. Because this is the student section, I wanted to mention really quickly, if you're interested in genetics and genomics, I would recommend any of these 3 books. They're all fantastic and they're written for, you know, a smart but lay audience. So they're accessible and they're all relevant. And then a big thank you to my lab and our funding sources. And thanks again for joining this afternoon. And please let me know if you have any questions.
Thank you, Brittany. That was great. If someone has a question, you can either unmute yourself or type it into the chat box. While we wait, I have a question. So one thing, Brittany, we're always worried about in analyzing our data is errors creeping in, whether or not it's in the algorithm itself or it's somewhere in the data set. So maybe some sample is misidentified or there's missing data. How sensitive are machine learning algorithms to errors in the data?
Yeah, so that's a great question. The simple, well, the long explanation is we spend a lot of time in our lab with data cleaning and QC. So as you know, Elliot, and perhaps some of your students know, you know, a large percentage of what you end up doing in computational biology and bioinformatics is that data QC and cleaning. So we spend a lot of time making sure that if we have patient profiles and they say they came from boys by looking at, you know, the genes that are there. Do they have a Y chromosome? We spend a lot of time on quality control, making sure that if we're using a gene expression profile, that it meets certain benchmarks so that we know that the data is at least of good quality, and looking for things like sample swaps or duplicate samples. And so past that, assuming that all of those checks have been done, that's why for us it's so important that you know, we make sure that we work with domain experts to make sure that the predictions that we get are make sense. And we try to only apply it in places where we think we can go and test it at the bench. And so that way we don't spend a bunch of time spinning our wheels. So there's always going to be errors that creep in. And so for us, the best way to be able to sidestep that and not spend, you know, lots of money and time working on something that is a dead end because there's an error we couldn't account for is to just go test it. And then if it doesn't work, we move on to the next thing.
Great. So we have one question on chat. What does it mean when someone says overfitting and underfitting the data?
Yeah, so overfitting the data is when you have made your model be so specific to the data that you use to make the model that it's no longer, it doesn't do very good when then you apply it to new data sets. So for example, I talked about the kidney cancer example here earlier. And so this is why it was so important when we made our model. I paid a lot of attention for this red line here. You want to optimize the area under the curve here, but you don't want to fit your data so perfectly that then when you go and apply that equation to a completely orthogonal cohort, you know, it won't do very well because you have completely modeled only your data points and not the disease itself. And so that's why it was so important that we have this orthogonal data set here that allowed us to be able to show that our model, we could have patients walk into the clinic and our model would do a good job of describing their situation and not just be caught up in the data that we use to generate the model. And underfitting is the opposite problem.
And one last question from one of our students. What is the next step in your project?
Yeah, so I'm pretty new here, as Elliot mentioned. So we just started the lab up last summer. So we've got lots of different projects that we're getting off the ground now, though we're you know, probably going to be slowed down a little bit. I just spent the last 2 days kind of shutting our wet lab down in anticipation of work from home. But for the glioblastoma project that I showed you here at the very end, our next steps are we're in the process of generating these profiles so that we can do this transfer learning and be able to make these predictions. And so we think about it as two ways. So one, we really care about being able to help in glioblastoma, but we also think about how can we take these methods and make them generalizable enough so that we can apply them to other diseases. And so we're working on trying to set these methods up as tools so that we can then apply them in other areas. So for example, we have collaborations with folks who work on Friedreich's ataxia and polycystic kidney disease here. And so we can apply those tools elsewhere. So kind of our short term is to do everything that's on this slide here, and more long term is to be able to move towards patient profiles and making predictions for patients in real time, as well as tool development so that we can make these methods available for lots of different use cases.
All right, well, thanks, Brittany. This brings us to the end of our workshop session. And so in just, you have 3 minutes before we start the next Tool Talk session. For those of you who are currently listening to me on Zoom, just stay where you are, and that's where we'll pick up Tool Talk, and we'll wait a couple minutes for the people to come over from the other concurrent Tool Talk session. And if you're interested in hearing Brittany again, she'll be talking again in about an hour. And so I'm going to turn things over to Chen, who will lead, I guess, the Tool Talk session 2.
All right, thank you. Is July here?
Hey, Jake, I am here.
All right, great. Make sure that you can share your desktop. Okay, go ahead, July. Okay. Well, there are people switching over, so I'll ask them to take a little while. Okay. So, July, let's wait for a minute. You can show your first slides, and then once I say start, and then we have 10 minutes to start, finish your.
Sounds good. I'll be off to the races.
Okay.
Can everybody see my screen?
Should be able to.
Okay, good.
As we're waiting, I want to make sure that our next speaker is also lined up. Chen, are you there? Okay, I see you. Darshan is there.
Yeah, I'm here. Yeah.
Brittany is there. Wayne. Okay. I see Wayne,
Hello.
you're mobile, but I presume pretty soon you'll be able to present on desktop.
Hello, this is Payne.
Okay, great. So we have everyone in this session. This is a two-talk session. Everyone will have 10 minutes to talk about a specific tool that they have in mind to share. The first speaker is July Wang. He's from the Informatics Institute. Go ahead, July.
Hey, Jake, thank you. So good afternoon, everyone. Hope you're having a good virtual meeting so far. So I'm very excited today to get a chance to talk to you a little bit about UBright, which is what we call infrastructure that we've been working on and setting up to help people do their data-intensive science, data-intensive translational research. So for my 10 minutes today, I'm just going to do very few slides to kind of set things up, give a quick overview, and then hopefully do a live demo that'll perhaps help current and prospective UBright users get a better feel for how you might engage the various abstractions of UBright. So at a very high level, UBright is definitely about three main things. So number one kind of bulleted point is we're definitely thinking about how to assemble clinical informatics and bioinformatics tools and be able to use them from UAB's very nice HPC infrastructure. Number two, it's definitely about setting up an environment to better help people do data analysis and data management. In particular, we have been exploring very heavily the idea of a commons design. So you may actually hear us refer to UBright as a translational research commons. If you haven't heard the term commons before, I pulled a quick definition out of one of the early papers, which simply defines it as infrastructure that co-locates data, storage, and compute with common analysis tools. So, and then finally, number three, as we set up this environment and infrastructure, we definitely are keeping a key eye on how it helps us facilitate team science. So that is how we can help the different members of a typical research team playing their different roles participate more fully in the research process. So kind of with the commons design in mind, if you look at the overall architecture of UBright, you can see we've organized it along three main abstractions. So there's a data portal, there's an analysis gateway, and there's an expo gallery. So if you imagine the maybe typical UBright new user that comes here, they may choose to engage the data portal first. And they might think to themselves, oh, I've got this omics data in flat files, maybe siloed off somewhere. can I put it in the omics data repo? Hey, I'm really interested in the clinical data repo, especially being able to programmatically query data out of the EDW and integrate it with my omics data. Or it might be, I've got all this great source code that I want to share, or I'm having trouble sharing with the rest of my team. Maybe I should think about putting it in the source code repo. Anyways, so whether you choose to engage one or all of these abstractions in the data portal, kind of the moment you do, you've accomplished a big part of the commons idea, which is to co-locate your data side by side with the biggest compute and storage here at UAB, which is the Chiha supercomputer that you see on the top right of this slide. And you're gonna, I think, hear more about this and research computing from Ralph Zottola later on this afternoon, but I just wanted to give a quick shout out to him and his group because we coordinate and partner very closely with them to do UBright infrastructure development. So kind of moving on to the analysis gateway, that's where we do a lot of our assembly of the toolkit. So certainly we think about putting popular data science and visualization tools here. But the way I like to talk about it is actually helping guide folks, UBright users, in thinking about their pipeline and workflow representations. So in past talks, you probably hear us talk about Jupyter notebooks and what those things are good for. For today, I'm actually going to try to talk to you about a class of tooling I call interactive research tools. So if you think about when sometimes I interview different research teams and I ask them, hey, what are your pain points? What kinds of things can I help you solve with IT infrastructure? Some of the time they come back to me and say, hey, you know, Jayla, I've got all this great code that I'm writing to do this amazing analysis, but I'm having a really hard time sharing it with the rest of my team, and especially getting their feedback and iterating with them because none of them want to fill out and change variables in my code or set up an environment to rerun it. You know, what they really want to do is be able to kind of point and click their way through interacting with my research tool. You know, how do I best do that? And so I'm actually going to do this example today with a new technology called Streamlit, which was actually released last October by the machine learning crowd. And we're excited about this tool because we think it solves some of our problems really nicely. And so I've got a Streamlit slide here, but the short version of this slide is if you think about the research team, the person typically doing a lot of the programming at that level is the data scientist. You may refer to them in your team as a data analyst or maybe a bioinformatician. But this person's very good at programming and but not very good at every kind of programming, right? So if you want to make a nice interactive user-friendly user interface, that's actually really hard. You know, front-end UI design and implementation is a hard thing, and you typically don't want your analyst, who's already probably pretty busy, having to spend time reasoning about client-server architecture and things like that. So Streamlit's kind of claim is that they can take your existing Python code and be able to instrument an analyst with just enough front-end capability and a simplified programming model to allow them to make good-looking, well-performing tools without having to go full-bore front-end UI engineer. And so this is kind of what we're excited about, because this is one way research teams can engage the non-programmers and other investigators in their team. So I'm going to switch away from slides now and kind of go to live demo mode. So just to orient you at a very high level, I'm just in my web browser and I'm showing a tech-oriented demo, but I've also got a number of co-authors on this demo from the GBM team, the glioblastoma team, and other teams that have helped me with realistic enough data. I'm not attempting to do a real scientific integration in here, but realistic enough kind of workflows that you can kind of mentally map and not have to use your imagination as much. So for the overview of what you're seeing in this demo, basically what's already happened is kind of in the left third of the slide. So I've kind of temporarily put on my data scientist hat, and I've authored some Python source code using the Streamlit app development framework, and I've iterated with the members of my team in the source code repo to kind of do the source code management and development. And once we were happy with what we had as far as the research tool, we deployed it to an app hosting infrastructure called OpenStack, which is very nicely maintained and supported by research computing. And that's actually what you're looking at right now. So I've put on the scientific investigator hat, and I'm now interacting with this running application. And I'm going to show you just a quick demo of how I can access different repos and access tools written by other teams doing methodology research. And I'm actually not going to go into each of these sections in a ton of detail. Towards the end of this demo, I'm just going to give you a link and invite you to kind of click around and play around yourself. So for the query clinical data section, all I wanted to show is that we're able to programmatically and securely query these demographic data from one of the GBM cohorts out, and you could possibly integrate it with the rest of the data you're assembling. The next section is just about parsing differential gene expression results. So a different part of the team, actually Lara and Christian for the GBM team, did a DEG analysis. And all I'm interested in is kind of pulling the list of differentially expressed genes from their analysis and kind of chaining it together with what I wanted to show in this research tool, which is actually a gene set analysis. So this is represented here by the pager analysis from Chen's lab. And they actually provide a RESTful API to be able to perform this gene set analysis. And what you're looking at here in this section is just the results from just the default parameters. And so you can imagine here, am I getting close to time?
10 seconds.
10 seconds, oh no. So you can kind of play around with the interface here and tinker dynamically with filtering the results. You can actually open this sidebar and set pager parameters. But basically, if you get a chance and you're interested in this kind of thing, you can play around. I wanted to just end on this link right here. This is a DOI record to this demo. And this is part of how our expo gallery works. We're trying to help UBright users think about how to get credit for their work. So this is a kind of place for you to store your research outputs where you can have tracking, automated tracking of the number of views and downloads you get. We've actually created a community called UBright where you can submit your research outputs, including software, pipelines, tutorials, posters, that kind of thing, and have it behind a stable DOI link. Unfortunately, since I'm out of time, I won't get a chance to go in depth about DOIs, but I think folks maybe are familiar with the idea from publications use of it. But I'll stop here. Didn't know I was going to run out of time so quickly, but hopefully I gave people a quick, reasonable taste test about UBright.
Okay, well, we don't have time for questions, so let's move on to the next speaker, Arshan.
Move to the right. Oh, right click.
I'm trying to share the screen. Telling tools disable the participant screen sharing.
I am trying to click on your icon and there's no
Try
way.
another one, see if it's.
So July, do you have to unshare yours?
I, to my knowledge, have already unshared.
Go up to the very top and click on
Okay.
it and remove those. I don't know why that's not working. Try more. Along to the right, maybe. That was working. Yeah, just working out for.
Or reclaim host. Oh, I see. Maybe click on this. Yes.
All right. There you go.
apologize. So go ahead, Darshan.
Okay.
10 minutes.
I hope all of you can see my screen.
Yes.
Okay. Good afternoon, everyone. Today, I'm going to present about a cancer analysis tool that was developed in our lab. So before I begin with my talk, I would like to thank the organizers for giving me this opportunity to present. So I'll begin with the motivation factors. So what are the factors that actually motivated us to develop this particular portal? So as most of you are aware of, the Cancer Genome Atlas has led to generation of huge amount of data from the samples of approximately 11,000 patients corresponding to 33 different cancer types. And this has led to multiple discoveries, including identification of new therapeutic targets, novel tumor subtypes, as well as understanding the molecular characteristics of the cancers. So this huge data is now public and has tremendous potential to uncover many more cancer biomarkers. And this data has been currently, or when we started the plan, there were many tools that provided easy access to data and also enabled users to do the analysis. But one thing that we observed after careful study, that none of these tools provided extensive, comprehensive gene expression and survival analysis on tumor subgroups based on race, stage, molecular signatures, histology, BMI, and many others. And this is what actually prompted us to develop a new tool, that is UALCAN. As the name itself suggests, the tool was built such that any cancer researchers from any part of the world should be able to use this web portal with minimal computational knowledge. So for the development of UALCAN, the first step was to obtain the data. So we actually obtained level 3 RNA-seq data, microRNA-seq data, and DNA methylation data from all 33 corresponding to 33 different types of cancers from TCGA. And recently, with the collaboration of Dr. Chad Triton from Baylor's College of Medicine, we also acquired mass spectrometry based proteomic data for 7 cancer types from CPTAC, Clinical Proteomic Tumor Analysis Consortium. So all these data were actually cleaned, formatted, and processed using open source R statistical packages and in-house Perl scripts, which led to generation of gene expression profiling, promoter methylation profiling, survival analysis, and correlation analysis for 20,500 protein coding genes, as well as 1,870 pre-microRNAs in 33 cancer types from the TCGA, as well as protein expression profiling for approximately 10,000 proteins in 7 cancer types from CPTAC. These analysis results are actually hosted in the web portal UALCAN, which was developed using Apache Server, Perl CGI, and HighCharts JavaScript. So we try to keep the design of the UALCAN as simple as possible so that most people can access the data and easily get the analysis result. So this is the snapshot of the analysis page of UALCAN. It basically has 3 panels. Each panel is designed to provide the output of different kind. The panel 1 actually allows user to obtain top 250 overexpressed and underexpressed genes for each cancer. So these are actually are ordered based on the statistical significance as well as the ratio of the median TPM values. So the panel 2 is actually designed for the users to upload their genes of interest and select a specific particular cancer, and they will be provided links to the different analysis set. So in this case, I'm just showing an example of a particular phosphatase, CDC256. So we have different analysis links. The expression link actually shows the user the box plot, which actually compares the expression level of gene of interest in normal and tumor samples. and also provides the statistical significance of the difference. So user can export the analysis result as publication quality images or PDF documents. So not only we compare the normal and tumor, but also, as I mentioned earlier, we are more focusing on the tumor subgroups. So user now can obtain the expression level based on stage, based on patient race, based on age, and all other things along with the statistics, which shows the level of statistical significance. And all this information can be easily downloaded, and most of them and many cancer researchers we have seen have been using this in their publication. So the survival link actually provides the information about both univariate as well as multivariate survival analysis that we did, considering gene expression alone as well as and gene expression in addition to race and gender and type also. So the methylation data that we got was a piece of processed data from Illumina bead chip arrays from the TCGA, where we are providing the methylation level of from only the probes that are associated with the promoter region of a gene. So by this, user can actually link the expression and methylation information if there is a correlation, and can understand how these expression could be regulated. So the correlation page will actually list the list of top positively correlated and negatively correlated genes, and user can also visualize this result as a scatter plot. And the pan-cancer view is actually enables user to analyze the expression of gene of interest across the TCGA cancers in both normal as well as tumor samples. So as I mentioned earlier, so this is the new addition that we have in the YOLCAN. Now user can also study the protein level expression for the gene from the CPTAC data sets. So the third panel that we have actually allows user to query the YOLCAN database using pre-compiled set of genes. So these were actually obtained from the KEGG pathways, and from UniProt database, we obtain the specific classes of genes, and user can use this and directly obtain information of the analysis result for each gene that are involved in particular pathway or gene class. YOLCAN was actually published in 2017 August, and recently the integration of the CPTAC data set to the YOLCAN and analysis were actually published in a Nature Communication paper. So one can find YOLCAN with the following URL, yolcan.path.uab.edu, and till now we have approximately 300,000 access visits the website from the cancer researchers from all around the world, and recently we have reached over 450 citations for the YOLCAN. So future plans, we actually think, I mean, we are almost ready with the link RNA data from the TCGA, and we are also in future thinking of including the copy number alteration data to the database. So I would like to thank all my colleagues as well as Funding University for the support for not only developing this tool, but also now maintaining it successful.
Great.
Thank you. So if I there again.
Anyone have questions?
Yeah.
So maybe I have a quick question. How do you keep up with the increasing amount of the cancer data, especially at the clinical side and also the single cell level? Are you planning to just keep integrating them or build API so that yours become a service to other more pipeline analysis? What is the plan?
As of now, the plan is to integrate with the link RNA, and I think we are also focusing now on including a lot more analysis tool where we will allow user to do a lot more integrative analysis rather than just looking at only gene expression and microRNA and link RNA separately. we are planning to build certain or at least integrate or any tool that is available so that they can do a comprehensive analysis, something like a competing endogenous RNA network and all those things. That's what the plan right now.
Thank you very much. Okay, so we'll move on to our next speaker, Brittany Lestain. Brittany, I've already shared the permission with you.
Okay.
All right, great.
Oh, well, let's see. Can you see it?
Yes.
Okay. All right. So today what I thought I would do is tell you about one of the biology questions that we had in the lab and about the R package that we're developing to help answer that question. And so just to jump right in, since it's a short talk, we are interested in genomic instabilities. And so these are molecular signatures of gross genomic alterations, and we see them a lot in cancer. So I've got a couple of examples here for you. So chromosomal instabilities are the altered number and structure of chromosomes. DNA methylation instability, or where when we have these altered DNA methylation patterns like the CpG island methylator phenotype. And nucleotide or genetic instability or increased mutational burden, also known as the hypermutator phenotype. And then microsatellite instability are changes in the number and repeats of microsatellites, which are these short, short repeat signatures. And so we're interested in these because they have key roles in disease etiology and are linked to clinical outcomes. So if you're a student who's still hanging around, or if you're a cancer researcher or clinician, you'll recognize this from the Hallmarks of Cancer and updated Hallmarks of Cancer paper. And it identified genomic instability and mutation as one of the enabling characteristics of cancer. So this is one of the biological processes that promotes cancer in the first place. So it's this key biological capability that's acquired during cancer development, and as I said, an enabling characteristic. It's associated with tumor biology, patient survival, drug efficacy. So when we look at these big databases like TCGA that you heard about in the last talk, or that I referenced in my workshop earlier today, for example, we see that across different cancers, whether or not a patient does well with a drug or whether or not a patient survives their cancer, there are these strong correlations with different genomic instabilities. So for example, and we'll come back to this here in a minute, is microsatellite instability high with PD-1 blockades. And so these gross alterations in one feature set result in consistent changes in others. So chromosomal instability, so changes in the C and V numbers, copy number variation, is also linked to widespread DNA hypomethylation, so loss of DNA methylation, and characteristic gene expression changes. And so what's interesting about that is that this gross chromosomal instability, when we see these DNA methylation and gene expression changes, it's across even different species. So we even see this in plants. So there's some conserved biology that's happening here. And so I got really fascinated by this idea. And, you know, I'm a biologist first and foremost. And so being a data scientist also, I wanted to say, okay, I want to take a look at what these chromosomal instability metrics look like across these large public data sets, as well as in clinical samples that we are analyzing from data we're generating here. So the problem with that, though, as you already see on the screen, is that there's no consensus on biological and clinical relevance of various chromosomal instability metrics. So you see up here an example of 6 different ones that my team and I identified in the literature, and they all show these different but potentially important correlations between different patient characteristics or biological features of the tumors, as well as these different calculations. And so what we've done is we've been looking at it across the Cancer Genome Atlas. And so here you can see for each of the different cancer data sets, You can see in blue the normal tissues that were associated with those patients, as well as the tumors. You can see for two of these different metrics, you can see that they're very different from one another. So the spreads, how they look across different tissue types, and their relationship to the normals are different. This is really just to kind of underscore the message that methods matter. So when you see folks say, you know, this particular drug is associated with chromosomal instability, what flavor or type of chromosomal instability matters a lot, because we see that these definitions are actually biologically different from one another. And to drive this home again, if we take in kidney cancer, if we take and look at two of them, so the number of breakpoints and the number of genes with a copy number variation, we see that they're only mildly correlated with one another. And we think that they're capturing distinct biological properties. So where the number of breakpoints is actually indicative of homologous repair defects, and the number of genes for selective pressures of CNVs with cancer driver genes. And we find that their association with different patient characteristics that we care about, like survival, are very different. So the number of breakpoints is strongly associated with patient survival, but the number of genes isn't. And there's also little consensus for other instability types as well. So we think this is really important because there have been recently approved drugs for tissue and site agnostic indications in cancer. So this is, the example here is for looking at immunotherapies. And so here what happens is that when there's mismatch repair deficiency, so microsatellite instability high, it actually is in the presence of anti-PD-1 or immunotherapies. It allows the T cells to kill tumor cells. So what's interesting here is that these mismatch repair deficiencies, these microsatellite instability high, are really a functional consequence of mismatch repair deficiencies. So instead of going in and testing every single member of the mismatch repair pathway to see if they make functional proteins or if they have variants that might be deleterious, we can just go through and measure and see, is there microsatellite instability? And it gives us actually a really good indication across many different cancer types of whether or not this drug might be helpful. So we're interested because other genomic instabilities have been shown to correlate with drug response. For example, chromosome instability high with enhanced sensitivity to cytotoxic therapies. We're interested in developing tools to be able to measure that across different data sets. So we want to assess the relationship between drug response and genomic instabilities and compare patient profiles. And so this would let us nominate more biomarkers. So we have an NIH-funded grant to do this, and we're working on how we can evaluate these different instability signatures and compare them within and across cancers, to identify features associated with signatures and characterize pathways, to evaluate the relationships between those signatures and clinical data, and develop classifiers to measure instability signatures from each omic data type. So folks who know me know I'm a big fan of looking at the transcriptome. And so we're working on ways where we can actually infer chromosomal instability from a transcriptome or methylation instability from a transcriptome, because it allows us a cheaper and easier way and a way to extract more information out of a given data set. So we're in the process right now. We've got kind of what I would consider an alpha version of this R package for instability signature analysis. And so what it does is it takes all of these different definitions, and we have functions that allow you to be able to go in and calculate each of these and then compare them and look at different biological relationships within your favorite cancer data set or other data set for that matter. So I mentioned that we used R, and since I had looked at the schedule and it didn't look like you were gonna hear from other folks using R, I just wanted to take a moment and talk about why we use R. So it's an advanced statistical computing and graphics language. It's really great for data manipulation, modeling, and visualization. We like it because it's cross-platform compatible. But really the biggest reason why we use R, I love Python. I learned originally to code in C. I code in half a dozen programming languages, but my lab primarily uses R is because of the vast and rich user community and contributed packages in Bioconductor and CRAN, which are these package repositories that include specialized tools. And the genomics community and the bioinformatics community for omics data have really latched onto R. And so if you're doing kind of bleeding edge genomics analysis, Those analyses are almost always first published and in R and as R packages, and so it allows us to kind of stay on the very bleeding edge. And it's written in a way that really helps for statistics, and the popularity has really been increasing.
30 seconds.
Okay, so we have this R package that, like I said, is an alpha package right now. Our plan was to have it out later this year. It'll probably be out sooner because we're planning on working on it from home now. And so if you're interested, get in touch with us, but be watching for it available on GitHub and CRAN sometime in the next 1 to 3 or 4 months. Thank you.
Thank you. So as we're switching over, anyone have a quick question? So if not, we'll thank Bernie again. Our next speaker is Wayne Liang. He's a professor from Children's Hospital. Wayne, go ahead.
Yes. One moment as I share my screen. Can everyone see my screen and hear me?
Yes.
Great, thank you so much. So as Jake said, my name is Wayne Liang. I'm an assistant professor in pediatrics. I practice pediatric hematology oncology, and I'm also in the Informatics Institute as a clinical informatics researcher. And today the topic of my talk is pediatric cancer data commons. So as some background, in the United States, over 1.7 million new cases of cancer are diagnosed annually. However, less than 1% of those are diagnosed in individuals under 20 years of age. And that is all types of cancers, from leukemias to solid tumors to brain tumors. In other words, pediatric cancers as a whole are rare diseases, and individually are all rare diseases. Because of that, institutions and individual investigators do not have enough N to be able to really move the needle in terms of scientific progress, and data sharing and collaboration are critically important in my field to be able to advance our knowledge of pediatric cancers. If you're interested, you can look on, you can track this hashtag on Twitter for folks who are working in the space. So thankfully, pediatric oncologists have really developed a culture of collaboration. In the North America, United States, Canada, as well as Australia, the leading consortia funded by the NCI to perform pediatric cancer research is the Children's Oncology Group. And largely through the success of COG, about 50% of pediatric cancer patients or individuals with pediatric cancers in the United States are enrolled on clinical trial, compared to less than 5% in the adult space. Similarly, there are other similar organizations in other countries, including SIOP, led by French institutions, the BFM group, led by German institutions, and the CCLG, which is a UK-based group. And there are other groups like that in Japan and the Middle East and South America and elsewhere. These are very large consortia. The reason for their existence is so that we can pool all of our resources and our patients so that we can actually conduct clinical trials comparing regimens. And it's largely due to this collaborative research that we've been able to move the needle in terms of survival in pediatric cancer. The blue bars here represent survival rates in the 70s, and the yellow bars represent the same survival rates but in the early 2000s. And you can see a significant increase in survival, largely because of collaboration between institutions. However, more needs to be done. So not all cancer patients do well. In certain diagnoses, we have over 95% survival, but in other diagnoses, we have 0% survival. And certainly the diagnoses that are the rarest are the hardest to study and have the worst survival. And in our world, tissue samples are limited. So first of all, we have less patients. 2, the specimens we obtain are smaller because the patients are smaller. And 3, there's a higher ethical bar to overcome if your initial biopsy or specimen collection was insufficient. So whereas in adults, you may not think twice about having them go back to surgery to get another biopsy of their brain, for example, we would be very reluctant to do that in pediatrics. So every single specimen we have is very precious. It's very important that we share our data and our specimens widely. Unfortunately, childhood cancer data are fragmented. They often live within the silos of different research consortia, and sometimes on individual researchers' laptops or an Excel file somewhere in a statistician's desktop. They're not broadly available, especially outside these consortia, and they lack standardization. If we're able to provide data in a shared interface, data commons, if you will, as Jay-Lai went over earlier, a place where data and analysis tools and compute can live in the same place and make it available to the community. That way we can maximize the value of the existing and future childhood cancer data. And that leads us to the Pediatric Cancer Data Commons Consortia. This is a major effort funded by the St. Baldrick's Foundation, a private entity that is a major funder for pediatric cancer research, and is led by Sanford Chen at University of Chicago, but is really a major undertaking by the US and Canada pediatric cancer community, and having involvement from other consortia internationally. This slide just reviews what a data commons is. Given that Jayla went over that earlier, I will skip through this and talk specifically. In the PCDC, we are collecting data and data dictionaries for various different pediatric cancer types. So the cancer types that we're collecting is primarily clinical research data. We're not focusing on genomics or biobanking data or imaging data at this time, but the way that the data commons is constructed is meant to be able to interact with other data commons through an API model. And on the left, you can see here the type of diagnosis that we have currently collected from different leukemias, so germ cell tumor, Hodgkin's lymphoma, neuroblastoma, osteosarcoma, Ewing sarcoma, and soft tissue sarcomas, which is primarily rhabdomyosarcoma. And within this commons, the data will exist together and then be able to drive the data platform for analysis in the cloud. I want to mention that this is very significant, not just from a technical point of view, but also from a policy point of view, for different consortia to all agree to deposit the data in the same place, including consortia across the globe from different continents. That is the effort that we're undertaking here. And in addition, we need to harmonize the data dictionary so that we can have common data elements so that we can compare, for example, acute lymphoblastic leukemia results from North America compared to regimens used in the UK and Germany and Japan. In order to do that, we need to harmonize the data elements used to collect the existing data and to collect data going forward. Not only that, in the age of precision medicine, where the site of cancer is not as important as a mechanism, we need to be able to compare responses to therapy across cancer types. So comparing a leukemia to, for example, a brain tumor or a soft tissue sarcoma, we need to be able to speak the same language, if you will, so to speak, in the terminologies. And so the work that I am primarily involved in with this project is to harmonize the data elements with between the different disease entities, and going forward also harmonizing them across consortia so that we can compare data going forward. So current progress, the data from the International Neuroblastoma Risk Group, which is an international consortia, that data has been deposited into the data commons and is live, and I'll show you a screenshot that soon. So investigators now can export that data. Now we're working on ongoing data dictionary harmonization between the various different diseases data that have been contributed to the project, and also working on common or a data governance structure so that we can govern how data is contributed to the commons and how to handle data requests. As you can imagine, data come from the UK or European Union have different requirements, policy requirements than those contributed by North America, and so on and so forth. And so that is being ironed out. Working towards a minimal viable product to enable unaffiliated investigators to be able to start doing analysis on a platform. And this is in close collaboration and integration with other NCI-funded efforts, including the Center for Cancer Data Harmonization, the NCI Source effort, and the NCI Childhood Cancer Data Initiative. And I will also add one benefit of this living on the University of Chicago infrastructure is that they can very quickly do an API call to the genomic cancer data commons to be able to pull relevant genomic data for and connect it to the clinical data in this commons. So here's a link for what is already live, and I will post this also in the comments for everyone to see. Instead of doing a live demo, I just want to show you a screenshot. So anyone today can log in, request an account, and start performing queries. At the right, you can see the 4 different consortia that contributed data to this commons, and then you can start filtering based on common data elements. For example, cancer stage, MIC, MIC status, treatments that they have received, and do cohort identification and preliminary counts analysis. And then if individuals would like to get line level data, they can request that. And so the future vision is that any qualified investigator would be able to register for use and be able to do preliminary analysis with a limited data set and access or request access to deep final level data governed by the executive board in the contributing data source. With that, I'll pause given time, and I'll thank you so much for your attention.
Okay, we're exactly on time, but let's just see 30 seconds, one question. Anyone? Okay, well, I guess people want to take a break. Let's take a break and come back in 10 minutes to start at 3:10 for our next session.
Hey, Rob. This.
Hello.
Hi. Yeah, I can hear you now. Yeah, we have a.
Unmute.
Yeah, we have a 10-minute break. So after that, we can start. Great. So I have made you as the co-host so that you can share the screen.
Okay. I think I'm ready to go.
Zechen, can you hear me?
Yes, I can hear you.
Can I test my computer once quickly before Rob speaks in this break?
Sure. I will make you as co-host as well. And I'll also notice Steve is here, and I will grant him the co-host as well.
Can I start sharing? Let's see what it was.
Sure, go ahead, please.
Right, I'm not sure.
Yeah, I can see Rob's slides already.
Okay, all right.
Yep.
Perfect.
I figure I'd start with the video from my bunker.
Hey, Ralph, nice bunker.
Yeah. Well, the president just declared a state of emergency out there, so I figured, you know, it'd be useful to be in the bunker.
No corona in the bunker.
Nope. I was debating whether to use a bunker or put on a Hawaiian shirt and have waves crashing onto the beach.
Malay, can you share and test a little bit?
Okay, let me test that, okay?
Yeah, go ahead.
I'm sharing now. Okay. Can you see my presentation?
Yes, yes, I can see that. Perfect.
The human genome screen, right?
Sorry?
Human genome screen, right?
Yeah, yeah.
Okay, all right. Yeah, we're good to go.
Perfect. And Steve, I have granted you as the co-host as well. So if you like, you can share your screen for a test.
Okay.
Thank you. Ralph, can you stop sharing for a moment and let's see if Steve's screen sharing will work.
I'm sorry, I didn't get your question.
Yeah, can you stop sharing right now? And I would like to see.
Oh, sure.
Steve.
Sure.
You want me to stay? Okay, let me go ahead.
Yeah, Steve, you can share. Yeah, perfect. Yeah, I think it works.
Yeah.
Perfect.
Yeah, I've been doing this for a whole course. I had people coming to take the course I'm running this semester from outside the university in several places.
Okay, nice.
Yeah, it works fine for us.
Good.
So you can go back as you need.
Molly side, she will be late for a while, but I think she will be here very soon. So our talk is 20 minutes. Please control your time a little bit. So I will remind you and at 15 minutes and 18 minutes respectively if you have not finished. And we will leave 2 to 3 minutes or 2 to 5 minutes for Q&A.
Okay. Yeah. No, I'll go quick. I'm intending. I wasn't quite sure who the audience would be, so I've kept this high level just to be of general information to everyone. So I will go fast.
Ralph, are you by any chance in a bunker?
It's a virtual bunker.
Oh, yeah,
I thought it would be appropriate given everything that's going on today, and unfortunately, I've not been able to attend most of today because I have been in meetings all day long doing business continuity planning, you know, for remote work options and all of that sort of stuff. So, you know, that, and I was contemplating wearing a Hawaiian shirt and having a background of waves crashing onto the beach, but I figured the bunker is probably more appropriate today.
Yeah, it looks like a real bomb shelter.
No, so in Zooms, so here's an in-service training at the moment. On the bottom left, where you have the ability to mute your phone and start and stop your video, if you click on the arrow for the next to the play video or stop video, you can select a virtual background.
Oh, okay.
And,
Yeah.
so you can change it to any number of things. So for the first meeting that I was on at 6 o'clock this morning, and we were all in different locations, I Googled Prepper's bunker, and there it is. I've been using it all day.
Yeah, good one.
Is anybody on site, or is it completely remote today?
I think completely remote. Even in the Informatics Institute, we stay at our own offices. So yeah, I think we can start now. So our first speaker is the Professor Raf Zotarla, and he's the Assistant Vice President for Research Computing UAB. You can go ahead, Raf.
Are we starting right now?
Yeah, we can start now. Sorry.
Okay. So thank you, and I'm happy to be here virtually, actually from my virtual bunker even. Let me share my screen. And today what I'm going to discuss is what we've been doing here at UAB. General overall big picture, I call it the research computing system. And really what our objective is to work alongside the research community here, building gateways to science. Let's see. Start with the requisite disclosure. I have a pretty simple life. So why research computing? In the early days in science, you know, we had theory, we had experimentation. Today, and as I'm sure we've heard from many of the presentations today, we can't do any science without significant computation behind it, whether it be the type of research going on, large science, the amount of data, et cetera. It's really gone to an extraordinary scale with potentially extraordinary opportunities for discovery. And the other thing that I think has evolved over time is the fact that it really is a collaborative endeavor. It requires people across multiple disciplines. I sometimes joke about the fact that, you know, we've heard the phrase a lot of times that it takes a village, and I always say that's not enough because villages often tend to be very homogeneous. We really need a very diverse group of people. I think that's clearly evident by the type of people that have been presenting here today and people in attendance. We come from a lot of backgrounds, and although there are some things that we share, there are many things that are unique, and it requires all of our expertise working together to make things happen. So why research computing as one of these things? Well, For one thing, as the need for compute has grown, so has the need for computers, and they have gotten more complex. The photos in this picture here, this little screen where it says less of this little image, is actually from someone who wound up being very high up at the National Science Foundation. What you can't see in this is that he's got a cluster sitting here, some box fans moving air to window air conditioners. And what we're trying to do is get away from this and go to this. And we just heard in the previous talk about creating repositories and sharing data. We can't do it if everything is in a silo. The amount of computing that people need, there are plenty of studies that show that the local clusters tend to be very underutilized. So it's a lot of money that we're spending for very little output. In contrast, shared resources tend to be heavily utilized. So you're really getting a return on your investment. Our cluster currently at UAB is probably it's generally in the 80 to 90% utilization. So we're taking full advantage of our investment. You get somebody else to manage it, swap drives, install software, making sure it's secure. I think having this resource available, when we put it in our facilities descriptions, et cetera, I believe it makes us more competitive in our proposals. And the real measure from my perspective is if it enables us to do research that would otherwise not be possible by having this resource here and available on the day a faculty member steps foot on campus, I think that's really important and a huge success. Some of the rigor that we put into running this, why it matters, sometimes things happen. And this was from last summer. We actually had the data center explode. Not really completely explode, but this dehumidifier circuit caught fire, set off the Energen smoke fire suppression system. The pressure blew out a wall in the data center. We had an emergency shutdown while all key personnel were in another state attending a conference. So the good news about all of this is that because of all the protocols we had in place, the documentation, we were able to engage teams from facilities, the staff that were here, teams from enterprise IT, and had graceful shutdowns. We were up and running partially within 8 hours and fully functioning within 24 with no data loss. I've been in scenarios where a pipe broke over the system that was in the lab closet, and that was not recoverable. So I think this matters. When I talk about the research computing system or research computing platform, one of the things that I'd like to shift from here is that I think for a little while here, when people think of research computing, they just think of high performance computing. But really, research computing is a lot more than that. We're still in the early growth stages, early maturity stages, if you will, of what a cutting-edge research computing group can be. But I think you'll hear in the next couple of minutes that we've made some great strides in a short period of time. And I think one of the main reasons for that is the fact that we get to work really closely with the research community here on campus. And people are I find that people are very open about telling you what they need and how they would like things to work. So it's moving along quite well. Our system consists of several components, and really all of them focus on our goal of trying to enable data analysis, data sharing, and scientific software application. A component to talk a little bit about is the high performance computing system known as CHIHA, named after the tallest mountain in Alabama. Some work we're doing around science gateways. Something that's been relatively new, we call it the Ruffner Cloud. It's an application development environment. And the underlying all of this, because if nothing else is constant, this is one thing that always is true. We're generating more and more data every day. So we're taking a very strategic approach to how we handle that data. It's becoming a serious endeavor, I'll say, because we actually have a real logo, a new logo these days. Apparently, what was originally done, which was to put a piece of tracing paper over a picture of the Chiha mountain range and take those squiggles and make those our logo, didn't cut it. So now we have something that professional graphics artists put together. So I think it's pretty cool. And our cluster is what I would call a mixed use environment. And by that, we have several different types of hardware configurations in place. We've got standard type of compute nodes, high memory compute nodes, GPU nodes. All new equipment that's going in is going in with a very fast InfiniBand networking fabric behind it. The critical item becomes moving the data to and from disk when you have a lot of processes. Our system, even though it's currently the fastest supercomputer in Alabama, at least at academic sites, it's still a pretty modest system. The university does have aspirations to deploy a top 500 system, and I would love to see a statewide consortium occur where we pull together research computing resources and infrastructure from the leading research universities across the state. I came from a place that did that, and it has given those institutions a huge competitive advantage, and I think we can do the same here in Alabama. Currently, we're about a half a petaflop as far as our computing power. And it's gotten extremely popular. This slide is from last year, and excuse me. The thing that I find most exciting about this is the breadth of use. When I started seeing departments in the list, like the Department of English, Philosophy, Media Relations, facilities, it shows, again, going back to the first slide, that computation has become an essential part of just about everything we do. This makes it a lot easier to justify resources on campus because it's not something for what I used to call myself, a techno geek. It's not just someone in the traditional sciences, it's really across all disciplines. And I think the widespread adoption has really increased dramatically since we deployed late last year something called Open On Demand, which was developed at the Ohio State Supercomputing Center. Open On Demand essentially gives everyone access to the compute resources via a very familiar web page, a web portal. And we've got a number of different applications that are embedded in and accessible from within the web page directly. And you can also start an interactive desktop session. So if an application is not yet webified or available in the portal, you could just run your desktop, standard desktop within the portal. What that's done for us is moved us away from having to do support around how to log in, making sure you've got any one of a half a dozen different packages installed on your computer and all having the exact right configuration, to just saying, here's the URL, log in. So the team is able to spend a lot more time working with you on research problems rather than technical problems. And another big priority for us has been to get the environment approved for analysis of HIPAA data. And we went through this process. It was a 6-month process, although I think now that it's been done once, it'll be a lot faster going forward for other systems that we want to add. But I think it's really important. A significant amount of our research is clinical research, and at this point now, it's no longer just looking the other way. We are authorized, or you are authorized, to analyze your IRB-approved data sets on the cluster. And we're working with the IRB office to add a checkbox on their form to make things go a lot faster through the review process. We've already done a couple of in-service sessions with the IRB so that they are aware that when you as an investigator submit a protocol and say you want to use this environment, they don't have to call up and ask. They're already aware that it's approved for use. Another tool that we're in the process of implementing is something called XDMod. It's essentially metrics on demand. I find this really exciting because for us, this is our data science world. We have a rich amount of data on how the cluster's used, the jobs that are running, that sort of thing. And with this kind of environment, we're going to be able to predict, number one, do data-driven decision-making about what type of computers do we need to add in the future. Do we need more large memory? Do we need more GPUs, et cetera? It's real-time data. But additionally, it allows us to monitor performance. So we had a group that wanted to buy dedicated hardware to add to the cluster. Now, I'm not against taking your money, but when we looked at this, we realized, looking at the performance data, that it really had nothing to do with the availability of resources on Chiha. It was really the code base. So we worked with that particular group on a couple of the scripts that they had going, and it turns out that now their jobs are running and starting just fine and running quickly. So that need to spend money on hardware as opposed to on the research is not there. And we hope to be very proactive in the use of this tool, and rather than being in a reactive mode. Science DMZ is a network architecture that's in place to move data very quickly. It's allowing us to create 10 gig links to various instrumentation labs and move the data to the cluster, as well as to connect to collaborative sites around the country. So if you're engaging in that type of activity, give us a call and we'll work with you to create a link a network link that allows you to move data at a very high speed. Globus is another part of that. Globus allows you to transfer files in an easy and secure way, share files. But for a lot of the people that you heard from today, you can incorporate this into your workflows via the programming interface and make it fairly straightforward to move large data sets. Final piece is something that we're calling the Ruffner Cloud. Ruffner is in another mountain range in Alabama. My team is full of hikers, so I think this is going to be a common theme. This has gotten a lot of use by many of the people you've heard from today to develop scientific applications. It's essentially something like an Amazon or a Google Cloud, but on campus. So you can build things, test things, tear them down, make mistakes without incurring huge costs in the cloud. And it's been so popular, it actually got submitted as a priority item in our upcoming budget. So I'm hoping that we'll get approval for that to be able to deploy a production version. So all of the applications that you develop we'll be able to host for you as well. The neat thing about this too is that it's given a standard environment for a lot of the application efforts that are occurring here. So you're gonna, I think one of the things we're gonna see as this develops is that what's developed in one domain can be applied or modified and easily adapted in another. So the holy grail of data sharing, data integration, that sort of thing will be much more possible working together. Storage, since I'm running short on time, I'm just gonna just say on storage that we're creating a tiered architecture of storage. The high-speed storage connected to the cluster, which is very expensive, nearline for things that you use frequently, but are not actively being used in the high performance computing mode, and then an archive pool for things that you just need to keep for a long time, and that's what often is referred to as cheap and deep. And there is a significant cost difference and performance difference between them. So having a well thought out strategy on campus for how we do this, how it ties into other data management type of activity is going to be critical as we generate more and more data and start to link it together. Shout out for the Data Science Club. This is one of the other benefits that occurred from the transition with XDMod. People are talking amongst themselves in a virtual club every Friday to solve problems.
Rough 2 minutes.
So, what was that?
2 minutes left.
Okay, well, I'm done here. So all I was going to say is our goal, you know, Darwin said that the survival of the fittest, it's actually survival of the most adaptable. And I'd like to think that as a research computing group, we're very adaptable. I hope you think so too. And here are some links to get more information. And I will stop there.
Thank you. We have 1.5 minutes for questions. No one's typing. Ralph, I think we are everyday users of HPC, and HPC really helps us a lot. We have a fabulous staff. They are extremely helpful. But I found there's many new users. I think you need to provide some trainings for the new users to get familiar with the HPC a lot. For example, job scheduling and all of this stuff, so that they can be a good citizen using the HPC.
Yeah, we do have regular training sessions, and the other thing that I'll add to that is any group on campus, if there is interest in having some sort of custom tailored training for your particular area, if you help us gather the people on your end, we're happy to come to you and cover any and all topics as needed. We're also starting to put a lot of these things online, like the Data Science Journal Club as an example. All of that is on a YouTube channel and a Twitch channel, so everything we're doing is going to be available on an as-needed basis.
Okay, cool. That's very helpful. So we have to switch to the next speaker, Dr. Malay Basu, Assistant Professor of Pathology. And Malay, can you share your screen? And you can start. Thank you. Thank you, Rob.
Can I see the slides?
Yes, I can see. I think
Can you hear me properly?
Yes.
All right. So I will very quickly describe this work that we have been doing for quite a long time now. This is one of the major emphases in my lab. So I already talked about this, some part of this talk before. People who know what I've been doing know this work that I presented earlier. So today what I'll do is that I will present some new results that we have got out of this, but let me start and we'll go very quickly. I will skip most of the technical aspect of this work because this work is huge, humongous. You won't be able to do justice to it. So I'll skip most of the technical aspect of this work. If anybody's interested, can contact me later and I'll provide you the necessary materials. All right, so the work actually started long time back when I was a postdoctoral student when human genome came up in 2001. And in that paper and in subsequent papers with a lot of commentaries and other review articles, people started calling it metaphorically as a book of life and the language of the gene. So these sort of sentences were getting used most in metaphorical sense by most of the reviewers and the commentators. So that's the time. So the book of life was mainly compared with like a manuscript, and like for example, this Voynich manuscript is a very mysterious manuscript that is now thought to be sort of a joke by Francis Bacon. but people have been trying to decipher what this means. This particular manuscript was identified as a scroll, and there are a lot of weird plants and other creatures in this manuscript, and this is written in a language that nobody knows. So people have been trying to decipher this language for a long time. So when human genome came up, people started comparing it to the languages, and then this seminal paper was published by by David Salz. He's now sort of an emeritus professor in GlaxoSmithKline pharmaceutical company. And in that, he described in this particular paper that the linguistic tools are very important in understanding, and there is a language of the genes. So let me skip these highlights. So basically, the idea is that this genome is basically like a book, and the individual protein sequences on that is basically like a sentence, and the amino acids are basically like alphabets. And then the protein domains, these are domains, those of you who do not know, domains are basically the unit, the structural and functional unit of proteins. These are specific regions that can be identified using HMM and other methodologies. These are units that get reassembled and recombined to create new proteins. And there are databases available like SCOP, PFAM, there are several databases. So the domain can be compared as words. So given this analogy that this genome is like a book, proteins are like sentences, and the words are like domains, in that paper, the Salz compared the domain is actually like a word. So what he did is basically compared in these, if you see my pointer, this is a graph where the y-axis is the frequency of individual word in Shakespeare's Romeo and Juliet, and domains, protein domain is genomes. The y-axis is the frequency of the word, and in the x-axis is the rank order of the frequencies. This is a typical way a particular plot is drawn, and we'll come back to it. It's called the power law graph. And what Salz showed very strikingly that this distribution of word in Shakespeare's Romeo and Juliet is almost identical to the distribution of domains in East genome. So basically this graph, what he was talking about is basically a power law distributions, and power law distributions are generally recognized as a straight line plot, and we'll come to it. And this slope of these, if you take log of the x-axis and the log of the y-axis, it becomes a straight line. And the slope of these is basically the parameter of this graph, and this is determined by this parameter called gamma. So power law distributions I am not going to discuss. It's been like a golden ratio has been identified in many different cases. That is generally identified as straight line log-log plot. So if so what I did is that after reading that paper, we actually did it for the human genome. In this case, this is the domain distributions in human genome as a raw count, and then the log-log plot on the x and the y-axis, and it has a straight line slope. So basically and I calculated it to be, it is minus 1.013 as the gamma, or the parameter of this power law distributions. And it turns out natural languages also has the same slope. So given this, a particular technique that generally used very commonly in linguistic analysis is called n-gram analysis. Now what n-gram analysis is this, that if you take a sentence, in this case, the protein sequence, and these are domains A, B, and A and B. And what it does in n-gram analysis, it actually takes the pairs of the words and calculate the probability of a particular word given there is a previous word. So for example, it calculates the probability of the AA and AB and the pairs of words like that. And if you take these are also called n-gram analysis. If you take 2 words, they are called bigrams, 3 words called trigram, 4 words called tetragrams. Though there are many different ways. The simplest one is basically the bigram, or 2 words at a time. If you take the distribution of these bigrams, and it turns out that they also follow a power law. So in this case, there are 4 different types of genomes as shown, and this is the slope of this graph is basically minus 1.8. And it turns out this is identical to natural languages. So this is shown here. So far, seeing that the domains and the words have typical the power law distributions of both unigram. Unigram is the single word distribution, and the bigrams have this similar power law distribution. So domains and words are mathematically very similar. Now, n-gram language modeling has been extensively used to identify the grammar of an unknown language. So what n-gram really is basically, I've already discussed, is basically the probability of a word given the previous word. And a typical example of this is extensively used in case of the spell check, in case of automatic filling up or autofill properties. Like, for example, in this case, the Google search engines, if you type Birmingham, it will most probably give you all these other auto-completion parameters. And this is calculated based on the probability of news given Birmingham as a previous word. And it turns out you can actually calculate the entropy of going from a completely random distribution of words in a language. And so basically, if you take Shakespeare's Romeo and Juliet and you completely shuffle it, you can calculate the disorderness, the entropy, and then you put them back into the proper text, a meaningful text. You can actually measure the difference of the information gain or the loss of entropy. And this has been extensively used to identify an unknown language, an unknown script, whether this is a language or not. And it turns out there are 17,000 languages in the world, and any human languages actually has a constant parameter of 3.6 bits of information gain, regardless of whatever the language. So this is used as an universal constant to identify whether an unknown script or an unknown book actually determines language or not. So given this parameter, what we did is that we actually tried to identify whether similar parameter exists, and universal constant exists or not in case of the genomes. And so we took a large bunch of genomes from different idea, and we actually calculated the differences from going from a completely random distributions of the words or the domains, and then we calculate the difference between how much information is actually gained by going from this completely random distribution to the exact genome that we currently have. And so basically you have now the entropy, the two different types of entropies. These entropies are the complexity measure of going from, you can calculate for the single word frequency distribution as well as the bigrams. And if you do that, you basically have these different criteria of different, I'm showing only the three major super kingdom of life, the bacteria, archaea, and eukaryotes. You see they actually differs quite a bit. The unigram entropy in this case always has a positive slope, but the bigram entropy on the other hand actually has a sort of a negative slope in case of the eukaryotes. And this is shown here. In a graph, you can ignore completely all the other peaks. What is important for us to know is this cyan color peak. This is the differences going from the unigram to bigram entropy. Now remember, unigram entropy is a completely randomly shuffled genome, and bigram entropy is the restricted version of it that actually makes biologically meaningful sense, or biologically meaningful combinations. So that means that the differences actually determines what the biology of the system is. So these differences, if you now plot for different subkingdoms of life, you will find what is important for us is the cyan parameters, that the differences of going from a completely random genome for the existing to the existing protein structure. And it turns out we identified 1.2 bits to be a sort of an universal constant across all subkingdoms of life. The only two major differences that we find, one is Crinoarchaea. This is basically a sub-branch of archaeal genomes, and human. Human is this peak. And so what we actually conclude from this, that there exists a 1.2 bit differences that is universally recognizable information gain across all genome in evolution, except two instances where the human being, where it's the complexity of the genome is the highest, and the crinoarchaeota turns out to be the most simplest genome of all. So with the exception of these two extremes, we generally have a constant information gain. Now, if you took, we can actually use this to measure how similar two genomes are based on these grammar. Now, these n-gram probabilities can also be called, it is basically reflecting the constraint of the genome, and thus reflects the grammar of this language. So what we can actually determine is that we can actually determine the probability of a genome given another genome. That means this is the probability of a genome coming out given the bigram models from a reference genome. And that reflects the how close two genomes are. So the closer the genomes, the higher the probability. So this measurement is also called the cross entropy. So if I show these graphs, so these are basically showing how close two genome languages or these bigram models are in several branches of life. And these are measurement of the cross entropies. Cross entropies are basically the probability that I described. Is the probability of genome given the bigram model from another genome. So we can actually do some mathematical trick of this and convert these cross entropy values into a distance matrix. And we can plot this graph, and it will show this near perfect separation of the existing taxonomic tree. That means this language is no longer genomes are not languages, and it has a constant universal entropy. It also follows the evolution of genomes across the tree of life. So given this methodology, we actually now ventured into what are the identifiable phrases in these languages. That means these are combinations, the bigram combinations that are specific to a specific taxonomic branch. We can consider them as particular phrase. So we use a technique called partial least square differential analysis to identify features. So the features are basically the bigrams that can separate two taxonomic branches or two clades. And when we can select the bigrams that are most informative in differentiating this specific clades into specific clusters. And if we do that, we find that individual functional, these are bigrams on the y-axis, and the bar represent the weights in specific clades. And we find that the specific functions are very, very important. There are well-known functions for an origin of specific clade are actually represented by this. So that means that this technique can identify features that are already known, biological features that are specifically important in specific clade. Like for example, the eukaryotic versus prokaryotic, everybody knows the ubiquitin and spliceosomes are the two features of eukaryotes. And we find that those are informative branches, like the same way the bacteria versus archaea, the cell wall actually is very important. In archaeal branch, for example, heat shock or the heat response proteins are very important. And the kinases are very important in plants. The cell-cell addition and hedgehog are very important from transition from fungi to metazoa. Metazoa are the animals. That means that the origin of animals are mostly dominated by cell-cell addition and the hedgehog genes. So this that means that the technique can identify specifically the specific bigram combinations that are important in specific clades. So the conclusion of this work was basically there exists a quasi-universal because we have these two exceptions. One is crinoarchaeota and human. We call it as quasi-universal grammar. And this value is 1.2 bits that are minimal complexity that is required to maintain a functional cell. So this work has already been published in 3 of my papers. People can follow this. The last one was published last year in PNAS. So what we are doing now, and this is the unpublished portion of the work, is that now we are trying to determine the actual parts of speeches of these particular language. And this is done by large-scale modeling of the protein family databases. Like in this case, I've shown a small toy example of the probability tree of cyclin proteins in Panther database. Panther is a protein family database. and identify these models that actually can identify all the proteins in the panther genome. This model can identify all the cyclins. So what we are trying to do now is to basically trying to find out the linguistic properties, particularly the features of the languages that is originally proposed by Noam Chomsky. The specific properties are called Chomskyan properties. We are trying to identify the Chomskyan properties of this particular language. We are also specifically trying to target two specific biological problems. The one is the origin of multicellularity, as we can actually, I already showed that we can identify specific phrases and we can cluster specific clades together. So for example, in this case, we can identify the bilateral animals. These are true multicellular versus all the all the unicellular organism. On the other hand, we can identify specific features that are origin of multicellularity. Now, multicellularity has come up several times independently in the tree origin of life, as I've shown by these red arrows on this tree. And there has been no universal properties of multicellularity that has been identified so far. That means that the plant multicellularity is completely distinct from the animal multicellularity. And the major multicellularity is known to be the animal multicellularity here, shown in the bottom red arrow. So what we're trying to identify is the universal feature of the multicellularity using these linguistic properties of the genome. And the second biological properties that we're trying to address now is basically complexity of individual human tissues and whether they speak a specific dialect of this language or not. And here is shown, and we use a mathematical trick for doing this. This is basically a language of differences in human tissues, and we see that the liver has the highest amount of information gain across the human tissues. So we're trying to identify this. We're trying to identify this dialect in human tissues as well as the abnormal stages like cancer. So okay, I'll stop here. Just acknowledge my collaborators here. This work was mostly done by Ligia, Deepak, and Diego in my lab. This work is strongly done in collaboration with Eugene Conant at NCBI and Yuri Wolf as my mentor, and this work was largely funded by the UAB Health Science Foundation grant. So I'll stop here for any questions that you might have.
Thank you, Malay. I think we have no time for questions, and I haven't noticed that anyone posted any question on the chat box. So I think anyone have any question, you can shoot the email to Malay and ask for more information. Let's switch to Dr. Barnes, our next speaker. Dr. Barnes is a professor of the Department of Pharmacology and Toxicology of UAB, and he will talk about the pathways to success in biomedics.
Okay.
Yeah. Okay.
So get to the beginning.
Yes. Thank you.
Okay, let me go to full screen for you. Okay, so I'm going to talk a little bit about my experiences in getting bioinformatics. I was funded when I first came to UAB in informatics by the NIH, and this was back as computers were coming into medicine. And so I've had a lot of experiences and some thoughts. So one of the questions is, do you merely provide a service or do you create one that can be used by others, and how do you go about doing that? So if we go back more to the memories of the people who are listening to this call, if we go back to the mid-1980s, it was when we started getting to look at not just one item at a time, but everything at one time. And we thought that was entirely cool. So we could measure the entire ohm. In this case, we're looking at a microarray. And the problem was it generated too much data. And computational methods were needed to map the results back to some form of knowledge base that we had to maximize how we could use it. Many of the biologists...
Your slides are not advancing.
Sorry?
I don't think your slides are advancing. We're seeing not the presentation view.
Okay, then that's because I went to full screen. So let me go back out of full screen. Okay, can you see them now?
Yes.
They are. Okay, well, you'll miss the emotion, I guess, that's associated with these slides. Okay, so I'll try and do it from this particular method. So there were too many data. Biologists were not experienced enough to write the necessary programs, and yet there was a need to do it. And it led to computational scientists coming in to work on the data and provide the links. So the scientists were looking at the data and not able to fully interpret it, at least on the time scale that they needed to do so. The computational scientists would send results back to the biologists. And I have had many occasions where the information that came back, it seemed to make sense, but then something changed in the way the data analysis and the result that came back was different. And so this was very confusing to the biologists. What's really needed is for the two groups of people to get much closer in terms of their overall language and their appreciation of each other. And we had a meeting at our national meeting to discuss this issue of respect between the disciplines. So that's a problem. The other problem I've had is when I run national workshops, as I've done here at UAB, and want to take people through the computational work that's needed in order for them to understand their data, we run into what I call the Goldilocks effect. And the problem is that the people in the group, I was teaching 40 people at a time, and they had very, very wide ranges of capability. They all wanted to learn, but had wide ranges of learning ability. And what we learned was we shouldn't mix the trainees into one group, because one group will almost always fall behind fast, and the others will be bored if you slow down enough. And I've learned this in school when that we graded our groups into different groups, so we taught appropriately. When you do this, one of the things that happened to me was at age 8, I'd learned to read far better than anybody in the school, and I had become a pest. And so the smart teacher had me training the students who hadn't learned how to do this. So I think in the format, we need to have smart students to train the more senior faculty, particularly the ones who want to learn, because their ability to learn is much reduced compared to when they were graduate students. So I'm going to walk you through an experience that I had here at UAB that required a substantial integration of different efforts, and everybody's effort was needed in order to make it work. In proteomics, we have more letters than we have in genetics. In genetics, you essentially have 4 or possibly 5 letters. In proteomics, we have 20 letters. The language is much more complicated. In order to analyze a particular protein, we use a specific tryptic peptide, which is representative of that protein. And it's very much like an oligonucleotide being selected for a particular gene or transcript. What we actually operationally measure is not words, but numbers. And so we take, if you look on the left side, or the right-hand side here, I don't know if you can see this, yes. So a peptide has a mass dependent on the amino acids that comprise it. We isolate that, but in the gas phase, we isolate this peptide, and then we cause it to fragment by heating it. And it generates a series of fragments based on that sequence, either coming from the N-terminal end or coming from the C-terminal end. And by combining this filter with this filter, we end up with what we call a mass transition. And it's numbers, it's not letters anymore. And numbers are even more diverse than letters are. In letters, in the alphabet we use is 26 letters. Numbers are basically infinite. So a big question for us when we're measuring a peptide is whether a particular mass transition is unique and truly representative. And when we started this, the mass spectrometers at the time actually had the ability to only recognize words as integers and not as the full definition of the mass, because mass is not defined by integers, it's always minor fragments of numbers. And there's a very big reason why this is, and it's the reason why you're here, but that's for another story. So the task in order to try and implement this was to download the entire proteome of a particular species from UniProt, then to digest that protein amino acid sequence into the rules that govern biochemistry to make tryptic peptides. We then needed to calculate the mass of each peptide, which was found doubly charged, and a particular, the human proteome gives you about 1 million of these. We need to calculate the mass of each tryptic peptide product, and there's about 8 million of those, make a two-dimensional plot of all precursor and product ions, and then start building interfaces and mouse over tools that can allow us to reveal the identity of each dot within that mass space, giving us peptide sequence, the protein it came from, and a link to UniProt. So it couldn't be done by one person. This is the team that set about doing this. I had the idea that this was a good thing to do because it was something I had to do when I was getting concerned that we weren't measuring the right things. I was very good friends with Chiquito Crasto, who's now down at Texas Tech University, and he was an assistant professor in genetics and an informaticist. And between us, we recruited Chaz Nan, who was a master's student in computing science. Now, Chaz had absolutely no idea about biochemistry, and therefore we had to sort of define what the rules of how you break a protein up into its pieces. And so he did the bulk of the work of getting this converted into a computational form. Mikako Kwai, whom many of you have probably encountered, was the website developer that turned this into a tool that we could use with our browser. And Jonas Almeida, who preceded the current bioinformaticists team, was a key person to generate our very first picture, which I'll show on the next slide. And so this is a mapping of all the peptides, and the X and the Y's represent the peptide ion and of the precursor, and then the fragment ion. And in total, there were nearly 4 million B ions and over 4 million Y ions that were in this plot. And what we did for each mass window, which was 1 Dalton wide, we asked how many peptides fit into that spot. That then allows us to determine how much degeneracy there was occurring. And this was a very massive plot, and Jonas was the one who enabled us to come up with this. And so it's color coded, and so where you see red, which you see in the limit of along here, it's when a tryptic peptide loses either lysine or arginine in its fragmentation. If you use that particular peptide ion as a fragment to measure the peptide, then you may have a degeneracy of over 100 different ions. So we were able to find out by practice where the sweet spots were in the peptide sequence, where we could get the best selectivity for an individual peptide ion. And so this is the website as it looked. We could set up a mass window. Let's say we were going to do an experiment and wanted to measure a particular peptide. We could put the mass range in that we know that will be the peptide mass. So in this case, we've gone from 705 up to 715. And then we created a window, and this could be in any other place that you wanted, but we've selected a 20 Dalton mass window for the fragment ion. The fragment ion can be bigger than the parent, because the parent is doubly charged and the fragment is singly charged. So we don't have to divide the molecular weight by 2 as we do for the precursor. So what do we get? Well, when we open up that mass window for the human proteome, we see that most of the ions are actually they're found at the interval of either 1 mass unit or half a mass unit. If they're doubly charged, they go at half mass. And you can see even at this resolution that there are multiple ions at each dot that you see in the chromatogram. There are also some empty spaces. I've shown those by these open circles. And these where in the proteome there isn't anything to be found. we can make the window expand out a little bit. So now we've got a 2 by 2 window, and we can see the individual ions separating out from each other. The red ions are Y ions, and the blue dots are the B ions. And we can take it even further. So now we're down to a 1/2 by 1/2 mass window. And I can't show you this in real time, but you could mouse over each dot and it would produce the mass of the ion. That's the precursor. This is the fragment ion. This is the sequence of the peptide. This is the protein, whether it has a name or doesn't have a name, where it's from. Of course, it's going to be human because we're in the human domain. And this is the link. And so by clicking on that, you can immediately go and see the full record for that particular protein. So this proved to be something that was of great interest to the community. And I was an invited speaker at our national meeting in 2012. And the chances of getting on that meeting is about the same as dying from the coronavirus. So a very low percentage of people who submit actually get in the meeting. So that was nice. People from the scientists followed up with me and had an article featuring on it. And I get quite a few people contacting me about whether the program is working. And sometimes we had to reboot the computer because that particular computer had gone down. And Macaco was the one that went on to develop the smartphone adapted version of this so that we could see this on our phones as well. So can you review MRM space now? Actually, you can't. It's gone. And this is just some of the emails that occurred about 12 months ago. So I noticed that we lost MRM path from our Temple website due to the transition to HTML5. Is it software recoverable? Macaco said that it's on the Tomcat server. The server is not connected to the internet. Landon, one of my staff, asked IT when it had a problem last fall. We haven't had a reply. Tomcat server is very old. I think many applications need to be upgraded. So we had a problem of transition. The people who built the program had left. I guess this is fairly classic. And we need to find a way to go forward because we regard this as a valuable program. So that's not the only thing that's going on. We have other projects that I'm very interested in and would like people to interact with me. I do work on metabolomics. Metabolomics is a lot harder than doing proteomics or genomics. There is no real database. You can't, in proteomics, we're able to convert DNA information into protein information. And so we had a protein database long before people ever actually studied the protein. So we got the sequence from the DNA databases. So when we do metabolomics at the moment, we have a mass spectrometer that separates out the different items and identifies ions, what we call ion features. They have retention time and they have mass. And so there's the output is of a formatted form that comes from the manufacturer, and we can view that with some of their software. But that's not what people want to do. The NIH wants us to go out and do deeper searches into the database. So we take this and submit these data over to XCMS Online, which is available, but it's sometimes rather slow, but it aligns peaks, gives us quantitative associations for each sample, for each identified feature, and then exports it as an Excel spreadsheet. I then process that, turn it into .csv files, do a statistical analysis on metaboanalysis, and then do pathway analysis. So there's a lot of changes of the basic collected data going through these different packages, and it would be very nice to produce a sort of an umbrella form of this, that whereby you could put your data in here and all of this would run, all of these different softwares would run automatically, and you would have an item at the end a lot faster than we currently have. I spend most of my time processing data through these different steps. There has been one attempt by colleagues. This is something called MS-Dial. Again, you take the files coming out of the mass spectrometer, and they can be converted to what we call .abf files. Takes about, on my computer, it takes about 20 seconds per file. You can't get the code from this. The code is held by the original developer, so you can't do that. But MS-Dial, you can run that on a laptop, but you can also get the code and run it on a much faster machine. And they have an automated mass spec library we use to identify things. We study the patterns of the fragments that we get, and there is library matching of this material to the database, which is not just gene products, but also includes drugs, drug metabolites, food intermediates, food additives, and all sorts of chemicals that you have. When we study people, we see sort of things that you use in your home to keep the bugs down, all sorts of compounds, chemicals that come out. They're all detected by the mass spectrometer, and some of this is relevant. It's very important to know, for instance, what a patient is really taking. If you ask anybody, clinician who's doing a study trying to find out how effective a particular drug is, that what they don't know is what the patient is really taking. Adherence is one problem, but the fact that they take other things, or their friends, spouse, whatever, it's a real problem. So we end up not being very precise in the medicine that we're currently using. Anyway, so to sum up, the bioinformatics support of translational science really, to my mind, falls into two features: the use of existing packages, and if they're in R, my experience is that the effectiveness is actually inversely related to the age of the trainee or to the faculty. The younger people pick it up much faster. I walked up the hill just before I came here with one of the people who attended my workshop, and even on the second time round of the training, he was swearing in very, very discreet terms. He was not happy because he was just getting frustrated. And so it's very important to make the training meet the level of the recipient, just like we train anybody else when we get away from being here in the university. And then to build new packages, it has to be a collaborative effort. All the different people have to bring their best science or their best capabilities to hack the problem. And then they should all respect each other, because everybody is contributing to the entire project. And what I find is that this is a wonderful place in which the students, either at the undergraduate level, the master's level, or even graduate students, can come in and learn a lot about what it takes to be a scientist. Because it's not just being you and trying to be better than everybody else. It's actually sharing your knowledge with other people and making it work. And my last point, and I learned this from a colleague in Cincinnati who's now a dean for core facilities, that what they realized was that there was a need for development of these types of capabilities to come from the core facilities program, and that people should apply to have small funding to try and get programs like this that would be of benefit to not only to the people here at UAB, but generally around the world. So finally, if you have any questions, I'd be happy to take them.
I think we are running out of time, Steve, and no one's hoping up any question. I think we need to move to next speaker, Dr. Molly, please, from School of Medicine, Department of Medicine. She will talk about the coronavirus update. We may be very nervous today because we have a case in Alabama. So, Steve, can you stop sharing your screen, and Molly, you can share your slides.
Okay. Let me see if it'll work.
Thank you, Dr. Bars. Thank you.
Not sure.
At the bottom of the screen, there is a share button.
Oh, I see it.
Yeah.
That work?
Yeah, we can see the slides.
Can you hear me?
Yes.
Okay, perfect. Hi, everyone. I'm Molly Fleece. I'm an assistant professor in infectious diseases at UAB. So I'm going to close out your day today talking about an update on the 2019 novel coronavirus that, if you have turned on a TV today, has been all over the news today and for the last 6, 8, almost 12 weeks now, with a slant towards epidemiology and informatics opportunities. And so I'm going to start by just briefly touching on 2 important coronaviruses in our history, talking about SARS and MERS, and then touching on COVID-19 or the virus SARS-CoV-2. And then what I would like to do is highlight what I think to be some great applications of informatics during this rapidly evolving pandemic. So coronaviruses circulate rather commonly in our community, and the ones that circulate frequently are the second most prevalent cause of the common cold. These are enveloped RNA viruses that are named for their corona or crown-like appearance of the virions. And as we know, every winter, that control of transmission of coronaviruses is quite difficult, and that is been a big topic in the news and a big topic in our scientific fields of the current situation of COVID-19. So real briefly on 2 very common or 2 other clinically significant coronaviruses, SARS-CoV or severe acute respiratory syndrome, and then I'll switch to MERS. So the timeline of SARS started in November of 2002, where an index patient was thought to be infected by a civet cat in the Guangdong province. And in this infographic, you can see this one starts in March of 2003 at point 1, where a physician who treated a patient with SARS in a teaching hospital unfortunately became infected and then spread to residents within a hotel at point 2. And you can see in this graphic, and the point I'm trying to make is just how quickly this virus spread, first through the hotel to other guests, and then how quickly everyone dispersed and spread the virus to their respective countries. By April of 2003, SARS had spread to every continent on the globe, including 29 countries. And SARS is a quite deadly virus with a case fatality rate of about 9.5%. The outbreak lasted from November of 2002 through July of 2003, involved almost 800 deaths and 8,000 infections around the world. On this map, gray indicates infection only and red indicates infections and deaths. And from the SARS worldwide epidemic, there are many public health changes and many public health infrastructures that were put into place that really helped to reduce the transmission and ultimately stop the spread of SARS. And many of these concepts are coming back up into play as we are facing this global pandemic. Toronto had the largest outbreak of SARS. This is looking at the epidemiologic curve of number of cases that were investigated and then the number of cases requiring quarantine, lasting about 3, almost 4 months during this map. And you can see in the gray bars that those were cases that met the SARS case definition. Blue were the ones that were investigated but did not. And then the yellow line are the number of contacts requiring quarantine. And you can see sort of the 2 peaks here on this map. During this time, hospitals in Toronto implemented many emergency plans to try and reduce spread in the healthcare system and exposure to healthcare workers. And I draw your attention to this because I think these are going to be many systems that we implement here in our country now to try and reduce the spread of SARS-CoV-2. Switching gears just a little bit to MERS. So MERS was first diagnosed in September of 2012, and it was isolated initially from the respiratory tract of a patient in Saudi Arabia. And this patient had a severe pneumonia as well as acute renal failure that developed in June of 2012. Since then, there have been about 2,500 cases around the world, about 900 deaths, And opposed to SARS, where I just told you the case fatality rate was high at 9.5%, the case fatality rate for MERS is 34%, so exceedingly high. And it was thought that this virus originated not from a civet cat, but either from camels or from bats, or potentially both. On this map, I am showing you tracings of how the virus spread from person to person. That was traced back through genetic sequences of the virus from isolates that were obtained in clinical specimens. And you can see the large red circle in Saudi Arabia, and because we live in a very international world, you can see how this virus was able to spread rather well throughout Europe, the Middle East, Northern Africa, and beyond. Again, to show you another epidemiologic curve, this is looking at the number of cases of MERS, and to show you that most of the cases of MERS have been isolated in Saudi Arabia, which are the dark blue bars, with a distinct red peak of the Republic of Korea in about 2015. All of the other countries have had relatively low numbers, thankfully. We still see cases of this in Saudi Arabia. Thankfully, case numbers every year are relatively low. Now to switch gears and talk about SARS-CoV-2 with a focus on epidemiology and informatics. So this is an article from Science talking about this deceptively simple number sparking coronavirus fears. And now this article is a little old, but I think it highlights the very important point of what a reproductive number is. And so the basic reproductive number, or the R0, is the rate of spread of an epidemic and whether that spread is self-sustaining. And it depends on the magnitude of this number R0. And this defines the average number of secondary cases generated by one primary case in a susceptible population. And so here I've shown you just a nice graphic of the number of people that one sick person can infect on average. And that's called the R0. And you can see where common viruses fall, like hepatitis C and HIV. Ebola, thankfully, had a relatively low R0. And that's opposed to measles, which is on your far right, where one infected person on average infects 18 people. And from the early studies in Wuhan and in China, and now ongoing around the world, the estimated R0 for SARS-CoV-2 is about 2.2. So to bring you back to the beginning, this is an epi curve from the New England Journal of Medicine, published this early this year, looking at the very beginning of this outbreak of the virus in Wuhan specifically, and in China. So you can see towards the beginning of December, there are a couple of cases. Most of them were linked to the Hunan market. And at the end of December, you can see more cases of pneumonia, where they started case finding activities. Around the turn of the new year, the seafood market closed, and that was to try and help reduce spread of this virus. And you can see as the number of cases rose, the emergency response escalated. By early January, it was defined, the virus was identified, and rather quickly was genetically sequenced. PCR reagents were sent to Wuhan, but unfortunately after that, cases quickly were identified outside of this local area. And since then, we can see how those early cases compare to the fairly exponential rise of cases that we have seen since that time, the beginning of January. And so on this, it's just to show you the sheer number of cases and to show you the distinction between the date of onset of symptoms and the date of diagnosis, and the onset of symptoms in blue and the date of diagnosis in yellow. At the end of January was when the WHO declared a public health emergency of international concern, and earlier this week, the WHO has now declared this a global pandemic. A little bit about the virus. This was as of February 11th. There are many cases, 72,000 identified in China. Most of them were confirmed, many of them were suspected. Age distribution tends to be in our older population, certainly greater than 30, with very few cases in people younger than 30. And spectrum of disease, about 80% of these infections are mild, with about 15% being severe and less than that being critical. Case fatality rate for SARS-CoV-2 is about 2.3%, with the highest mortality rate in those over the age of 80. And unfortunately, we have had significant healthcare workers infected, with several deaths in China and more around the world. Clinical course, just to talk a little bit about symptoms before I move on to some other informatics graphics. Most people have fever, cough, and shortness of breath, or dyspnea. What is important and what we talk about often in the news at this point is a significant number of people who require hospitalization, ICU admission, mechanical ventilation. It's not small. And you can see how long there has been circulating RNA in these patient samples. With a similar clinical course between survivors and non-survivors, other than the multi-organ system failure and the invasive ventilation in those who do not survive. And this is based off of patients in Wuhan. This is just to show the differences between SARS outbreak that I described earlier and what we saw early in this COVID-19 outbreak. And you can see how the number of cases is strikingly different. So what's happened since the early days in China? This is a graphic looking at the number of cases in China, daily new cases in red. And you can see how primarily early in this pandemic, the cases were centered in China. Since about the end of February, we have had a stark rise in number of daily new cases around the rest of the world. And there have been a few places where we have had a significant rise in infection, South Korea being one. This is showing the number of new cases and total number of cases in South Korea, and then the total number of deaths in the gray line graph at the bottom. Another place that's having a similar rise in cases is Italy, and I'll come back to that towards the end. So coming back to some informatics use that I think has been, wonderful, certainly as a clinician and as a researcher, to try and keep up with what is going on around the world, putting it, into a great graphic and something that can be updated in real time. You can see I pulled this morning at 6:33 in the morning. And on here, this is a infographic from Johns Hopkins University and the Center for System Science and Engineering, where they have been keeping track of the total number of confirmed cases around the world, total number of deaths, and total number that have recovered. The size of the circle corresponds to the number of patients, and you can see the significant number of patients in China, but increasingly larger red circles are popping up in other areas of the world, including the Middle East and then now Europe. And coming over to what is happening here locally from the same group at Hopkins, as of this morning, we had 1,700 cases here in the US, with certain areas where they're having more local or community spread, that being in Seattle, places in California, and also in New York. A different graphic, and I apologize for going fast, I just recognized that we were running short on time, but another infographic that I think is great. This is coming from the New York Times, and it's also being updated every day, trying to track number of cases here in the US. And I think this is a wonderful application of informatics and how it really helps during this global pandemic where things are changing every day. And as we know from 10 a.m. this morning, Alabama is no longer white on this map. We have had our first reported case in the state of Alabama. So updates soon to come. What I wanted to talk about quickly here is the transmission of the novel coronavirus and how genomic epidemiology has been wonderful in helping us map this virus. This is from a group out of Fred Hutch in Seattle, where they have described how this novel coronavirus mutates on average about 2 mutations per month. And after someone is exposed and the virus incubates for about 5 days before the patient develops symptoms. The virus is then usually able to be transmitted around day 7. So on average, 7 days from one infection to the next. And as the virus transmits, it mutates at about a rate of 2 mutations per month. So about every other step in this transmission chain, there is a mutation, and this is shown graphically. Now, through genetic sequencing, their lab was able to reconstruct which infections were connected to others, trying to help teach us about the epidemiology of this virus, as well as transmission in a rather novel way. They used a next strain software platform, which they also used for Ebola, Zika, and the flu. And they were able to look at genomes of 140 SARS-CoV-2 coronaviruses that were shed from all over the world, and they incorporated these genomes into a global map. So this is an up-to-date view as of this morning of the genetic epidemiology of this novel coronavirus, linking where the viral strains originated on the map, and then on this phylogenetic tree. Here is where they looked at the first case in the United States, USA WA1 2020, and this was from a traveler who returned directly from Wuhan to Snohomish County in Washington on January 15th. They were able to sequence this virus and share the genetic sequence within 5 days of sample collection, and then placed on the genetic map and found that it had an identical genome to a similar sample obtained from a person on 1/21, who was also a travel export from Wuhan, suggesting a close relationship between these two cases. Now, the second arrow on this graphic is looking at the first community spread case in Snohomish County. It was someone who sought treatment for flu-like symptoms, was tested for flu and sent home because they had a mild disease. Once COVID-19 was confirmed and sequenced, they were able to identify that the sequence was identical to the WA1 strain, except for 3 additional mutations, suggesting that that viral strain had been circulating in the community for about a 5-week period. They looked at the probability of random genetic variants, felt it was very low at about 3%. Just real briefly on case fatality rates versus confirmed cases in 20 places around the world. So you can see the United States in the middle of this infographic, China up at the top in red, and then Italy far over to the right, with rising number of cases and a relatively high case fatality rate at this point, which probably just has to do with the need for more surveillance to get that denominator data in better form. But I bring up Italy to show that as of yesterday, Washington State and the number of cases was tracking almost perfectly along with Lombardia, Italy, and this has caused quite the concern in the public health arena and in the infectious disease arena, both clinically and with research, for how can we slow the transmission of this virus in our community so that way we do not overwhelm our healthcare system. And as I'm sure all of you have seen in the news as of late, this push to try and lower and delay this epidemic peak. And what we're trying to do is reduce that uncontrolled transmission peak shown on your left through proactive measures like social distancing, limiting large gatherings, reducing travel, and more assertive approaches to where we do not have such a high peak over where our healthcare system has the capacity to manage such an influx in the number of patients, and whether that's ER visits, ICU beds, etc. So hopefully through our public health efforts, we in the United States will be able to flatten this curve. And with that, I will answer any questions, and just as a gentle reminder, everyone wash your hands.
Thank you, Molly. There is a question from Wei Liang in the chat box. Is there anything Informatics can do to help these coronavirus efforts in Alabama?
So in Alabama, you know, this is an evolving situation. Since we had our first case this morning, we are able to run more tests now. And so we will see how the situation in Alabama evolves. I think the best thing that we can do in Alabama right now are things like wash your hands, stay home from work if you're sick, try and avoid large gatherings, especially if you are sick as well. And pay very close attention to what's going on here locally. I think the situation will be changing, certainly day by day, potentially, hour by hour. So just stay up to date on what's going on here locally.
Molly, this is Steve Barnes. Can you hear me?
I can hear you.
Yeah. The president, when he made his speech the other night, was stopping people coming from Europe. And made the very interesting to me, almost embarrassing comment that, of course, the English would be allowed to continue to travel across the Atlantic. Do you have any sense? I've got some input, but I was curious, do you have any sense of how well they're doing?
On how well the English are doing? I'm sorry.
Yeah, so how are they doing it differently to what we're doing or other countries are doing that would allow the president to make a comment like that?
I am not sure if there's anything particularly different that they're doing. I'm very interested in your inside information, though.
Yeah, so my son is a lecturer at the Solent University of Southampton. And so he started there during last summer. And so I contacted him this morning to find out what was happening in his university compared to UAB. And so they have another week before they take their 2-week Easter break.
Okay.
And they're still having the students meet, despite the fact that I gather on proportion, the numbers in England are higher than here, if you look at the head of population. And so he ended up with contact with the student and got no instruction about what to do. So he's self-isolating.
Oh, goodness.
Yeah. So,
yeah.
So I'm rather concerned that we're having a mismatch of information. It sounds like UAB is approaching this somewhat professionally, and I hope that's the way you feel.
I do, and I'm helping certainly I'm the assistant healthcare epidemiologist here as well, and so myself and Rachel Lee, we have been working a lot with the hospital leadership and other leaders throughout this institution to try and ensure that we are as prepared as possible for what's going on. I think, we're seeing more and more universities around our country trying to move towards virtual classes after spring break, which is about to happen for almost everyone around our country right now. And I think it's a good idea. Hopefully by being very proactive, by reducing the number of students who are together, the number of large gatherings, the sports, NBA, basketball, baseball, and all of those events that are being canceled. My hope is that by being very proactive with all of that, we are going to be able to flatten that curve down and try and prevent what's happening in other parts of the world from happening here.
Yeah, so the coach of the Arsenal team in England, so one of the big clubs in England, was identified as having the virus. And finally, the Football League, with these big crowds meeting, multiple days a week, finally called off the season for 3 weeks. So it's rather like the NBA here.
Yeah.
But it took a case to actually force them to do it.
Right.
Whereas it was obvious they should have done it anyway. It's where money, unfortunately, trumps, sorry, I used that word, but trumps decision making.
Yeah, I think.
Sorry, we are running out of time. So thank you so much for all of the speakers and the audience. And we will have a poster award. Jim is here. He will announce awardees for the poster, and Jim will make the final remarks.
Okay, thanks, Zechen. So before I do the poster awards, since people will still be listening to me for a few minutes, I want to say thank you to the staff and faculty for pulling this together in 48 hours. We made a decision on Tuesday or Wednesday, really, the university said no meetings unless they're absolutely essential, and I couldn't think of the reason why this essentially had to be in person. So we really pulled it together. Special thanks to Zechen, Alex, Jake, Elliot, Amy, If I left somebody out, for running the various sessions, it was a huge help. And for Amy for running the poster session. So with that, let me announce the third place winner with a prize of $100. Maggie Chen, Inspector, a tool for inspecting long-read de novo assembly results. Second place for a prize of $200, Samantha Carlisle, sex-based differences in the activation of peripheral blood monocytes in early Parkinson's disease. And first place with a prize of $300, Chris Taylor, comparing 16S sequencing and shotgun metagenomics to vaginal bacterial communities leading up to incident bacterial vaginosis. So congratulations to all the poster winners, and also thank you to the poster presenters for pulling that together so quickly. And with that, we'll turn it over to Jake, who is a remote. And
yeah, he's driving.
Yes.
Here he is.
Okay, can you hear me?
Yeah.
All right, great. Well, I also echo Jim in acknowledging everyone in the organizing committee and putting together a wonderful program. And especially, I think we are probably very creative in the country in the sense that that everyone can enjoy a great conference in the comfort of their own chair or bed, whoever. We don't know. So I think that many of you noticed that we placed the last talk, but the most important topic of this month on coronavirus last. And I want to emphasize that that informatics is not only about solving basic science problem, but also about solving. Okay. Okay, apologize. Okay, so informatics is not just about solving basic science problem, but also about solving emerging translational problem of a great society concern. And this is an opportunity where the nation is called for informaticians to look at the epidemiology data, look at genomics data, to look at everything that we know and maybe working together. So the concluding remark, and I want to actually present people an opportunity to have another virtual 1-hour meeting next week. We're planning that meeting. We want to bring together people from this community, whoever is interested, to come and talk about a Ubright research portal that Informatics Institute is planning to lead. So this research portal will be populated with all of the coronavirus genome sequences and will also be placed and then we'll also be placing these genomic sequences on the Ubright platform with all the tutorials. So we want to actually brainstorm what can we do as informaticians to make this resource available to every UAB translational researchers that may have an interest in developing diagnostic tests, in studying the virus evolution perspective, or maybe in studying the potential therapeutics. So the meeting is likely going to take the place of next Friday from 9 to 10, but we're actually trying to make sure that everyone can attend virtually. So, so expect to hear that from Amanda. And that's all I have to say. Thank you.
Okay, well that will close our 4th annual ATTIS conference. And thanks to everybody for your participation.
Okay. Thank you everybody. Have a nice weekend and take care.
Stay alone.
Yeah, thanks everyone.
Nice weekend, everyone.